Good morning, everyone. We're going to go ahead and get started, so if I could ask you to please start taking your seats. That's fine. Okay, good morning. I'm Greg Daniel. I'm Deputy Center Director at the Duke Margolis Center for Health Policy. Welcome this morning to our public workshop, Understanding the Development Challenges Associated with Emerging Non-Traditional Antibiotics, which we at the Duke Margolis Center are convening under a cooperative agreement with the FDA. As you all know, antimicrobial resistance is a serious and growing public health threat. As many of you have likely seen, earlier this week, the FDA proposed some new approaches to address some of the clinical and economic challenges. They released a draft guidance on a limited population pathway for antibacterial and antifungal drug development, or LPAD, which allows for streamlined clinical trials for patients with serious and life-threatening infections and who have unmet medical needs. In Commissioner Gottlieb's press release, he also proposed working with the Centers for Medicare and Medicaid Services to develop a licensing agreement for antimicrobials, which would alleviate the need for volume sales. While these are exciting developments that have the potential to support all types of antibacterial antibiotic development, today we'll be focusing specifically on the scientific, clinical, and regulatory challenges that non-traditional antibiotic developers are facing. Traditional approaches to antibiotic discovery and development do not encompass all possible strategies to treat or prevent resistant bacterial infections. Traditional approaches are typically thought to be small molecule compounds that have a direct bactericidal effect. Today we'll be discussing what we're calling non-traditional antibiotics, which don't have all of those characteristics, including products that may be narrow spectrum and may be designed to be used in combination with current antibacterial therapy. Non-traditional antibiotics include antibodies, bacteriophage products, antivirulence therapies, immunomodulatory agents, microbiome approaches, and many others. We'll not be discussing vaccines in this discussion today, as those types of measures are being addressed through other avenues. Our workshop today is intended to bring together stakeholders to share information on non-traditional antibiotic research and clinical development, with a particular focus on potential developmental and regulatory challenges. Speakers and panelists will discuss a variety of non-traditional antibiotic products, including products that we'll be covering in session 1, which have the potential to be used as monotherapies. Products in session 2 that we'll be discussing are designed to restore activity to existing antibiotics. Session 3 will cover products designed to enhance the elimination of bacteria when used in combination with existing antibiotics. And session 4 will be covering products designed to prevent infections. At the end of the day, we'll hope to identify areas that need additional focus and to consider next steps to better support these important therapies. We're greatly looking forward to the discussion today. Just as a quick overview of the agenda that I just went through, for each of those 4 sessions, we plan to have a couple of presentations from companies that are actually working in the field and developing non-traditional antibiotics, where we'll hear some of the scientific and evidence development challenges that we might be having. Then, for each session, we'll have a bigger panel to engage with the company and discuss those challenges. And then at the end of each session, we'll leave room for audience Q&A. Before we get started, just a few additional housekeeping notes. Wi-Fi information is on the registration tables in the foyer outside of the meeting space. The restrooms are located outside of the ballroom. We'll have some time in each session discussion with audience participants, as I've mentioned, and we'll be live tweeting this meeting at using our Duke Margolis handle at Duke Margolis, using hashtag AMR and hashtag antibiotics. For those of you joining our webcast audience, welcome. We encourage you to participate in the day's discussion, and you can do that by emailing your questions for the panel or speakers. Please send them to duke.abx@duke.edu, and we'll try to address as those come up. I want to remind everybody that this is a public meeting and the event is being broadcast online. So everything you say will be part of the record for this event. For those of you in the room today, feel free to help yourself to coffee and beverages right outside the room. And lunch today will be on your own, but you can bring food back into this room as we get kicked off for the afternoon. Finally, a reminder that although this meeting is being convened under a cooperative agreement with the FDA, it's not a federal advisory committee. We will not be taking a vote. The meeting will be a success if there's an exchange of information and open and active discussion among all of us today. So we'll begin with opening remarks from FDA. Dr. Ed Cox is Director of the Office of Antimicrobial Products at the Center for Drug Evaluation and Research, and Dr. Cox will provide further context of today's discussion and FDA's perspective on the goals for today's meeting. Ed?
Great. Thank you, Greg, and thank you, everybody, for joining us here today for our workshop on emerging non-traditional antibacterial agents. We're here today, but I think I just want to start out by recognizing all the work that went into getting us here today. All the folks on Greg's team at Duke-Margolis and John and Kevin's tireless energy for putting this all together. And also, too, the companies and the panelists, the speakers that will be joining us here today. And if you think about it, today's a little bit unique in some ways because usually companies don't talk too much about programs that are in development. If you think about it, that's usually the time when there's a lot of confidentiality issues that make people afraid to talk about things. But we really think it's great that companies are here willing to talk about some of their programs, talk about where things are currently in development, because this is, I think, a critical part of sort of seeing what's out there and thinking about what's down the road, how to develop such products. So we're thankful for that folks are willing to come and talk with us about the different types of products that they have, where they are in development. If we think about today's meeting and sort of what we're hoping to get out of it, this is really our, one of our early opportunities, if you will, to talk about non-traditional therapies. And I think if we can get an inventory and start to think about what are the range of different products that are out there, that'll be particularly helpful to the field. We may find that certain types of products, in essence, can be grouped together. That may be helpful as we start to think about what's up the road. How will such products be developed? How will such products be studied? So I think today is sort of a first attempt to start to do that. I oftentimes joke around and say, don't ever take development advice from somebody wearing a tie or standing at a podium. There's a lot of particulars about development programs that can make them different, make them unique, and other things that you need to think about. So think about today as sort of a contextual discussion of how one might approach development. If your product is a little bit different in some ways, there's some unique aspects. By all means, don't be afraid by today's discussion because there's always particulars that need to be thought about as we think about a drug development program. So if you are developing a product, today is a good chance to listen, to contribute to the discussions, to exchange, and then I would expect most would go back and think about, what have I heard today that impacts on my development program? Do your best to put your best development program together, how you propose to study your agent, how you propose to show that it provides clinical benefit to patients. And then, when you've done that, then I'd say, you're ready, come in and talk with us at the FDA. We're very interested, both in CDER and CBER, to have discussions with folks about development pathways, particularly for challenging products. And if we think about non-traditional therapies, obviously this is an area where one may be taking on greater risk, a new treatment paradigm, something that hasn't done before, so there may not be a particular model to follow here. But that greater risk could lead to greater benefit. So rather than just taking a step forward, it may enable us to take a leap forward. We see the challenges that we face with regards to antimicrobial resistance, patients who have few treatment options, outcomes from serious infections that we hope could be improved. And looking at different treatment modalities, different treatment approaches may be the recipe to try and get to success and to improve patient outcomes and have new options for patients with resistant infections. And I think that's about what I wanted to cover. I do greatly appreciate all the folks that have joined us here today, all the folks that are joining us here online, and we look forward to a fruitful day, good discussions, and we see this as really sort of an early step. I expect there'll be more opportunities for further discussion, both in public fora and then also, for individual companies with folks at CDER and CBER through discussions that are development programs. So we won't answer all the questions today. We do hope to get to this inventory. We do hope to get to this first blush of what these different development programs might look like, how things might be grouped, and then use that as a foundation for further public discussions and further discussions with companies developing this area. So thank you for your attention, and I'll turn it back to Greg.
Great, thanks, Ed. Okay, so to give us an introduction to what we mean by non-traditional antibiotics, we have two opening speakers. Kevin Outerson is Executive Director and Principal Investigator at CARB-X and Professor and Neil Pike Scholar in Health and Disability Law at the Boston University School of Law, as well as Dr. John Rex, Chief Medical Officer at F2G Ltd., Expert in Residence at the Wellcome Trust, and Operating Partner at Advent Life Sciences. Kevin and John will deliver a scene-setting message, and their remarks are meant to provide an intellectual framework for the conversation that we'll be having throughout the day. How to approach talking about non-traditional antibiotics and what they are. Why novel antibiotics must showcase distinctive value and some strengths and weaknesses within different categories of non-traditional antibiotics. Without further delay, Kevin and John, thank you.
All right, so thanks for that introduction. I'm grateful for this opportunity for us to have this conversation well before the moment in which a company needs to approach the FDA for these decisions. We're going to talk about some hard things today, some hard science issues, and I just wanted the room to understand, and the people watching as well, the perspective, what John and I are really trying to achieve here. We want to see these things succeed. We want to see these clinical trials designed and successful. For CARB-X's perspective, we're investing a lot of money in companies that have non-traditional programs. And so the goal today is not to have an optimistic day in which we ignore the problems in front of these clinical development, but actually that we confront them head on and try to find optimistic solutions to move forward.
So with that context, let's talk about the core problem here. Next slide. Actually, I should advance the slides. Great. I've been thinking about this problem for 20 years, and so I'm delighted that we're here today to talk about it. And the deep problem is that everything has to show its distinctive value. This is not a regulatory issue. It's what you ask of anything. Prove to me that it works. How is it better? When is it better? And for antibiotics, it turns out that the limits on routinely possible studies create a substantial hurdle that we're going to go through in detail. And here I just want to share the quote that I like from William Swanson, the CEO of Raytheon. Beg for the bad news. If you're not clear on these problems, you're heading into a world of hurt. So, as context, Trial Design 101. Every trial ever done has one of these two things at its heart. It's either a superiority design, where you're trying to show that X is better than Y, and that's actually what you'd always like to do. It's unambiguous. Everybody likes the idea of better. Or it's this other thing called non-inferiority, where you're trying to show that X and Y are not too different from each other. This is messy, harder to do, confusing. But we're almost always going to be using NI studies because of the paradox of antibiotics. We want new drugs for bad bugs, and the advantage of a new drug is easily shown in the lab on the basis of MIC testing or animal models. But asking for clinical data leads to a problem. Consider a limb-threatening infection due to MRSA. It's not ethical for me to randomize you to methicillin, which I don't believe will work, versus new. Instead, it's got to be something like vancomycin versus new, because vancomycin works. In short, I can't put you in that trial if I believe that either arm is a bad idea for you. Hence, antibiotic trials are typically designed to avoid showing superiority. Now, this thing about superiority is very, very, very hard. Because we do it in other places, like, migraine. I can easily do a delayed effective therapy study. Even in cancer, where I can't do placebo, but you can show incremental improvements, because there's always room to improve on 5 and 10 year survival. But infections are different. We routinely cure a potentially fatal illness, and it's hard to get better than cured. Still, the idea of non-inferiority is confusing. I want a better drug. I get it. But insisting on clinical superiority before approving new agents means progress only when or if the pipeline is again inadequate for the studied population. I'm going to poke at this two other ways. One way to think of it is that superiority means in infection that something bad has happened. And here our case study is plazomicin, a recently studied novel aminoglycoside versus carbapenem-resistant Enterobacteriaceae. So in 2012-13, when this product was getting into its pivotal trials, colistin was the only serious alternative for CRE. And at that time, you could really design a study of plazomicin versus colistin-based therapy and say, we actually think plazomicin ought to do better. And so they started that study, and indeed, the study is shown on the right, where you can see that looking at mortality on the y-axis, you were more likely to die in the colistin arm. So plazomicin won, but now look at the denominators. 20 in one arm, 17 in the other. They really couldn't enroll this study at a reasonable pace, and it ended up costing nearly $1 million per studied case. And 40% mortality is, of course, not good. So going forward, future studies won't use colistin as its base. You're going to use plazomicin, or there are several other drugs that now have data like plazomicin, drugs that actually have a much better outcome than colistin does. So that's the first lesson. But this superiority thing, just try it one more way. We do it in these other settings. Why not infection? Well, it's really this thing about durable cure being routine. And, you know, you can't do placebo in cancer, but you can improve. There's also, but the reason we have to solve this problem, the reason we're here today, is that they're talking about transmissible agents and transmissible resistance. We actually have to solve this so that we can develop agents for tomorrow. So the notion that we have to find ways around this sort of runs deep, but we actually have to find strong ways around this. And part of the answer is what I think of as the emerging two-study path for more traditional antibiotics. You do one standard trial versus a good comparator in a usual drug resistance setting where both agents work, get a nice data set, and then you do a salvage trial. And that's what plazomicin ended up doing. They added a complicated UTI trial to the study that I just showed you. It makes perfectly good sense. So now, language to guide conversation. We're getting closer. So what is a non-traditional? We're going to differ a little bit from prior papers and say that mechanism or chemical structure doesn't help. What matters is what it does, or really what it doesn't do. And for purposes of today, we're going to define an idea called a Fleming versus a non-Fleming antibiotic. Fleming is like penicillin. It has the spectrum for a defined syndrome and the speed required to be useful as sole therapy. And if it doesn't meet all four of those S's, if you like acronyms, it's non-Fleming, it's non-traditional. It kind of doesn't matter what it is at a molecular level, but it doesn't fit into our classic paradigms. Some other language briefly to comment on alternatives to antibiotics. I have no idea what this means. It could mean anything and everything. So we're mostly going to treat it as meaning non-traditional. Ditto, potentiator, or enhancer. These terms are very ambiguous. You may want to use them, and I'm not going to argue with you if you want to define them and use them, but we're going to largely avoid them in this talk. So back to the mainstream. If you hit all four of those S's, the great thing about that is there's at least one setting where you can enroll empirically into a standard study of new versus a standard comparator. And this is a predictable path to registration. You can run those studies in a non-geologic time. You know, it's very straightforward. For prevention, you probably have to just hit the first and the last S, spectrum and being the only agent required. But there's some other issues about prevention we're going to come to in a bit. I mentioned the lesser problem of the MIC. We're very used to using MICs as a way to calibrate our anti-infectives. Some products don't have an MIC. I think it's a problem we can manage. There are other ways to look at affinity of a product for its target. We don't require it for other drug classes. It may mean that your PK/PD support's a little different, but I'm sure we could work it out. There are also some other benefits that sometimes get mentioned for non-traditionals, like it's narrow. Less pressure on antibacterial. It works via the host, not going to get resistance, has fewer side effects. Some of that stuff may be true, but it's actually relatively hard to show those things, like less development of resistance. Carriage of resistant bacteria is imperceptible. Who among us got up this morning and said, oh, my ESBLs are just killing me, they hurt so badly? Now, you don't do that. Trial endpoints have to be based on things you can actually feel. And safer, you know, AE rates are pretty low with most modern agents, and it's hard to be convincing in terms of superiority on safety. It's possible, but it's just kind of hard. Will diagnostics fix any of this? Unfortunately, they're not the answer you might like. There are two issues. First one is that diagnostics don't create the cases. If a rare bacteria is seen in 1% of people, you still have to screen 100 to find the one. And with infection, time is ticking and referral is not a path. In cancer and rare diseases, we don't dawdle, but there is enough time to make a clear diagnosis and refer as needed. With infection, minutes, and I do mean minutes, count. The patient typically must present at a site where the study is already running. So this magnifies the problem of finding those rare cases. We will look for ways to use diagnostics, but they're not the fix I wish they were. We're also going to skip some product-specific issues like immune response and delivery of the product. If you had a good product, you'd figure these things out. So now, non-traditional products to treat. So we're going to introduce an idea that we call the 4 archetypes that go with the acronym STAR: standalone, transform, augment, and restore. And the thing about these archetypes is that in each one of these buckets, you have the same development challenge, and that's what makes them useful. So let's first talk about standalone and transform. So standalone is what it sounds like. It's something that is meant to be a monotherapy for its target, whereas transform is something where you take a known drug, like a gram-positive agent, and you add something to it so that now it does something really different, like it treats gram-negatives. And that idea has been discussed, the idea of adding a polymyxin-like compound to a known gram-positive agent to create a gram-negative therapy. So you transform. In either case, you have an entity that is complete in and of itself. Even if it has more than one component, it's still a thing that you can use as one unit. It usually has an MIC, and the advantage is that standard NI designs might be suitable. But if it's narrow spectrum or not fully standalone, you hit some issues. Narrow spectrum is worth a couple of slides. There are 4 patterns that I can figure out. Pattern A is the easy one. The organism is the syndrome, gonorrhea. That's actually very easy to study. Pattern B, the organism appears within a syndrome, and, helpfully, there's another agent out there in the universe that has a hole in its spectrum that is just the right size for your compound. There's a perfect fit. And a good example of that is ertapenem, which is a carbapenem that covers all the Enterobacteriaceae but misses out Pseudomonas. You put a new drug for Pseudomonas with ertapenem, and you now have the complete activity of an anti-pseudomonal carbapenem like imipenem. So when you see activity against Pseudomonas, it's due to new. What's left in is the low rate of Pseudomonas infections. Here's where a diagnostic might help you pick out people to enroll. Finally, the last 2 areas get a little even deeper into this. The organism appears inside a syndrome. There may be multiple organisms in parallel, and there's no agent that has the gap that you need in its coverage. And so this pattern then just divides up into, well, normal commensal versus pathogen. Once a commensal like E. coli, that's tricky. Every one of us would be PCR positive for E. coli. So you actually have to find a setting where you really believe it is mono-infection due to E. coli. It may be uncomplicated UTI you might be able to do that with a non-Star Trek diagnostic. And finally, there's always a pathogen. That gets a little easier. Salmonella, you're not supposed to be carrying salmonella. It might be a sweet spot for a rapid diagnostic if you wanted to focus on that pathogen. Finally, not fully standalone. The problem we're going to come back to again in a minute, but for one of several possible reasons, the new alone is not sufficiently active to be monotherapy. You don't get equipoise on new versus old. Instead, you really feel like you got to do new plus old versus old. And new plus old is going to have to be better than old on clinical endpoints for anybody to be interested in what you've done. And the classic mantra here is something that the patient feels or something that you can see in how the patient functions or survival. This problem will come back again in a minute in the augment category. So restore is our 3rd category. And this one we actually have a lot of good examples of where you restore an existing agent. So the best example is the beta-lactamase inhibitors that you add to an existing beta-lactam. Beta-lactam has worked in the past. Resistance mechanisms now block it. With the BLI, the MIC moves back from infinity to a normal range. And the advantage here is this is very clear and easy to develop. Prior history of the base product gives great comfort. Probably can do PK/PD easily. In short, this is actually very close to all 4 S's. And I should point out that these 4 categories actually really apply to whether it's a classic small molecule or not in many ways. Some of these ideas, as we develop them, you really can use them in both directions. The distinctive hurdles, PK's got to match. It's true of all partners. And there might be a narrow spectrum problem if you're chasing a rare bacteria, but otherwise pretty easy. Finally, the category that is the most characteristically non-traditional, augment. something like a virulence inhibitor, where the idea is that what you developed lacks an MIC, has no discernible effect in vitro on the base therapy in the laboratory, but it does something that you think will be helpful in the midst of the infection. Another example would be an antitoxin, which you put with a Fleming antibiotic. Distinctive hurdles here. First, the base therapy has to work. You might have an add-on that protected against the development of resistance, but it doesn't solve existing resistance problems. Second, the lack of an MIC, we've already talked about that. You can probably figure that way around that. Superiority, though, returns. You are going to have to show that new plus old is better than old, and you may need a novel endpoint. Let me expand on this on the next slide. Ultimately, AUGMENT forces you into a study of the form new plus standard of care versus standard of care. And we're going to want to see that new plus standard of care is better than standard of care. Are there settings where you might envision this with a sort of a classical infection? Maybe. Endocarditis is one good candidate. More rapid bloodstream clearance might have a measurable clinical benefit. Last night, Mark Gitzinger maybe think about the notion of duration of therapy. You might be, TB, a disease with a very long duration of therapy, maybe shortening that would be a benefit you'd be interested in. But these are, very specific examples that don't generalize as broadly as you might like. Something that we've been debating is whether or not different endpoints might be useful. And it's a challenging question. Whatever you propose has to be compelling. And I've mentioned the idea of feels, functions, and survives at the individual level. Is there a population level equivalent of that we should be beginning to recognize? I'm not really sure what that is yet, but it's worth thinking about something where maybe preventing resistance is something that benefits your neighbor but not you, but maybe is worth investing in. And finally, I say again, this is not a regulatory issue per se. The agencies are simply the first to point out the issue to you. Why should I use this? Why should I pay for it? So if you compare the archetypes, standalone and AUGMENT, this is where the greatest novelty tends to occur, but they're often narrow. They may need superiority studies, and they may lack an MIC. Transform and restore, typically one known component, usually has an MIC, may face a narrow spectrum problem. Preventing infections. This is something that is harder than I thought. Lots of folks want to eliminate the carriage of something as a way to prevent infection. It's true, if I'm not carrying Staph aureus, I can't get infected with it. But you actually do, it's not just reducing carriage, you really have to show the benefits. You have to show a clinical consequence on top of best available prevention. This turns out to be frustratingly hard, and the effect size must be interesting. The number needed to treat must be reasonable. And you also have the question of what replaces the displaced bacteria. Getting rid of VRE and replacing it with Candida might not be such a good thing. So very helpful case study recently at advisory committee. Pfizer Staph aureus vaccine back in November. Pre-surgical prophylaxis for Staph aureus. Very logical concept. And the two questions at this meeting were, how big does the study have to be to show a reduction in a serious infection, and where can you do it? And Pfizer has set out and is doing a phase 3 study in the population with the highest rate of surgical infection they could identify. Open, posterior approach, multi-level, instrumented, spinal fusion, orthopedic surgery. Read that carefully. You've got a bad back, we cut you open top to bottom, put in a big rod, and close you up. With best care, that still has an infection rate of 1.4%. It's a big surgery. And so Pfizer was able to say that, well, if we studied 2,600 people, a 1-to-1, we could detect a 70% reduction in infection, 1.4 to 0.42%. And the question of the AdCom wasn't about this trial. It was that if we do this, rather, now that we've done it in the back, what about hips and knees? Because it's the same pathophysiology. And the FDA briefing book says it the best. As rates of invasive Staph aureus disease across other elective surgical populations are 0.25 to 0.5% within 90 days, conducting a randomized placebo-controlled clinical endpoint efficacy trial that includes other orthopedic surgical populations would be operationally impractical. Yeah. My math suggests you're going to need 10 to 20,000 per arm in this trial. And think about what it would look like to reduce the rate from 0.25 to 0.125. That's a number needed to treat of 800. What do you do in other things like influenza? It's 10 to 40. human papillomavirus vaccine and cervical cancer, 300, 400. Yeah, it's on the edge. It might be acceptable. But put it all together, it's just harder than you might imagine. So the perspective then is Fleming-style things hits all 4 S's. We know how to develop these, and we can apply those ideas to some of the non-traditionals. But outside this zone, when you get into things in the buckets, particularly augment, but also all 4 of them can get you there. Standalone and augment are hard. Restore and transform are a little bit easier, but that doesn't make them easy. And prevention can be surprisingly difficult. And let me turn it back over to Kevin.
One of the things I do at Boston University is teach FDA law, and this is really a science problem. It's not a regulatory issue with the FDA. And again, we're trying to emphasize that we're trying to make things work in this sector. I know that this presentation can be something of a reality check or a downer in sometimes. So for CARB-X, which the organization that I am the executive director of, we're investing a significant sum of money over the next 5 years. We're focusing on priority drug-resistant bacteria. We're agnostic on what sort of approach that one takes. You see our impact, our goals here. And then this is a snapshot of the types of companies that we've invested in our first 2 years of operations. Right now, we have 2 rounds that we opened in 2018. Over 400 applications in those 2 rounds. This doesn't reflect any of those applications. This is just the older material. But note that we have some companies that fit into a very traditional paradigm. It's a known class, known mechanism. But the non-traditionals, the things we're talking about today, are represented in that box in the upper right quadrant. 8 in this diagram that are non-traditional, new mechanism, new class. It's the riskiest, but also the most innovative. And so from our perspective, we want to bring these forward, and we also want there to be a successful regulatory pathway for the clinical development of these products. And so why are we here? Why is CARB-X here? We're trying to support the ecosystem. I'm not here on behalf of any particular company. We don't represent the companies. We're non-dilutive, nonprofit. We make a grant to these companies. But we want the ecosystem to work. We're trying to restore the entire field and to make some advances. And so we're grateful that FDA and Duke-Margolis has supported this workshop and that we can have these conversations. It's also, in talking with Ed and others at the FDA, it's really helpful for them to have non-hypotheticals, right? To meet with actual companies, with actual programs. And if this was a more traditional approach, you would already know what the path is. It's helpful to have this discussion a couple of years earlier with real live examples and companies that are actually making progress. And so we're funding them and some that we'll be funding in the next round, and we hope to have an open dialogue with the FDA with that. We also want the companies to have realistic pictures, because it doesn't do me any good at CARB-X to fund a company through preclinical and phase 1, and then for them to run into an unexpected roadblock on their phase 2 trial design. And so that's also my motivation, our motivation for CARB-X in doing this. So a couple of things to think about. And these are just examples. They're just examples that John and I came up with in discussing it. It doesn't reflect, it's just the beginning of some free thinking in this area. So John already mentioned population-level clinical benefits. So imagine that you had a trial in which you could show non-inferiority against usual drug resistance, right? But then you needed to show, as one would typically do in a non-inferiority trial, something else that makes you want to use this agent. What if you could show clinically relevant, changes in resistance, or clinically relevant changes in carriage, or something else, bending the curve of resistance, doing some other population-level benefit, in addition to the non-inferiority trial against usual drug resistant? Just throwing that out there. Look at what was used for the approval of HPV vaccines, right? It would have been impossible, perhaps, to design a trial in which what you're trying to show is reduction in mortality from cervical cancer from the HPV vaccine. The latency is measured in decades. So the pivotal trials published in the New England Journal of Medicine and approved by the FDA, instead showed other things, intermediate, precancerous lesions, which we know based on the pathology would lead in a certain number of cases to full-blown cervical cancer and mortality. In addition, a lot of data out now showing the changes in the serotypes, showing that actually in the bodies of vaccinated men and women, a reduction in the covered serotypes, that were covered in the vaccine. So reduction in carriage and intermediate endpoint, not 20-year mortality, but still enough for approval of a very successful vaccine. The other bullet point there is just to begin to have the discussion about human challenge models. If it's difficult to find, in prevention or in therapeutics, there's a reference there, a recent article in Lancet Infectious Disease, which lays out, the number of areas in which human challenge models are being used today. And it's just to put that there as a potential. It's not an exhaustive list. This is the sort of thing that we're hopeful that today generates creative thinking in this area. But I couldn't go very long without having a little more bad news. And this is for us to think also about payers at the end of the day. FDA approval is not the end of the road. You actually want to sell your product. Recently approved products in antibacterials have not sold well. So you want a trial that is appealing to CDER and CBER, but you also need a trial that actually the clinicians are going to be impressed with and they're going to use, and that the payers will be impressed with, especially if you want a price that's higher than generic vancomycin or generic colistin. And we have to do that in a way that also preserves stewardship. Stewardship, will reduce the unit sales of your antibiotic. That's a good thing for public health. We have to take that into account as well. And so pull incentives, because I can't give a talk without telling people we need pull incentives. Pull incentives are a potential way to solve some of these problems simultaneously. And here's the unique thing about non-traditional antibiotics and pull incentives is that we'll reemphasize this. If there is one core problem for this area is that we're designing trials today for tomorrow's patients. We have a small number today, but we're worried about the larger number of these patients tomorrow. For other diseases like cancer, or cardiovascular or depression or whatever, if you talk to the companies and the analysts, they know what the market size is. They have a very good idea in 2 years and 5 years and 6 years how many people will need treatment for a certain type of cancer. And it changes, but it doesn't change dramatically. For us, we're faced with an uncertain path for bacterial evolution. We don't know what the epi future looks like. There's all sorts of efforts by the CDC and others to reduce the rate of infection. And then there's the wild card of you don't know what will happen with bacterial evolution. This is an additional barrier for antibiotic companies, especially those seeking innovative therapy, is that we don't actually know what the market size is. You're asking a company when they start a development program to guess, to predict whether or not CRE is going to be a huge problem in 10 or 15 years, right? And we need to develop that program now instead of waiting 15 years because we don't want to have 15 years in which we have no available therapy. This is an insurance policy. This is something that a pull incentive can address some of those issues in terms of payer side, not necessarily address all the issues on the regulatory approval side. Okay. And you see my suggestion at the bottom with regard to pull incentives. We have some supplemental slides. The slides are available, I believe, and some of the literature that describes some of these issues that we've talked about today, these are useful references. And John, we had some more slides in on animal health we had to take out as a result of time pressure. But just to make it clear that the animal health side that are doing some interesting things in this area that are relevant for you to read up these references and to get a sense of some ideas that might transfer over. This is our final slide. It kind of summarizes, John's presentation, giving you a sense of our views on the subject. We hope to have great questions and have great discussion at this workshop. And we're grateful for the opportunity to do it. So, I don't know if we have time for Q&A or if we're done.
Okay, yeah, we can spend, if there are any quick questions, we are going to move on to our next session, but we do have a couple of minutes here, so I ask Ed, John, and Kevin to stay up here for just a few minutes. Any questions from the audience? There are microphones in each of the aisles. Okay, yeah.
Is there a desire in
The short answer is yes. We want to spread across that spectrum, deliberately so.
Okay, go ahead.
Would these slides be available to us?
Yeah, we're gonna, so the whole video of today is gonna be available online, but also we'll have a separate file for the slides. Yes. So it'll be on the Duke Margolis website. Yeah, okay, go ahead.
Hi, I'm Dave Mantis from Arsanis. This question is really for Ed. You had mentioned that we should come to both CEDAR and CBER, and I'm kind of curious because I do hear, at least amongst companies, that they get different feedback from CEDAR and CBER, particularly on things like definition of endpoints. We know that the application of, for example, the combination product guidance is a little different. We use the HPV example, right? That's a 9-valent set of antigens, but certainly they didn't do a 9-arm or a 10-arm phase 3, or 12-arm phase 3. So I'm curious as to whether CEDAR and CBER are trying to work together so that their application and interpretation of these things are consistent.
Yeah. No, thanks, Dave. Yeah, I mean, we are trying to work together and preparing for the workshop, and we expect we'll have greater opportunities to overlap in the future. There can be reasons why people get different advice. There may be scientific differences that lead to different approaches, or that endpoints are different. But certainly in those areas where there's good reason that things should overlap or be consistent, we'll work with our CBER colleagues closely. And I think we've learned already from working with each other, and I expect we'll continue to learn in the future. So thanks for the question, Dave.
Okay. Okay, great. So thanks to Ed, Kevin, and John for all of your comments. Okay, that takes us into session 1, developing non-traditional antibiotics with the potential to be studied in clinical trials as a monotherapy. During our first session, we'll hear from presentations from companies that are developing these kinds of therapies that have the potential to be studied as monotherapy. As I mentioned before, each presenter will come up and present, and then after all of the presentations, we'll have our panel discussion. So we'll go in order of the presentation. So our first presentation will be from Greg Merrill, CEO at Adaptive Phage Therapeutics. Greg, can you join me up here? Great.
Okay. All right. Well, thank you. And thanks to the organizers for pulling together this workshop. This is going to be a really interesting discussion today. What I thought I could do is start with a little bit of background on phage and tell you the Adaptive Phage Therapeutics story is really an amazing story, and it's thrilling to be part of it. The handout that I saw outside has some really nice background information on bacteriophage. I just wanted to throw out a couple interesting factoids that I think are just amazing about phage. When the Earth was formed, something about 5 billion years ago, pretty quickly little life forms that resemble contemporary bacteria evolved on Earth, and at the same time, co-evolving on Earth were viruses. So these viruses have been attacking these bacterial entities for about 4 billion years and have evolved to become the most powerful bacterial killers on Earth. So I've heard that every day 40% of all the bacteria in the ocean are killed by these viruses, these phage, bacteriophage. The phage, because they host on the bacteria, and they're very specific with regard to even down to the strain level, tend to be generally safe to humans and even safe to non-targeted bacteria. So that's just a quick primer on phage. So the story starts, we'll pretty much start in the middle of the story, really. The Navy Medical Research Center out of Fort Detrick, and particularly the group, the Biodefense Research Directorate, started a program that originally was envisioned by my father, who was a research scientist at NIH, and had thought of an idea for creating a diverse library, a phage bank of a phage that would host on the worst of these superbugs. So the Navy in 2010 started working on collecting raw materials, sewage from around the world, from all of the military bases and from ships and other places, environmental samples, and they'd ship those samples back to Frederick, Maryland at Fort Detrick, where they would isolate phage using clinical isolates. They'd find the phage that would host on the escape pathogens. They would then screen these phage for deleterious genes. So they do the sequencing and the annotation, and the resulting phage would end up in a collection. So that started in 2010. Then one of the things that the Navy realized was that, well, this collection, in order to be used, you have to figure out how to match a phage to a particular patient's infection. So they developed a technique, it's an assay that they called the Host Range Quick Test, or HRQT, in which you can take a 96-well plate, put the patient's bacteria in the wells, and then you can load in all the phage from your phage collection. Maybe you'd have 100 different strains of phage against that particular species of bacteria. So you could put those all in these 96-well plates with some media so the bacteria would grow. You'd add some dye that would change color based on the metabolic output of the bacteria, and the result would be that in the wells in which the phage were killing or inhibiting the growth of the bacteria, the color would not change, whereas where the bacteria is growing, the color would change. So if you image those plates over time, you'd start to generate these kill curves, and you could figure out which of the phage would be effective therapeutic agent for that particular strain of bacteria. So that was all happening in the lab, and it was amazing, interesting work, but it was very much an in vitro research effort until Tom and Stephanie, this is Tom Patterson and his wife Stephanie Strathdee, decided to go on vacation in November of 2015. They went on a river cruise on the Nile in Egypt, and Tom got sick. And he was feeling terrible. He was throwing up, and he was having severe abdominal pain. They called their friend, infectious disease doctor Chip Schooley, and said, "Hey, look, Tom's really sick. What do you think we should do?" And Chip said, "Look, don't mess around. Go to the local clinic." So they went to the local clinic, which was in Luxor, and somewhere along the line, Tom became exposed to Ebumani. Became extremely sick, was evacuated initially to Frankfurt, Germany for 10 days, slipped into a coma, was flown back to UCSD Medical Center, which is where they lived, and was just deteriorating. They're trying every antibiotic, nothing was working, and Tom was dying. And fortunately for Tom, his wife is an associate dean in the medical school, has a PhD in infectious disease epidemiology. She went to the doctor and said, "Well, what if we try something out of the box?" She knew about phage. She said, "Well, is anyone working on phage?" And I believe she reached out to the FDA and she asked the FDA, "Do you know anyone working on this Ebumani phage?" And the FDA knew about the Navy program. So Stephanie reached out to the Navy program and convinced the Navy to try this on Tom. So on March 11th, 2016, so at this point, Tom had been in a coma for 3 months. They sent a culture of Tom's Ebumani infection to Fort Detrick, where Dr. Biswas took the culture and developed, ran this process for the first time on a human subject. So the sample arrived at Fort Detrick. They pulled the collection of phage from Phage Bank specifically for the Ebumani species. They had about 100 phage in the collection at that point for Ebumani. So they ran this HRQT assay, and out of the 100, they found 5 of the phage were lytic against Tom's particular strain. So on March 17th, in that week, they grew the phage that were specific for Tom's strain. They purified it so that it could be used for a IV infusion, and amazingly, within 48 hours, Tom was awake after being in a coma for 3 months. And this was just an amazing story. In fact, it's so amazing that Tom and Stephanie now have a book deal. They're talking about a potential movie deal. And it's just an incredible success story. And it really made the Navy realize, hey, maybe we're really onto something here. A few weeks later, they tried the same technique on a 2-year-old child at Children's Hospital who had a Pseudomonas heart infection. They were able to clear the infection there. And both of these case studies have now been peer-reviewed and published case studies. So at this point, this is where APT steps into the picture. The Navy realized that there was really a potential therapeutic approach here, but they needed to bring this to market and needed to work through the regulatory process. So they looked for companies that could help with that effort, and really there were no existing companies that had been working with phage that really wanted to take on this precision approach to phage. So the Navy opened up an opportunity for companies to propose taking this on. And that's where my father, who had developed this original idea of using this phage bank approach, my dad and I teamed up. We proposed to the Navy that we could move this forward. We'd have to build out a GMP manufacturing facility for the particular challenges related to this approach. We'd have to put together a team and really look at the regulatory process. And the Navy agreed, and we negotiated a worldwide exclusive license to the whole phage bank collection and to the assay to match the phage to the patient's clinical isolates. And we also entered into a collaborative research agreement with the Navy, a very close relationship in which the Navy is continuing to expand this phage collection. The Navy's helping us with the sequencing and screening of the phage. We're working on commercializing this approach, and we're also continuing to treat patients on an emergency or compassionate use basis. So one of the big challenges for us has been scalability. So if you think about that Tom Patterson case, it was Dr. Biswas didn't sleep for something like 3 days working on trying to come up with that therapy for Tom Patterson. He had to acquire the isolate. He ran the assay to find the phage. Then he had to grow the phage and purify the phage. Took 7 to 10 days to achieve that therapy. Too slow, too expensive to really be scalable as a commercial approach. So our biggest challenge was to look at that and say, how can we translate that into a commercially viable approach? And one of the things that we recognized was that a lot of that time was spent in the growing and purification of the phage. But while the phage bank is large, it's currently hundreds, it's going to be in thousands of phage, it's still finite. So what if we were to take the entire collection and go ahead and grow it and purify it and put it into single-dose vials ahead of time? Now we don't have to do that in the critical path of patient care. Now we just have to find which of the phage will work for that particular patient. So that's what we're currently pursuing for our clinical trials and as our current path to commercialization. So we've reduced what took a week down to less than 1 day because the HRQT assay can be done in 8 hours. So that's the time for that. We've had to build our own manufacturing facility because of the unique requirements of having to manufacture so many different phage, and that facility involves special robotics for aseptic filling that allows us to do rapid turnaround. And I think this is my last slide, just as an overview. But we see the big challenges as the speed to match the phage to the patient. So we're working on optimizing our assays and adding robotics. We're working on artificial intelligence to speed that up. The time for screening and growing and purification we're dealing with by pre-manufacturing the phage. Cost, because we are doing bulk manufacturing of each of the phage components, reduces the cost. Logistics is still a big challenge. We're initially centralizing the assays and the distribution of the phage, but eventually we'll be distributing that so it becomes much more local to the patients. And we're working with the FDA. The FDA, I'd have to say, has been extremely supportive in talking through the methods for doing this unique approach. And then showing efficacy. I'm sure we'll be talking about this as a panel, but we have to be really careful because these cases are very complicated. And you think of the Tom Patterson case, extremely complicated case, so very difficult to show efficacy in cases like that. So identifying patient populations in which we can show efficacy, and we're approaching it in a very iterative process. So we'll eventually get to become more of a frontline therapy than as a last resort therapy, but that's something that I'm sure we'll discuss in the panels. So that's a good overview, and I look forward to speaking more in the panel.
Great, thanks, Greg. Our next speaker is Paul Garofalo, CEO of Locus Biosciences. Paul?
Thanks. Is it clicker right there?
Yeah.
Just forward. Hi, everyone. Thanks for having us here today. Thanks especially to Duke for inviting us. It's great to be able to participate today. Just to sort of get this all in 10 minutes, I think we're just going to do a brief overview of the company and then sort of focus in on what we might be able to do with this technology from a trial design perspective. So just a couple of words on Locus. So Locus is the worldwide exclusive owner of CRISPR-Cas3 technology. And I'm sure many people know CRISPR-Cas9 from all the news that's been recently available on that. But instead of being a pair of molecular scissors for gene editing, the Cas3 enzyme works more like a Pac-Man. It essentially makes a small nick in one strand of the DNA in the genome of a bacteria cell, and it unzips, unwinds, and permanently destroys that single strand around the genome. What we do is essentially, through synthetic biology, we use a viral vector like most DNA editing technologies, and so we use bacteriophage. We open up the genomes of the bacteriophage and embed either CRISPR-Cas3 arrays, RNA guide arrays, which would activate an endogenous Cas3 system inside of a bacterial cell, or we'll take the full cascade Cas3 complex and load it into the phage genome for delivery into the human body. And we have actually advanced through a Type B meeting for a lead asset. I'm going to talk a little bit more about that. But that phage safety profile, coupled with the efficacy that we see with CRISPR-Cas3, we think is one of the big opportunities with advancing this platform for all kinds of indications, not just in infectious disease, but also in the microbiome. The company is focusing on 3 assets. I'm not going to go into great detail about each of the individual indications, more a focus on the different routes of administration. And so our first asset, we are going to be approaching trying to go after complicated urinary tract infections. The leading bacteria that we're after is E. coli. I'll talk a little bit more about that, but we're going after a route of administration that's intrabladder. We'll follow that up with a Pseudomonas indication, likely anchoring on a HAP-VAP lead indication through a nebulized inhalation route of administration, and then we'll follow that up with a Clostridium difficile indication for recurrent C. diff going through a gut route of administration. The idea here is to systematically build 3 different routes of administration to get into 3 zones of the body where most of the infections take place. We think there's some huge opportunity in being able to stack those routes of administration to be able to use that as we build the safety data sets against the platform over time. The company's done very well over the past 24 months. We've raised $21 million. Very happy to say that we have the funds in the bank to be able to advance our first asset through a phase 1b trial. So we will hopefully get to have that data set in hand of what that human PK is going to look like with these drug products. We'll also have money in hand to get a Pseudomonas indication all the way through an IND, and we'll be working in some of the CMC scale-up opportunities for the C. diff asset. It has some uniqueness to it as an anaerobic target. So to talk about trial design well, I thought it would be really important to give a little bit more under the cover view into what a product is off this platform. And so our discovery process is very bioinformatics heavy. And so we will download hundreds, if not thousands, of genomes. We'll create huge isolate panels of clinically relevant isolates for the target bacteria that we're after. And what we do essentially is we search for conserved genes. We build RNA guides that can be arrayed so that we can actually build CRISPR-Cas3 command sets that can then be loaded in to trigger after targeting those conserved genes. Take E. coli, for example. We make sure we can cover all 180 serotypes so that when we build these RNA guides, they actually go after genes that we know that are throughout the population of bacteria that we're trying to kill. And then we also search for the corresponding phages that would essentially provide the right host range to those isolates, so that we then can move into construction of the drug product, which is through synthetic biology, to open up the genomes of these bacteriophages and load in these CRISPR-Cas3 array commands. And we can essentially then take all of those drug product candidates through a battery of pretty standard in vivo tests: tolerance, efficacy, distribution, and persistence. And we have advanced through 7-day toxicology despite some of the FDA's flexibility in what you might be able to not do there. I'm not going to go through the data in grave detail, but I think the punchline is we very typically see 4 to 5 log killing above and beyond what you would normally see from the phage lytic cycle alone. And so we typically move something forward as a drug product when we get into the 8, 9, 10 log killing range to be able to make sure we can fully eliminate a bacterial infection in many times less than 30 minutes. So that's what a drug product from our platform would look like. I think it might be important to then sort of have that grounding to talk about the trial structure. So we're going to advance our first asset as a monotherapy into a phase 1b trial. We're going to do that. We're going to go after E. coli-based presence in the bladder. We're going to be targeting colonized patients, so patients that have not yet advanced to the infected state. And what we'll be primarily looking for is a safety endpoint, but also to be able to have a secondary endpoint of PK in human. And that should give us the efficacy indication. It's not obviously statistically powered with only having 30 patients in this initial phase 1b study, but that indication of efficacy is something that we believe we can then lean on for every other trial and every other asset that comes off of this platform. A lot to work out there with the FDA, but it's a decent set of assumptions in this serious and unmet need arena. When we move from that phase 1 monotherapy, it is very likely that it will move to a pretty traditional non-inferiority model for what would be in a phase 2 study. A concomitant dose with some form of standard of care, head-to-head against standard of care. But what we do think we can do is to look at a single species, multiple site approach. And I'll talk about that next, but what that would lead us to be able to get to is to potentially have assignment into the Tier C framework that would either reduce or eliminate the need for huge pivotal trials to get the drugs approved. And so just a little bit more on this theory, which I think this is where the Duke discussions have gone and where we really think we're able to leverage quite a bit of opportunity here. So we talked about the E. coli first step as a monotherapy to establish that safety profile. But other assets that then lead off of that from this platform, we believe we can have an anchor indication, let's say Pseudomonas hap-vap, but we can bring in patients that are cultured for MDR strains in other regions of the body, be it the bladder, be it the sinus cavity, be it the skin, so that we can get that patient population set up to a level that's potentially high enough that we could potentially have composite POC and small pivotal study groups together in the same trial. A lot to work out there with the FDA, but we believe this is where the opportunity with phage safety really gives a huge step forward into the flexibility of how we might be able to design trials to essentially attack the escape pathogen systematically. And maybe a better way to really kind of understand this is to take a look at it for sort of a more traditional view of a drug development process. Let's say 10 years, $1 billion to be able to develop a drug. And at the end of that time period, you're essentially in a position where you have, for one zone in the body, the ability to eradicate a number of pathogens in that zone. Let's say complicated urinary tract infection. What we're going to be working on is to look at stacking a set of POC pivotal combined Tier C framework products that are bacteria focused across multiple sites of infection. And you get to the same endpoint to be able to have a platform of drug products that attack MDR-based escape pathogens, but you get that in a way where you're going at this in a far shorter period of time for far less cost, which inherently provides a lot less risk. And so with the safety profile of phage and the efficacy profile of CRISPR-Cas, we believe that we have a solution to be able to attack the escape pathogens in a way that no one else can. Again, a lot to work out with the FDA, but I think that was really the point and purpose of what we wanted to try to share today in the context of this discussion with the folks from Duke. We have probably all of the same challenges that Greg has, maybe even a few more. I think, we just talked about the trial design one. I think just a note on resistance, because it's always the first question that comes up when we talk about phage. We're really fortunate to have been assigned to the vaccines division of CBER. The thought process there about how essentially drug products can move through in more of an annualized basis to address the mutations that you're seeing out there with your drug product, that's the thought process that we think is needed to be able to figure out how to have a phage-based platform actually work for the long haul for society. And so while we don't believe that our drug products will be immediately resistant by any stretch, because they're cocktails of phages, we could easily see resistance within 2, 5, 10 years, and we'll need to pretty rapidly be able to address that. For us, we almost look at the CRISPR-Cas array as a drug substance and the phage as an excipient, or something that can be thought of along those lines, such that the phage delivery vector can essentially be rebuilt time and time again to carry those arrays that attack the conserved genes repeatedly. Manufacturing, I think Greg stated it well, and Greg's probably one of the most advanced in this space with his CGMP environment. Pretty impressive stuff, but we completely agree that this challenge does become a great opportunity. It takes a lot to put a manufacturing site in place. I actually, in a past life, was the chief technology officer of Patheon Pharmaceuticals. So building a CGMP manufacturing facility for phage would become very much a barrier to entry to anybody else that could come into the market. And we think the last one is capital. It's been a tough space. Frankly, raising money in infectious disease alone is incredibly hard. Trying to do it with phage is even harder. But what's really been the anchor for us, and things have changed dramatically in the last 12 months, has been the acknowledgement inside of the strategic big pharmas that this platform has applicability between both infectious disease and the microbiome. And when we look at the safety profile of phage, the efficacy profile of CRISPR, and the ability to take that platform across indications that are both infectious disease and microbiome, we have a lot of interest from that group. And I think everybody in the room would agree that one of the big things we have as an opportunity as sort of a cluster is to entice big pharma back into this game with the checkbooks that they have. And we certainly hope to be on the forefront of being able to break that through. So thank you very much for the time. I know that was really quick. There's a lot of other information related to the company. But again, it's a pleasure to be here and look forward to the panel discussion.
Great, thanks, Paul. So our next speaker will be Alan Joslin, President and CEO of Oragenics.
Okay, there you are.
Well, thank you for inviting me to give this presentation. My approach is actually a little bit more along the lines of the traditional antibiotic development. Oragenics is a company located in Tampa, Florida, and we have identified a new class of antibiotic that allows us to hopefully introduce treatments for a variety of infections. What we do is we actually develop genetically modified bacteria. This slide just shows you the makeup of our portfolio. AGO13 is a lactococcus bacteria that has a human gene inserted into it for treatment of oral mucositis. And we essentially use a bacteria as a drug delivery vehicle. OG716 is our lead compound for treatment of Clostridium difficile, and I'm going to go into some detail about this. It's a brand new class of structural antibiotics that we are developing, and of course we have the rest of that library within the antibiotic class that we're just trying now to better understand how it works. So antibiotics, and this slide just kind of highlights for you that the CDC list in 2017 for multi-drug resistant pathogens. And as you can see, there are still a large number of patients, about 3.5 million in the United States, that are infected with these pathogens for which new treatments are needed. We've heard two presentations on phage approach, and now we'll go back towards more the traditional antibiotic approach. In this particular case, our lead compound is under development for treatment of C. diff. C. diff infections occur in approximately 500,000 patients still in the United States annually, and about 29,000 of these patients are going to die, either directly or indirectly as a result of the continuation of that C. diff infection. Of particular interest to us is that very bottom bullet, and that is that as these patients relapse, approximately 8 to 10% will develop a co-infection with enterococci, for which vancomycin, which is standard of care in C. diff, will not be effective in this group of patients. So right now, as a company trying to introduce new therapies for treatment of existing infections such as C. diff, we essentially have to climb systematically up the mountain, so to speak, to actually gain usage. In this particular case, the slide shows you the treatment algorithm commonly used to treat C. diff, starting with very inexpensive treatments, including metronidazole is first choice. If those patients relapse, they'll move on to vancomycin. Fidaxomicin or Dificid is also approved for use in C. diff. So you can see there's not only a lot of activity going on in the area, it's also a pretty significant hurdle to introduce a new product, even though one is needed in this particular population of patients. So what are antibiotics? Antibiotics are a novel class of proteins that are produced by certain gram-positive bacteria to prevent overcolonization within the microbiome. So in this case, Streptococcus mutans will put out tiny amounts of this antibiotic to essentially preserve their space in the microbiome, much the way a skunk would put out a spritz of sulfur and protect its area. And so the biggest issue that we have had from a technological perspective is how do you produce enough of this antibiotic to actually turn this very, very potent protein into a therapeutic? We know that it's manufactured in tiny, tiny amounts, and what do we need to do to improve upon that? And so for about 10 years, the founder of Oragenics worked in the area of trying to manipulate the fermentation and purification process in order to grow the amount of antibiotic that it's produced within that fermentation vessel, and that was, quite frankly, an abject failure. And so we turned to a company called Intrexon, and Intrexon is expert in manipulation of genes through their synthetic biology platforms. And what we were able to do was two things. One, we turned off the regulator gene, because as you can imagine, that if a bacteria like Streptococcus mutans produces too much of this particular antibiotic, it'll essentially kill the host. And so what we're trying to do is not only turn off the gene that stops production of the antibiotic, but also develop a fermentation process that allows for the sweeping away of the produced antibiotic and allows that bacteria to continue to thrive for a period of time in the fermentation vessel to make the enough of the antibiotic in that particular production run that allows us to then harvest the supernatant and purify the antibiotic. Along the way, we also were able to use a process of saturation mutagenesis and essentially substitute out, down, as you can see, the structure of our lead, actually the original antibiotic, MU1140, where we substituted out each of those amino acids to better understand the structure-activity relationship for this particular class. And we now have a library of about 700 different structural analogs that we're now trying to call and identify the best antibiotics for specific infections, along with very specific physical chemical characteristics, which is also an important consideration here. So the antibiotic profile, we know from early mechanism of action work with the class that we have a very, very unique mechanism of action as far as binding to lipid-2, which is the foundation for cell wall biosynthesis, very different than other cell wall biosynthesis products that work through various other pathways. We actually identified a lead compound out of this library, OG716, that was actually impervious to proteases. So you were able to swallow a solution of this, it would arrive intact in the colon and thereby treat the C. diff infection. And this slide just shows you from a standard animal model of the Syrian hamster, C. diff infection, clindamycin treatment with OG716, which is our lead asset, positive control vancomycin. You see all the animals with OG716 have lived even through the follow-up period where spores would regerminate. We did lose the vancomycin animal. Our second compound, OG253, did not work in this assay, even though microbiologically it was very, very potent. The reason being it was being chewed up by proteases. So the combination of physical, chemical, and microbiological evidence is really important in trying to develop a brand new class. Now, here's some of the issues that we face as a public company in trying to advance this library. First, we have a BSL-2 pathogen in Streptococcus mutans that we use for production, and clearly finding a facility willing to take on that class has been a challenge. We've actually tried and surveyed about 80 companies to come up with one that was willing to work with us. Right now, we're only able to scale this at 1,400 liters, which results in about 100 to 200 grams. And it's a very expensive purification process because it's a large protein. Commercially, as the previous speaker spoke of, Big Pharma's not really interested in antibiotics. And as a result, investors aren't coming into public companies yet because of this lack of an exit. And so this is a huge challenge for a company that's about a 10 million market cap company. And as a result of stewardship, the likelihood is that I'm going to be successful and be put on the shelf for quite some time. So I'm encouraged by some of the recent developments with its CMS and FDA. And then, of course, the cost of production and clinical trials in the area is very, very high. And so with that, I'd like to thank you, and that's it.
Okay, thanks, Alan. Next, I'd like to invite the other speakers back up, and then also invite Scott Stibbets is Chief of the Laboratory of Mucosal Pathogens and Cellular Immunology at Center for Biologics and Evaluation Research, CBER, at FDA.
Wow, that's bright.
Okay, so a lot to talk about. So those are our first introduction to non-traditional antibiotics, and I think everything I heard sounded pretty non-traditional. So Scott from FDA, may I give you the first dibs at reacting to what you heard and give some comments and even questions back to the presenters if you'd like.
Did anything surprise you?
Well, not really, because we work in an area of surprising development. So I think we did see a spectrum of new ideas, incredibly exciting ideas that seem to have great potential. And at the FDA, we welcome these. They actually make our job even more interesting and fun than it normally would be. But I think what I'm sort of assessing as I hear it is, are we up to the regulatory challenges for these things? And based on what I heard, I have to say, yes, I believe we are. I mean, I think the Center for Biologics and our office deal with a lot of very challenging products that we have to apply science-based decision-making. The regulatory requirements, the requests that we make of sponsors, we want to be sure are well-based in science, that we are asking for supplemental material or studies that really provide necessary information. We summarize this as the nice to know versus need to know. And I think that we've gotten pretty good at that over the years dealing with these types of products. So obviously people can have a different opinion, and we're always welcome to listen to that. But I think to some extent the challenges in the product development are evident. They are not necessarily, and I was glad to see people representing the FDA's role so far in their development plans as being largely positive and guidance oriented and not so much restrictive and draconian. So I realize that's a little bit of a soapbox speech, but I'll be happy to try and answer any more specific questions.
Great, thanks, Scott. Let me turn to the full panel here, and each of your presentations did discuss some of the challenges particularly in trial design, challenges in recruitment, especially for rare issues and potentially small populations. Can you sort of expand on some of those challenges and sort of steps you're taking to address the recruitment and small trial design issues?
Well, I thought it was interesting that Paul's company and our company sort of landed in the same place with regard to the UTIs, because I think that the UTIs present an opportunity for non-acute illness. So you have a little bit more time, and it's an opportunity to better elucidate the efficacy signal from a less complicated case than like the Tom Patterson type of cases where there's just so much going on. It's hard to figure out what the curative agent was. So I thought that was really interesting.
I guess to add on to that, I think, and maybe just to give some props to Scott as well. I mean, I think the FDA, in our interactions with them, you always I've been dealing with the FDA for 25 years, so you're always a little nervous to interact with the FDA, so you don't say something that hurts you down the road. But in this particular area, I think we found them very flexible and very open. And so the idea of being able to start to work through some of these, I mean, patients are scarce. So the opportunity to do a single species run where we might be able to look at multiple sites of infection, I think is perfectly positioned for phage, frankly, because that safety profile, that sort of known entity from even the GRAS designation over on the USDA side, I think gives a little bit of flexibility to everybody involved in these trial designs. And so I think you have to have some caution when you're going after each new route. You likely need some pure safety data with each route, say nebulization, for example. But I think once you get that body of evidence for safety, I mean, there may even be opportunities for companies like Greg and ours to actually sort of share safety data sets as we prove those regions of the body get safe. So I think there's some pretty interesting opportunities here. And I think the problem is we need patience, and we need to be creative about how we're going to get them.
Alan, any additions to that?
My development is more traditional in the sense that in my case, I'll be looking for treatment failures on existing therapies in a very, very specific population of patients. It will require, even with the newest guidance, the draft guidance that just came out, setting up a clinical trial at tens of centers around the United States, hunting for those few patients that will fail and require a new class. And so the startup costs of that type of a trial may actually be higher than some of the more traditional approaches, because you have to set that trial up at so many more centers than you normally would have to run a more traditional, run-of-the-mill anti-infective type clinical trial. So I think that that's one of the challenges for moving forward with some of these new classes of treatments, is just the breadth of what's required from a study setup perspective in order to rapidly accumulate patients once they do become available. And so we just have to be mindful of that.
So Paul, you expanded a little bit on some of the sort of more efficiently designing approaches that look at products across a range of infections and sites. Maybe expand on that a little bit, on
Sure.
how that's helped, and challenges unique to that, and maybe get some of the other panelists to sort of opine on that as well.
I guess the way we look at it is, in large part, phage in its drug product form is a liquid that works best when it topically touches the infected cells. So for those who don't understand the biology of phage, it's an active agent, and so it essentially inserts a payload of itself with command to replicate, and then bursts that cell with hundreds of copies of itself. And so you need to be able to get that phage to almost topically touch that bacteria cell before it can really get into that mechanism of action. So I think the idea, and what led us to sort of think like, we could do a lot with this is, if you just take inhalation out for a second, that same drug product that you would use in the bladder is likely what you would use for a skin infection. It's likely the same formulation potentially, although maybe in an aerosolized form. You may also have some form of sinus wash that gets used, but that drug product being the same drug product adds to, I think, an opportunity to be able to reuse that product. For the record, for inhalation, we also believe it's the same drug product, but needs to be added to a nebulizer, which would require safety as an initial step, but once you establish that safety for each route, Greg and I are likely making product that could be interchanged for those different zones of the body. So why not enroll patients across those zones to be able to get a larger swing at a nicely sized phase 2 that doesn't take 10 years to get 20 patients, and then maybe you have enough data coupled with the known safety factors of phase to waive phase 3. I mean, this all has to be worked out, obviously. But I think that's a real opportunity for us to attack escape pathogens that don't exist with anything that has a typical tox profile.
Yeah, no, that's interesting. So we have been treating patients under this EIND program, which has provided us an opportunity to see exactly what Paul's saying in terms of different routes of administration with the same basic therapeutic prep. We've had one patient who had a lung infection, and the plan was to use the phage therapy both as an intravenous route of administration as well as nebulized. Now, to prepare the phage for the nebulized route was going to take a little bit of time, so they started right away with the IV approach, and that cleared the infection right there. So then they decided, well, we'll just skip the nebulized approach. But that's an interesting point. And one of the things that we're looking at with our clinical trial design is an initial step where we have an adaptive design that allows us to try a couple different things prior to going into our blind mode.
So Scott, you talked a little bit about nothing you heard was sort of a surprise, and you all are seem to be gearing up to address these. But we are hearing about products that are very, very different from each other, right. Non-traditional, taking different mechanisms of action, and maybe if you can expand a little bit on how your group is addressing this just diversity of products that are available.
Right.
Coming on
I mean, I think in the phage space, we've had a lot of discussions about what the CMC requirements should be for phage. And again, they are what we consider to be absolutely essential, and I think they're easily achievable. But basically, the phage should be as pure as possible. Again, depending upon route of administration, you do want them to be free of endotoxin. Now, the exact limits of endotoxin for different routes of administration is something that we still have ongoing discussions about. We really only have proven or accepted limits for parenteral administration, so there's some questions around other types. But I think the really novel thing with phage is what makes phage unique, maybe more unique than other anti-infectives, is their ability to affect genetic transfer. And I can't think of a product that's like that. And so, that's not a reason not to go forward, but it is a reason to give a lot of thought to what types of characteristics you want in a bacteriophage. We want them to be lytic. That's largely, I shouldn't say largely, but at least partially due to an efficacy issue. If they can lysogenize, the lysogenized cells will be resistant to the phage. But then there's the issue of generalized transduction, and I think that's an area that there will continue to be a great deal of discussion on. But we think, we wonder, sometimes we worry about putting phage into a context where, by definition, you're treating a highly virulent, highly antibiotic-resistant organism, and you're introducing an agent for genetic transfer. In a sense, it's a question that's larger than the individual patient, but that's something we think about. not sure I'm answering your question. I get the feeling. I kind of picked.
I think that was good.
Just started
Yeah, that was
rambling.
fine.
But, I mean, the other thing is that even the issue about transduction may be dependent on the context. For example, if you have a phage that is capable of transduction, but it's been selected from a bank to treat a particular pathogen, a particular patient isolate, and you're growing it on that patient isolate, essentially you're talking about the possibility of transducing back to the original pathogen. Maybe that becomes of less concern in that context. In other contexts, say you're going into the gut, there may be many, many other bacteria where you could be transferring these weapons, and so it becomes a different issue.
So we are going, any other comments on, so we are going to turn things over to the audience for Q&A in just a minute. If you're on the web, just a minute, if you're on the web and watching, if you can email your questions to duke.abx@duke.edu. And I'll get one more question to the panelists, and then we'll go to the audience. But, you know, across your presentations, challenges and ways, you all have seem to come up with very innovative ways to address the challenges that you're facing across trial design and recruitment, demonstrating clinical benefit, manufacturing challenges, and then capital. My question is, what would be your single most sort of challenge that you all are trying to work on, and aside from the capital, because I think I'm not going to give you that much of a softball. So aside from the capital, what would you say is in clinical development and in the scientific side the biggest hurdle that you all are dealing with?
Yeah, I just want to say, before we started this morning, I was talking to Paul and Scott a little bit about the historical challenges of antibiotics, and I'm not a historian, but the little bit I know about the introduction of penicillin involved significant challenges in manufacturing, and that they had to reconstitute the penicillin from the urine from the initial patients because they just couldn't get enough penicillin to treat patients. And so I think that while we have challenges, I think they're probably relatively modest compared to the initial challenges of producing the first antibiotics. So I just wanted to throw that out there. But otherwise, I think that we covered a lot of the challenges. Certainly for us at Adaptive Phage Therapeutics, we have this challenge of having a large library, which involves the manufacturing of many different phage. And in a traditional drug manufacturing process, you might have to produce one large batch for a clinical trial, whereas we have to produce, let's say, hundreds of individual batches. And that challenge is what drove us to create our own manufacturing facility. So that was a very difficult decision point for us, and it's one of those field of dreams moments where you have to build it, and they will come in building that manufacturing capability.
I mean, I'll add to what Greg said, and I think if I take capital out of it, I think the biggest challenge are the scientific unknowns, like what Scott just brought up. So we exist in sort of a world of two edges of the sword, right? So FDA has been absolutely fantastic, waiving toxicology studies, waiving dose range finding studies. We exist in a world, like John Rex said earlier, where MIC curves don't apply. So we go up against any group of really well-seasoned, MDs, PhDs, venture capitalists, whatever it might be, and you don't have any of the traditional data that's required to get anybody confident to let you approve something to get to another phase, to give any kind of comparator towards, let's say, a small molecule. You exist in this world that's brand new, which, by the way, is super exciting. It has a really cool part of it, but it takes resilience because you have to hear the objections, you have to figure out how to work through them. Like the transduction issue. So that's an issue that Scott and his team have given to us, and we've come up with a battery of in vitro tests to be able to essentially select phage that don't have those capabilities to be able to address it. But we got to go back to the mountaintop and have that discussion about whether did we get through that? And if we didn't get through that, what's next? So to me, the biggest challenge is we're on the bleeding edge of science. And I mean, a little bit of uniqueness for Locust is that we're also CRISPR. And so there's tons of questions out there about CRISPR. And so here we are trying to talk about selectively removing bacteria, and we get questions every day about, well, what does that mean for editing human genes? Well, it means nothing. We're using phage. They don't infect human cells. So, and we're not trying to edit a gene, we're trying to kill a cell. But you know, when you're on the bleeding edge of science, it has that cool part to it, but it has these other barriers of you have to be resilient to break through. You have to be able to work through answering those scientific challenges, and that's the biggest challenge.
For myself, with a new class of antibiotic, up until about 6 months ago, I would have said that manufacturing was my biggest challenge, but I've overcome that hurdle. I think that the biggest challenge now is creating the perception of value of a new class of antibiotic being available to society moving forward. And I think that once you are capable of developing that value proposition, it's at that particular point in time that I think all of us will benefit from having companies and investors be more willing to come back to the table and begin to invest in some of this once they realize the value in what we're doing, moving these extremely novel approaches to treating infection forward.
I'll just sort of reiterate what I said before. I mean, I think a lot of the basic knowledge that would help us in our regulatory role is not there. Phage have been understudied. Study of phage has been underfunded. It was amazing to me, having grown up more in the molecular biology space in E. coli before I started to work on pathogens. But when I came back to phage, for example, in the staph field, you go to the literature to find what you think are just answers to really basic questions, and work simply hasn't been done. So there's that. But I'll just also add that we have to keep in mind that each bacterial phage pair is unique. So in addition to all the novel challenges associated with phage therapy, and we tend to echo the common wisdom about resistance and cocktails and all that, but we just have to keep in mind that, for what pair of bacteria and phage, because you can find exceptions to almost every point in that common wisdom.
Okay, so we'll turn to the audience for questions. Hi, yes.
Hi, this is Sybil Tasker. I'm just so curious about the discussion with drug product and with phage, how do you define a drug product? It seems impractical to have an IND for each phage, but it also seems impractical to have an IND for the whole library. So I was just curious if the panel could discuss that.
That's a great question. That was probably one of my biggest questions when I started adaptive phage therapeutics is, well, what's the drug? It seems like it's a service. But we did have to come up with a definition of the drug. So what we've landed on is that the drug is phage bank, is this whole collection of phage. And then one of the big challenges in our discussions with the FDA is how do you, if that's your drug, and by the way, it's not a fixed collection, it's constantly growing. So how do you regulate that? And so that was, it's probably an ongoing, continuing discussion with the FDA, but that's our current position, is that the drug is the phage bank.
Yeah, I'll just add to that. I mean, I think there are at least two aspects to regulation. One is through the development process in the IND phase, and the other is final licensing. I think for single phage cocktails that are defined, the current pathways are completely appropriate. And for things like phage libraries or personalized medicine, whatever you want to call it, I think that the IND process is up to the task of getting these types of products, I guess, into clinical trials and testing their efficacy. But I would have to agree that I think the question in that setting of a phage library, a treatment center, whatever you want to call it, what the final product is and what FDA approval looks like is something that is still an evolving question. But we have a little time, I think.
Well, and maybe to build off that, since we're one of the groups trying to help answer that evolving question. So for us, it's probably a hybrid of the two. Our proposed E. coli product is a 6-phage engineered cocktail. Each of the phage drug products we consider as an intermediate. We do a final blend into essentially a cocktail. We are working on identity assays to make sure that the right phages and the right percentages are in that final mix. That's what goes up on final stability. That's the final drug product. I mentioned before about resistance and the eventual need to rotate in new phages. So we hope Greg breaks all that ground for us before we get there, and then we'll just leverage that with Scott.
John?
Fascinating. John Rex, talk to me a little bit about the pharmacology, I do mean the PK of phage. And Paul, I was fascinated by your idea that you were going to treat complicated UTI by putting something in my bladder. Absolutely. And so if I put it in the bladder, where does it go? If I inhale it, where does it go? If I put it in my vein, where does it go? If I take it by mouth, where does it go?
Yeah, well, and start. I mean, there have been some animal studies. We've been looking at a collaboration and started a collaboration with Stanford in which they can do some imaging in animal models to see where the phage ends up. But I know there's work that was done at NIH. Carl Merrill, my father, did some work showing where the phage ends up in terms of being filtered out of the liver and other organs as part of the immune system. But yeah, that's an area that we.
So you're saying you don't know yet where it is?
I mean, there's animal models, and we'll be looking. But yeah, that's an area that needs additional work.
So if I put it in my vein, does it distribute widely to body tissues? Would I expect to find it in the urine?
There's been studies that show that it ends up in the bladder after you put it into the
So I can build
bloodstream.
on that. I can build on that a little bit because we've taken our actual proposed drug product and moved it through a series of distribution and persistence models. Let's start with the bladder. So we'll dose a single shot into the bladder, contact time of, I think, 30 minutes. We see, as we then essentially take time points of sacked animals and look where it's gone, we see it's fairly localized in the bladder. It does distribute back into the kidneys, which is what we did want to see for a bladder, basically preparation for a bladder infection. We see essentially staying power for 6 to 8 hours at very high titers. And this isn't, obviously these are distribution and persistence assays, it's not reacting to anything. So they're not getting watered down with time. And we see, depending on the phage block, complete clearance of the phage through the urine and the feces within 24 hours to 48 hours. So it goes in, it hits for 6 to 8 hours, essentially in the area of the body that we want, and then it secretes out as you would normally expect. Find no traces of it after 72 hours in any of the assays that we have. In the gut, very similar situation, localized into the upper and lower intestines and then into the cecum. We see a high density of concentration in the cecum, and again, the 6 to 8 hour stay time, and then passing out through both essentially the normal exit pathways, urine and feces. Again, with the sort of 28 to 48 hours, no detection at all of the phage that we're putting in. And we typically see the same amounts that we enter. So let's say we put in something that's 10 to the 9, we see 10 to the 9 come out. So we're pretty happy with those distribution and persistence records. We've shared those actually as part of our Type B meeting on our E. coli asset with the FDA. I think that for at least the body of evidence that we have in animal, it looks like we're good to try this in human. There is some pickup in the blood. It's minimal, but it's, at least we believe it's known that the various organs in the body are built to filter phage. That's why we're trying to actually work around an IV to be able to get drug product to the site of infection so that the amplification works as potently as possible.
I think one of the challenges, John, is that there's such diversity in the phage, that there's some phage that will circulate for thousands of times longer than other phage. And so that presents additional challenges for PK.
So I'll just say that complicated UTI is not limited to the urine in the bladder. It's in the kidney, it can be in the perinephric tissues, and it can be in the blood. So if I'm going to treat somebody with a monotherapy with a phage, I actually would want to know that would also clear bloodstream infection. So the fact that it gets elsewhere, from my perspective, is a good thing rather than a negative. Anyway, thanks.
Yeah, Vance Fowler, Duke. Absolutely segueing off John's point. My concern here is how you fundamentally treat a systemic disease with a localized product. Now, the parenteral administration makes sense in the case in point that you kindly provided about the pneumonia. It makes sense because, in point of fact, that's what we do clinically all the time. The converse does not make sense, however. And in particular, the issue of instilling a product in the bladder to treat complicated UTI. Because if you're treating an infection in the bladder, guess what? It's not complicated UTI. By definition, it's a simple UTI. Complicated UTI has got, they're systemically ill. There's a mortality rate. There's a bacteremia, there's a rate of bacteremia. And the issue of route of administration and where that product goes, to my estimation, is going to be critical in where these incredibly exciting compounds are ultimately employed. I'll give you another example, the notion of the administration, localized administration in the lung for treatment of HAP-VAP. Great idea. We use it for a number of compounds already, TOBI for one, tobramycin. But it's localized, and we never use it alone. So we use, there's something else to treat the systemic element of HAP-VAP, because again, there's a high mortality rate. The patients are not dying because the infection is limited to the lung. They're dying because it's a systemic infection. So I guess what's the message? I guess the message is the route of how these agents are ultimately employed is going to govern, or going to be governed by what you're ultimately trying to treat in terms of this, and specifically treating a systemic disease with a localized product. Thank you.
Comments?
I would just say, I think that a lot of infections are more systemic than they are on their surface thought to be, like burn infections. You find that the bacteria can be spread throughout the body, can be sequestered in different areas, and so I think from our perspective, we thought that the IV route might be the best route to treat a lot of these things that might be intuitively thought to be local.
Yeah, I think, and I mean, maybe to build on that, we're certainly aware of those concerns, Vance. We get these questions all the time, and I think for the monotherapy approach, to be able to get safety profiles established for these drug products, to be able to reuse and leverage that safety profile, and then take that into unfortunately, some very standard non-inferiority trials, you're going to have the combined dose with standard of care. So I actually think it's going to be a long time until we figure out how to look at monotherapy for these kinds of drug products in actual infected patients, because as you well know, MDR infections that are this late stage inside patients, ethically you're not going to be able to just try a whole bunch of stuff out and figure out what works. It's going to take a number of, I think, layered approvals in this concomitant type of dosing structure to establish a body of evidence that might eventually lead us to the point where we could go into a different type of mix. So I guess our thought there, Vance, is that, you know, you go with a combined dose with standard of care to get, let's say, Pseudomonas, Klebsiella, and E. coli through to, you know, that type of label, and then, you know, flip it, so that you can then take a few of the phage cocktail from Kleb, Pseudomonas, and E. coli and put those together into something that's a lot more traditional, where you would be dosing it in a way that likely you would be more comfortable with. It takes time, and I think what our proposal is the way to address it is through modular product development. And you get these bug-specific, single-species, multi-site bodies of evidence created, and then you look to see how you can combine those in a modular way.
I've got a question from online that I'll go ahead and wedge in here. The question is for Paul, but around CRISPR-Cas3. So you indicated that the CRISPR-Cas3 had a greater kill than phage alone, I think is Greg shows dramatic clinical benefit with phage alone. Safety with phage is a good concept. So the question is back to safety concerns for CRISPR-Cas3, and then also is a two-part question. What are the safety concerns with CRISPR-Cas3 is the first one, and then how will you demonstrate the CRISPR-Cas3 payload brings enough added benefit, so safety and the benefit?
So I think all of our animal studies for in vivo efficacy now have control arms that are sort of two control arms. They have a wild type in addition to essentially an engineered phage. So we can see in vivo the difference between essentially what the wild type phage cocktail would do of the exact phages that we then engineered with CRISPR-Cas3 arrays. So you get a head-to-head comparator that shows efficacy differences between those two. I think on the safety side, and maybe just a little bit of Cas3 info, so CRISPR systems are in any number of bacterial species. It is a bacteria cell's immune system. Cas3 is the most dominant and prevalent Cas system of any CRISPRs that are out there. We believe they're in over 50% of the existing bacterial species that essentially have been sequenced. And so you're simply reusing the immune system that sits inside the bacteria cell on itself. And so if your point and purpose is to kill that pathogen, you're sort of 50% across that line by simply tricking it with an RNA guide that points it to its own genome. So I think we've done well with the FDA to actually look at whether there are any lead-on safety impacts, but the simple fact that phage doesn't infect a human cell leads to a safety profile that's consistent with phage. And so I think we feel really good about safety. And what we want to see is we want to see PK in human.
One last question, or one more question?
Yeah, Todd Black with Merck. So you touched on it a little bit, and it is with the safety side. It's delivering viable recombinant organisms. Has there really been any definitive guidance yet on what's going to be acceptable in terms of looking at shedding or, not just the host impact, but the environmental impacts?
So, I mean, I think we are constantly talking about the need for a guidance document, and one of the questions is when you start to write that document. And generally speaking, I think the guidance document fills the best role once you have a body of experience that you can draw on to provide guidance. And I think our feeling is that it's really early days. It's a moving target. We try to fill that void by speaking at conferences, about the CMC considerations, for example. I suspect that's a major part of it, of what we're looking for. in terms of CMC for phage. For example, we spoke at the Evergreen Conference, at a conference in Tbilisi, Georgia last year, many of these. So I take your point. I think writing a guidance document also takes quite a long time, and so we're looking for other ways to kind of get our message out. I don't know how satisfying an answer that is. But,
the ag industry was very surprised, I think.
beg your pardon?
The ag industry was very surprised, I think, by the backlash of recombinant organisms.
In terms of recombinant specifically. Right. I mean, our position is that we don't view recombinant organisms differently as a class. In other words, they will have different qualities and capabilities, and they're judged on that, but the fact that they're recombinant does not trigger a different type of treatment.
All right, great. So that brings us to the end of the session. I'd like to thank all of our presenters and panelists for a really good discussion around these products that are quite innovative. So thanks for all of your time. We'll go ahead and take a break. We'll start back up for the next session at 11:15. Thank you.
Thank you. Nice to meet you.
Okay, we're going to go ahead and get started soon. If I can ask you all to make your way back into the room. Monica, can you guys help round the people up? Yeah, thanks. Troy. Okay, you're here. At least you're here. Just try to get a few more people back, and then I'll introduce you. Yeah. Yeah, sit back down, and then I'll bring everybody. Yeah, I'll bring everybody up. Yeah. We're going to go ahead and get started in our next session. During this second session, we're going to focus on agents that restore activity to traditional antibiotics when used in combination. As in our first session, discussion will encompass scientific challenges and developmental concerns. We have two presenters from companies, and then we'll invite an additional two folks up here as panelists. Finally, we'll again allow Q&A time, and I think as we had a number of folks sending emails online. I'll try to move that Q&A time up a little bit earlier in the session to accommodate all of the folks who have a question or comment. So our first presenter today in this session will be Troy Lister. Troy is Vice President of Research at Sparrow Therapeutics. Troy?
Morning, folks. So I'd like to start just by thanking the organizing committee for the invitation to present. Let's get some public company stuff out of the way. So I want to start first. I'm going to focus mostly on one of the platforms we have at Sparrow, and focus mostly on the science, and sort of going to hit some of the challenges that we've come up against and overcome as we go through the presentation, and sort of circle back and exemplify them at the end as we summarize. But I wanted to take just a few minutes to sort of, reiterate, but mostly remind a lot of the folks in this audience who are experts in this field, why we're here today, why we need to be thinking about alternative approaches, especially for gram-negative bacteria. And as a recovering medicinal chemist, I'm acutely aware of a lot of these problems in that, it's readily easy to identify target-based inhibitors, even in the gram-negative space, but the real challenge comes in translating them into therapeutics, and even very early on, how to just kill a whole cell bug. Translocation in the outer membrane is primarily the most difficult resistance mechanisms that we come up against. And so I just wanted to run through a few of these because it really frames the approach that we've decided to take, which I'm calling potentiation, and I agree, John, is a very ambiguous and amorphous term. And I think primarily the rationale for that, for this particular chemotype, is it really translates across both the transform and restore. The restore part is something that we didn't anticipate coming into this and have elucidated as we've gone through. So as I said, there's multiple reasons why it's very, very difficult to kill gram-negative bugs. The primary reason is actually both membranes, but primarily the outer membrane of gram-negative bacteria, and primarily the architecture in the outer leaflet and the lipopolysaccharides specifically. It's an intrinsic barrier to penetration for a lot of would-be wonderful gram-negative antibacterials. And for you to successfully translocate that membrane, you've either got to rely on porins, of which there are multiple types, and they're very different across the gram-negative escape pathogens, E. coli, Klebsiella, Pseudomonas acinetobacter, and many of them are actually undefined. And annoyingly, some of them are very chemotype specific and some are very non-specific. You may be very, very lucky in that you have a chemotype that can passively diffuse across both the outer and inner membranes, but that is extraordinarily rare. And you also may be susceptible to some self-promoted uptake, but it is equally rare. But if you're fortunate enough to have a chemotype that can take advantage of any of these mechanisms, most often you come up against the second most predominant resistant mechanism, and that is efflux. And you're very quickly efflux back out of either the periplasm or the cytoplasm by the multiple promiscuous multi-component efflux pumps, which again are vastly different across the gram-negative bugs in the escape category. So, when you're optimizing a chemotype that you think is going to be a wonderful target-based inhibitor, you can try and optimize for these two aspects and keep a drug or a putative drug inside the bacterium for long enough to interrogate its target. But you've got to incorporate all that SAR to be consistent with actually remaining on target. And then also hopefully avoiding any target-based resistance that may come up. Highly mutable targets are obviously intractable in terms of development. And then there's also instances of, and these are quite rare as well, of chemotype-specific resistance, where you have modifiers or degraders, which can then, again, diminish the amount of material that you have inside the cell to interrogate the target. And these are all kinetically driven processes, and they're all simultaneously operative. And so you've got to optimize for every single one of these to enable you to have a sufficiently active, whole-cell killing gram-negative antibacterial. And then just for fun, throw on top of it human ADME. And so you've actually got to be able to get that drug to the site of action and keep it there for long enough to actually take advantage of all this optimization that you've gone through. So needless to say, these things all pile on to make it extraordinarily difficult to come up with novel monotherapy gram-negative antibacterials, which is really why we haven't seen one for multiple decades going through clinical development. And what it also means is there's multiple novel mechanisms of action and novel chemotypes that are unable to be deployed into the gram-negative space because they don't manage to get any one of these attributes that are essential for gram-negative targeting. And most of it is out-of-membrane penetration. And so when Spero Therapeutics got into this and really wanted to try and crack this, our goal was really to try and leverage and utilize that wealth of chemotypes and mechanisms of action which reside predominantly in the gram-positive arena purely because they can't get into their target, can't get into a gram-negative bacterium. Their targets exist. They're highly potent and conservative targets with very low incidence of resistance because they've never been deployed in gram-negatives, and they're completely undeployed simply because they don't get in. And so we really wanted to go at this target, and we really set up a platform to do it. And the chemotype that we decided to leverage to do this, to target the outer membrane specifically, were the polymyxins, being probably the best known outer membrane targeting therapeutics out there. But as John pointed out earlier on, they're also plagued. And this is actually data from the same plasmamycin study that John pointed out early on, in which there's an extremely high rate of mortality associated with colistin therapy. They're very well known nephrotoxins. And indeed, this was probably the first challenge that we came up against and had to overcome, is that there's a stigma associated with the polymyxins, and well justified, they are highly nephrotoxic. And that for us to deploy this chemotype in an adjunctive therapy, we were going to significantly have to defray the risk of nephrotoxicity. And so that's what we did. So a lot of companies and efforts have taken place over the last several decades to try and identify safer polymyxins. The breadth of spectrum that they possess, their very, very low incidence of resistance up until quite recently, are admirable traits and ones that we would want to replicate. but the safety issue was significant. 50% mortality in the setting of severe gram-negative infections, and very high rates of nephrotoxicity at the therapeutic doses. The only way to mitigate that risk in a preclinical sense, to give you any level of translation, is to study novel polymyxins in higher species, multi-day, multi-dose toxicology experiments, and specifically monkey studies. So the only ones that have predictability from our knowledge and our demonstration to clinical utility. And so that's what we did here. So I've got a lot of data on the slide, and most of the slides are pretty chocolate data to try and get this done in 10 minutes. But the first, almost essentially one of the first studies we did was to run 741 up against, which is our lead potentiator molecule, directly up against polymyxin B in a monkey study, looking specifically at renal biomarkers for nephrotoxicity, blood urine nitrogen, serum creatinine, and associated histopathology. You have to look at microscopic changes in the kidneys to have any translation of these biomarkers. And we did this study. It's a 7-day, 3 times per day study via 1-hour infusion under non-GLP conditions. You can see essentially very high rates of nephrotoxicity at quite a low dose of polymyxin B. Any higher than this, and you actually start to see mortality. 741 was essentially completely clean at what translated into be almost double the exposure. So you're seeing no effect, double the exposure to something which is quite nephrotoxic. So very nice early indication that we're on to something quite different. From a chemotype standpoint, exactly the same core as polymyxin B. What we've eliminated is 2 positive charges that lie in the side chain and a lot of the lipophilicity. Both of these are implicit in both nephrotoxicity, but essentially, critically, are also essential to target anti-infective activity. So you can see here, let me go back. Why didn't it come there? We've lost all of the antibacterial activity. So 741 does not kill bacteria on its own. It has to be combined with another agent. Very quickly through this, translated this study, went through GLP 14-day IND enabling studies, again using monkey, I keep doing the wrong button, I'm sorry, which recapitulated those earlier findings beautifully over 14 days. You have to do a rodent species for IND enablement. We chose rat, and this is really why no other groups have seen a credible translation of their preclinical models, because you can see rat as a horrible species for predicting nephrotoxicity. Its NOEL exposure is almost 100-fold lower than that in non-human primates. We then went and ran a phase 1 SAD-MAD, of which there have not been any successful polymyxin Bs to transit through a phase 1 study, and successfully dosed 741 for 14 days at 600 milligrams 3 times a day. It's almost 2 grams of a polymyxin for 14 consecutive days without encountering any of our stopping rules, which were hinged on renal biomarkers of serum creatinine. And that translation of our, what we call here as our effect dose, we started to see some elevations in serum creatinine in the high dose group. AUCs at a no effect dose, very similar, so this pointer's not working. The green, match up the greens there versus the effect dose. The exposures are almost identical, so wonderful translation of our preclinical model. But that alone doesn't get you a drug, because 741 doesn't kill bacteria, and that's where this comes into being a fully adjunctive approach. And really what we wanted to try and do was leverage, as I said before, gram-positive ad sets that John mentioned earlier on. And I'm just going to show you one example here for time. We've shown many, many examples at various conferences over the last couple of years, but I think this points out to where the limitations are from a purely gram-positive deployment standpoint. So this is azithromycin. We've done a lot of work on azithromycin. It's a macrolide antibiotic. It's used extensively in CAP. In combination with 741, again, this is just a very brief amount of data. You can go from MICs. This is a large panel of UTI pathogens collected by JMI. They're all genotyped. You can take azithromycin on its own, it's completely inactive. Add 741, you can drop the MIC 90 and 50 down to well below its current breakpoint in its indication. So very, very nice transformation of activity, to use John's term. You can see the same thing happen in vivo models, the Klebsiella 24-hour neutropenic thigh infection model, where you can essentially take inactive azithromycin, inactive 741 versus growth control, and drop it down to below static levels. So wonderful translation in vivo. But where do the problems arise? So the first problem, and one of the things that are the hardest things to overcome, is adoption. You're now taking not only, to John's point, colistin, which is wonderfully active broad-spectrum gram-negative antibiotic, and combining with something else to transform it and leverage that chemotype. You're now taking 2 completely inactive antibiotics in a setting of a gram-negative infection. And you want to deploy that now into the setting of a serious ICU hospital-based infection. So adoption from that standpoint is for this concept of taking a gram-positive asset and transforming it into a gram-negative is one of the highest hurdles that we've had to come up against. But then specifically for azithromycin, there's a bunch of other challenges. So again, being a gram-positive antibiotic, there's no idea and no way to really characterize what its incidence of resistance is in gram-negative bacteria because it's never been used there and it's very hard to study in that setting. When you combine it with 741, allow it to get in, and you can then understand resistance rates, it turns out that it's actually pretty high. There's actually some really nice posters at ASM Microbe just last week with the incidence of cross-resistance of polymyxin B resistance and ERM and MPH resistant mechanisms to macrolides is actually very high. So that's something you're going into this you don't expect, and then you find as you start to work through it. Azithromycin is known to have very poor, and macrolides generally poor exposure in the urinary tract. We actually showed very excellent efficacy in urinary tract models of infection. So I think we certainly dispelled this, but would have to play out clinically. You can't use azithromycin in bacteremic patients, which is about 10% of all UTIs, so you're taking a hit there. There's relatively high rates of phlebitis as a monotherapy, and then you combine on top another IV-administered antibiotic. It's only approved for short durations of therapy before oral switch, normally 3, less than 5 days. And so applying that now to even UTIs where the duration of therapy is longer, HAP/VAP certainly much longer again. And as I said before, adoption is probably the biggest hurdle in terms of 2 completely inactive components being used in that setting. This is 741, so I think this is just one case study. There are obviously many, many other things we've deployed in combination with 741. I think where we started out not intending to get to and where we've shown a lot of wonderful data to, especially in a type B setting with the FDA, was restoration, especially to beta-lactams and BL-BLI combinations, especially piperacillin and tazobactam, where you can essentially restore all of its lost ESBL activity purely by getting more of it into the periplasm. So there's very many other assets that could be deployed very well with 741. I think marginally gram-negative, making them more exquisite and broader spectrum gram-negative agents, or restoring gram-negative activity to beta-lactams especially is an avenue that is certainly worth continuing to explore. I've said most of this, and given time, I'm going to have to blow through most of it, but I think, certainly for this and certainly for any novel or adaptive approach or a novel approach to gram-negative therapy, it's really identifying what your killer experiments are. I think generally that's the case, but here especially. I think, certainly we had that with tox, identify that tox is going to be the liability of deploying a polymyxin in any avenue, especially as an adjunctive approach. And we did that, and I think we're able to overcome that very early on. And in vivo translation and PK/PD are always going to underpin any approach to the clinic for any kind of therapeutic, especially gram-negative. And our CSO has a great saying: if an experiment's not worth doing, it's not worth doing well. There's no point spending 2 years trying to understand something that's going to have zero relevance to you translating that into a clinical asset. And my last point is polymyxins can be safer. There's obviously a stigma associated with them, and a well-justified one. They are horribly toxic molecules. But we've done a lot of work over the preceding 3 years, and there's a lot more to show over the coming 6 to 12 months that you can do this. You just have to do it the right way. So thank you.
Okay, thanks, Troy. Our next speaker is Greg Mario. It seems like a lot of Gregs unusually for today. President and CEO at Taxus Pharmaceuticals.
It's a pleasure to be here. Thanks for having us. It's such an important space. At Taxus, we're making good progress towards our goal of reducing and possibly eliminating the threat to societal health of multi-drug resistant bacterial pathogens. Our science is focused on the disruption of the foundation of bacterial cell wall architecture to address elemental forms of drug resistance mechanisms that exist across a broad spectrum of pathogens. We have 3 programs that look very promising. The first is efflux pump inhibition. This is not a new target. There has been work done previously in the space, mostly on eukaryotic applications for cancer, but we've made some significant breakthroughs and have achieved where others have failed. FTSE modulation, this is a way of killing bacteria by not allowing them to divide. This is our most advanced program. We have a narrow spectrum oral and IV anti-MRSA drug candidate that's entering the clinic next week. Exciting times. I can now say that Taxus is a clinical stage company. It's taken a while. It always does. And MREB modulation, the most close human homologue is actin. Very promising but earlier stage. In the interest of time, I'm going to focus on the efflux pump inhibitor platform. So these are not bactericidal agents on their own. These are adjuvants. A good, I guess, analogy would be to beta-lactamase inhibitors. Totally different mechanism of action, but essentially we can resurrect the activity of some of the most widely prescribed drugs on the planet. So far, 28 antibiotics that no longer work or require very high doses to work. We can bring them back to the market and make them work again. Over time and exposure, these drugs have lost their potency. By knocking out the efflux pumps, we essentially bring them back to life. And as Tony was talking about, it's tough enough to get into a bacterial cell wall, a gram-negative bacterium, in the first place. And these efflux pumps act as bilge pumps, like on a boat. So foreign materials are swept up, including antibiotics, and kicked out of the cell. And the only way to overcome that is to, like Spiro is looking at, enhance permeability. But otherwise, you would just have to overwhelm those systems with high doses of drug. And of course, you get toxicity. So this is a nice opportunity for applications in stewardship where we have the potential to resurrect drugs that are already on the market, and we can apply them at a very significantly lower dose. And those 28 antibiotics come from 10 different classes. So it truly has the potential to be a platform play. Macrolides, cephalosporins, monobactams. We have a whole host of in vitro data. I'll share a little bit of it in the interest of time. But what's really critical is the first hurdle, and that's animal efficacy. And we, for the first time in this space, an industry first, have durable validated in vivo efficacy in the wild-type Pseudomonas model of infection. We have both broad-spectrum and pathogen-specific efflux pump inhibitors. It's clear that our targeting has specificity with respect to the R&D class of efflux pumps, primarily MexA-B, CD, EF, and XY. We don't know specifically that mechanism yet, but we're working on it. So this is an experiment. Actually, I'll go this way since the mic is turned that way. TXY842 is one of our efflux pump inhibitors. That's a broad spectrum, and it potentiates a macrolide against multiple gram-negative pathogens. These are side-by-side comparisons, 4 different pathogens, and side-by-side comparisons, the macrolide by itself, and then the macrolide in combination with TXY842. And you can see the significant enhancement of potency, where 64 microgram per ml, that's extremely high. That's not effective. And then when you combine it with our EPI, you have a 512-fold increase in potency. Quite remarkable. And you can see the similar effects in the other pathogens, E. coli, Pseudomonas, Acinetobacter, and Klebsiella. This is an example of a pathogen-specific efflux pump inhibitor, 9155. This is one of our most advanced leads. And you can see here that the effect is only seen in Pseudomonas, where we have a 32-fold increase in potency of the cephalosporin and no effect in Klebsiella and Acinetobacter. So we have the potential here for broad-spectrum agents as well as narrow. And again, in vitro data is very meaningful. It's very important. But the key is in vivo efficacy. So this is one of many experiments that we've run. We're looking at efficacy of the combination of TXY9155 with a cephalosporin in the murine septicemia model of infection. The vehicle is the dark square, and the cephalosporin by itself is the white circle. You can see that those two elements, they act in concordance. At 24 hours, all the subjects have perished. Unfortunately, over the 5-day study, none of those subjects recovered. That was some dark humor, just to see if you guys are with me. Anyhow, the good news is when you combine the cephalosporin with TXY9155, we see a very robust efficacy signal. In this case, 75% survival, and it was durable through the 5 days. When we give a second treatment at 24 hours, you can see that we have 100% survival over 5 days. So very critical. We know that if you have efficacy in the animal, you know you can get it in a human. The real question for us is toxicity. And certainly in this space, toxicity was one of the biggest issues. Clearly, we've overcome the gross toxicity of the past efflux pump inhibitors because it's not overcoming the efficacy signal. So it's very encouraging. So there are 3 ways to apply this technology. The first is a generic strategy, very critical. This is a way that we could provide life-saving medications in a cost-effective way across the global community, because the threat of multi-drug-resistant infections is already upon us. It just hasn't reached our shores. The best way to address it is to take it overseas and go to those underdeveloped countries and provide low-cost access to life-saving medications. And in fact, in this case, it's the same medications that they've been used to using for decades. We can just repurpose them and make them work again. So you talk about antibiotic stewardship, that would fit right into it, right? The second way, which is a little more exciting from a profit motivation, is branded life cycle management, much like the beta-lactamase inhibitors. Amoxicillin stopped working decades ago. It's still prescribed widely because most ear infections resolve. But you add clavulanic acid to amoxicillin, and voila, you have a new branded drug, Augmentin. And lastly, place that we haven't really looked at too much yet is R&D enabling. You can imagine that there are a lot of antibiotics that have failed in clinical development due to dose-limiting toxicities. Well, we can repurpose them, dust them off, lower their dose significantly if we have that synergistic effect, and push them through and perhaps get a lot of those drugs on the market that otherwise couldn't make it. The challenges are first in class. Like the panel before with ephages, it's exciting. It's cutting edge, but it can be bleeding edge. It's very difficult. I always like to say in this business, I like to be number 2, the second. Someone else will cut that path because there's an awful lot of risk aversion, and that's a good reason in the agency, because patient safety is number 1. But those things can be overcome. It just takes a little longer and a little more hand-holding. This is a platform technology, and that's a blessing. It's exciting. There's so many different ways we could apply it to so many different antibiotic classes, but we're a small company. So in some ways, it's a bit of a curse. There's so much that I would like to be able to do, like I used to be able to do when I was in a big pharmaceutical company, which is why we're actively engaged in discussions with pharma companies to see if we can carve up this project in some way, shape, or form among us. But the biggest challenge is, and we know this, it's always stated, in fact, in the panel earlier, besides the economics, what's the biggest challenge? Well, the biggest challenge is economics. It doesn't make sense. Even though antibiotics as a category are a fantastic category to invest in relative to others, because the numbers don't lie. The rate of clinical success is the highest of any category. Phase 3 success is higher than anything by head and shoulders, almost 2x times the average of other new chemical entities. So why is it that pharma companies wouldn't be interested? Economic incentives have been put in place, the GAIN Act, very meaningful. We received qualified infectious disease product designation from the FDA for our TXA 709. Very meaningful, additional 5 years of marketing exclusivity. Carbex, hallelujah, thank you to the team, Joe, Chris, Tony, and the rest. Very important program that also brings more private investment along with it. If you've been watching some of the companies that get this funding, they often go public or raise significant amounts of private capital. We are in the process of applying, just put our first expression of interest forms in last week. But the market dynamics will not materially change without pull. I'm not a bioethicist, but does it make sense that we spend hundreds of thousands of dollars on medicines that can extend the life of an octogenarian by 3 or 4 months, perhaps? But for an antibiotic that might save that octogenarian's life when he or she is in their 30s or 40s, we pay a fraction of that amount, 1/10 or even less? The answer is no. This is not a good time, it never has been, but especially these days, thanks Martin Shkreli, to be advocating for higher drug prices, right? Not so easy, but guess what? We have to. Chairman Gottlieb came up with a very interesting approach, a subscription model for antibiotics, We'll see how the hospitals respond to putting money out in front. I don't know, but at least that takes some courage, right? But we have to go further. It's the right thing to do. It's not popular, but we need to have courage and conviction and stop ignoring that elephant in the room. Besides economics, no. It's the #1 issue. We have to raise the prices, incent pharmaceutical companies to invest in the space. Otherwise, we'll lose it. And if we lose it, infectious disease management is the foundation of modern medicine. We go back to the days when we lived only until we were 40 or 50 years old. Not to scare you, but it's real. Hopefully we'll do something about it. Wish us luck. Thank you.
Great, thanks, Greg. So I'll go ahead and invite Troy and Greg back up to the stage, and also two additional panelists, Ed Cox, who was introduced before as Director of the Office of Antimicrobial Products at CDER, and Vance Fowler is Professor of Medicine and Professor of Molecular Genetics and Microbiology at Duke University Medical Center. So Ed, why don't I go ahead and give you a first chance to react to the presentations that you've seen and give some of your thoughts.
Yeah, great. Thanks, Greg. And thank you very much for the presentations, and thank you for working in this field and trying to push forth the frontiers of science. I think that's great. My comments will be not only, they'll be more general, because I think there's a few things that are probably worth talking about that I thought I'd share, at least as I think about this. I think mechanism is very important. And I'm a very strong believer in basic science, because I think that's where the new innovation is going to come from. It's going to come from our understanding of biology and being able to leverage that and to move forward. But I think as we start to think about products and we start to think about benefiting patients, it's important to move beyond mechanism and to start to think about how will this mechanistically exciting molecule that we have benefit patients. So that's it's something to always sort of keep your eye on the ball, so to speak. Where is this headed? Let's look down the road a little bit. If we look at antimicrobial drug development now, we do pretty well with animal models and preclinical data to help predict the likelihood of success. But even with all the work that we can do with traditional small molecule antibacterial drugs, a field that we know and understand pretty well, clinical trials continue to teach us new things that we didn't expect. So I think it's important to keep that in mind. We learn from these lessons, but I don't think that biology is done teaching us things that we haven't understood yet. So we just have to sort of keep that in mind as we're working through this. I think it's been interesting across the presentations. We've heard some of the ideas about where do you start? And the idea of, can you start somewhere where you can do a clinical trial to look at clinical activity in humans in a situation where it's still sufficiently safe? And where you start, I think, depends a little bit on the confidence in the molecule, the experience with a particular class of molecule. If you're doing something really novel, you may want to start somewhere where there's still, in essence, a safety net or a rescue to be able to show some initial activity. And then there's also practical issues. Depending upon what your molecule does, how large is the patient population? What can you do that's feasible initially? And depending upon the molecule, for certain molecules, you may need MDR pathogens. For others, you may not. So that may help you if you don't, even if your product is one that you see being clinically useful in patients with unmet need, if there's an acceptable safety profile, you may be able to jump in and do something that isn't extremely difficult to do right off the bat. A couple of other concepts to think about. It's hard to improve upon 100%. So if existing therapy is already highly, highly successful, to do better than existing therapy is going to be hard. So think about where there might be patient populations where you can actually show improvement, patients where there are needs to do better, diseases where there are needs to do better. And think about also the possibility of enrichment, because enrichment may help you get to a studyable population where you can actually demonstrate the effect of your drug clinically. And I think, there are differences in some of the molecules that are coming forward, but I think there's also a lot of important lessons to be learned from the experiences to date. We've seen a wide range of different products. We've seen a wide range of products for different diseases. We've seen, various different approaches, some of which have worked and some have not. And I think there are some higher level concepts that can be translated to, different and novel molecules that are helpful and can help to avoid certain problems or pitfalls that we've experienced in the past. So I'll stop there, but those are just a couple of the things that I've been thinking about as I've been hearing the wonderful presentations we've heard.
Vance?
Yeah, sure, thanks. Appreciate the opportunity to speak today, and appreciate the opportunity to hear more about two really interesting compounds. Both of them were fascinating, and I totally agree with Ed about the fact that science is ultimately going to guide innovation. Three thoughts succinctly that would be, I think, of importance considering how to develop these sort of restoring compounds. So where do they go? Broad versus narrow, and then the issue of number of patients and potential for difference, sort of an inverse relationship there. So with regards to the first element, where does it go? And I think this is going to be an important thing that, you know, has been touched on before about PK, PD, and the fact that there are going to be two elements that we're going to be tracking. If you can get one to the cerebrospinal fluid, for example, but not the other, you run the potential for loss of the potential efficacy that you had hoped to achieve. So I think, and this is going to need to be clarified, to my view, it's going to need to be clarified in more than the conventional platforms, because, you know, the reality is that when these compounds become available, people use them off-label. No big surprise to this audience. In fact, I'd say the majority of use for these compounds are often off-label. And so having some clarity as to where these various compounds go in various sanctuary sites, if you will, is going to be important for responsible use as we move forward. Second element, broad versus narrow, I was really intrigued by the fact that these compounds have the prospect to be simultaneous, to essentially have a portfolio of those with broad activity versus those with targeted activity. And the example was with the Taxus Pharma element of, I think it was 842, that was sort of broad element as compared to the Pseudomonal 9155 that was targeted. When I started thinking about with regards to that strategy and how that could improve some of the current stumbling blocks for getting drugs approved in registrational trials, particularly in gram-negatives. And the big element there is prior antibiotic therapy as an exclusion criteria. So for example, you know, in terms of HAP-VAP, obviously an important pathway that is commonly employed in this space, single greatest, you know, reason for exclusion of patients into that space is prior antibiotic therapy, more than 24 hours of potentially effective antibiotic therapy prior to enrollment. Big issue, it's been published earlier this year in CID that, you know, the cost of those trials is on the order of $90,000 per patient enrolled. And so the structure could then conceivably be you have a broad spectrum sort of restore agent, to use John's star analogy, until you have the luxury of data in terms of what you're actually treating, and then you could target it thereafter. So in this instance, sort of the 845 target narrowing down to 9155, for example. And so why would that be important? That'd be important because it would allow the enrollment of patients earlier in study before you have the luxury of data, and thereby facilitate patients in study. The final one is the inverse relationship between the potential to show difference in the number of patients involved. We heard a comment, I think it was John, that mentioned endocarditis. Tough place to do a study. Whew, tough. But the mortality that you see there, you can account totally to the disease state. And there are specific microbiological characteristics. Staph aureus is a pretty big player there. You can target a staph-specific agent in that space, and you could ultimately identify the event rate's going to be high enough that you can show a difference. So how you balance that, how you balance the need for adjunct therapy, cardiothoracic surgery, referral bias, expertise, regional expertise, geographic variation. I mean, we did, when we did the ICE study, International Collaboration Endocarditis, there were significant differences in how Staph aureus endocarditis, left-sided Staph aureus endocarditis, presents, is managed, and ultimately fares.
by geographic region. That would not have been intuitive to me. So I guess in summary then, that, you know, thoughts about where the compounds go, both of them, the broad versus narrow, and the fact that we needn't necessarily have to choose both of them, or, you know, either of them. It need not be an either-or scenario, but maybe it's a both-and. And then finally, the balance, careful need to balance, you know, the potential to show benefit versus the difficulty of enrolling the patients. And I'll stop there. Thank you.
Great, excellent questions to bring back to the these two presenters. So maybe I could turn to Troy and Greg to either, you know, respond or react to anything that Ed or the sort of three major areas that Vance outlined.
I think that, you know, as you had mentioned, Ed, the challenge is, you know, to focus on unmet need. If something already works, you know, why bother? But Vance, you put it pretty clearly. When you get to places of unmet need that are significant, you find very difficult clinical trial design issues and conduct issues. You know, ultimately, my personal goal is to next prove in vivo efficacy in an animal model of infection with Acinetobacter. Because if we could cover both Pseudomonas and Acinetobacter, then we really have a great shot at a HAP-VAP drug. And right now, you have 50% mortality in that space. So it's so meaningful, but try to recruit patients, right? And then try to separate the signal from the noise with all the comorbidities associated with many of these patients. You know, it's a real slog. So, you know, we have to be brave. You know, we have to go after the tough targets. And then hopefully along the way, we'll get some relief. You know, perhaps breakthrough status, you know, a single phase 3, and then post-marketing surveillance, you know, something that was done for the HIV drug development effort. Additional economics is definitely critical, you know, much like the tax program that was put in place for HIV, where you could write off your development costs. You know, then people will start paying that kind of money for these giant trials that, unfortunately, many of which will fail. I just think I can't get away from the economics on this at all. It takes courage and conviction. We have to get out there and get prices and reimbursement much higher, at least in the institutional setting. Otherwise, we're going to be talking about this until millions of people start dying, and then we'll play catch up, like we usually do, unfortunately.
Troy?
Yeah, no, no, go ahead, Ed.
Do you want to jump in on that last?
I was just going to say, yeah, you know, just to Greg's comments, I mean, you know, certainly we'd love to see more drugs for HAP-VAP, and it's an area where, you know, it's a very serious infection, and it's an area where we've seen some drugs that have not, you know, have fallen down with regards to efficacy. They haven't done quite as well. It is challenging, though, and oftentimes when we think about HAP-VAP, it may be not your first indication, but it could be a follow-on, because you may pick to do something that's not quite as challenging to start out with. There's significant challenges in doing a HAP-VAP study. So there's certain, you know, there are two approaches. I mean, one could be to, you know, to jump into HAP-VAP. You'd have to have enough confidence in the compound. It's a serious infection. You know, want to make sure that you were ready for it. And the other is it may not be quite as high a jump initially. Would be, you know, a more, you know, a somewhat less difficult indication to study. So it's just something to think about. And I'll throw one more thing out. This is a topic that's come up time to time, but it might be interesting to hear too. You know, when we get to these difficult indications, one of the ideas that's been out there to try and help the field, recognizing the economic challenges, the scientific challenges, has been that of clinical trial networks. So that's come up before. I'd be curious to hear any panelists' comments on that too as we're working through this.
Troy?
Yeah, so I might go back to a couple of the earlier questions that Vance raised. So I think, you know, certainly I understand the argument of, you know, why go into a patient need that's already been met. And certainly it makes a lot of sense when you're talking about, you know, the current multiple B or BLIs all running through the same UTI pathway, where essentially they're demonstrating efficacy against mostly wild-type pathogens. I understand, you know, continue to go down that path with that chemotype and that mechanism of action doesn't make a whole lot of sense, but certainly leveraging other mechanisms of action to account for future resistance development against that mechanism of action, I think, is really important. So, you know, I certainly think there's a path forward for a novel mechanism of action, novel approaches to go down a pathway that may be perceived as a met need, even though I think it can certainly be extrapolated further out to have that where the unmet need is much greater. In terms of you know where something like potentiators would be positioned, in terms of narrow or broad spectrum. I think certainly that's the other challenge of combinations, especially, well, it's not so much a challenge, it actually is quite a benefit for potentiators, is that it's not mechanistically driven, it's not attacking specifically a beta-lactamase, for instance. It's much more of a macromolecular phenomenological approach where it's interrogating the outer membrane. And so a spectrum of activity is defined. It essentially can target all of the gram-negative bacteria. Its spectrum, ultimate deployment spectrum of activity is much more dependent on the partner antibiotic, and it really allows you to tune that approach based on the profile of the partner agent and its resistance frequency and resistance profile and current clinical outcomes. So, I think there is a much more tunable approach of being either broad or narrow, but very much dependent on the partner.
So any thoughts on the, Ed, you brought up sort of clinical trial networks and potential opportunities there, views on how that might help in some of the challenges with recruitment and small trials?
Yeah, no, I think it's a challenge. I mean, certainly being able to leverage the network and being able to work pretty competitively would be a wonderful thing. I certainly think there's a lot of people going down, a lot of companies going down exactly the same path of clinical development, whether they be CUTIs, which is obviously essentially where most of the clinical development is being done at the moment. And so certainly there would be, it'd be a great benefit to be able to leverage a lot of the same clinical trial sites and enrollment statistics and distribution networks in a pretty competitive sense. It's tough for that to play out realistically in a competitive commercial setting that we're in, but I certainly would love to see how we could leverage that in a much more effective way than we are doing right now, which is essentially not at all.
Yeah, go ahead.
So it seems like, too, both Troy and Greg, for your products, too, there's a certain amount of knowledge that you would like to, that would be helpful to you as you're thinking about your clinical development program. The bacteria that a potentiator can actually impact upon and the opportunity to impact upon an efflux pump. So it seems like there's also critical knowledge that's needed to understand the likelihood of encountering patients where your particular therapy can demonstrate effect. Any thoughts on that and types of data that you might be pulling together or need to help you plan for your clinical development?
Yeah, I mean, I think, the one example that I pointed out, which certainly could be a challenge is when you're trying to deploy an asset that's never been used in that setting before, like a gram-positive asset, if you're looking to transform it, there's very little knowledge that can be gained about current resistance rates and where it would be most useful. So, certainly that data is very hard to get, and I think some of the data that we've leveraged and some of the great work that some of the organizations are doing, like JMI Labs, is essentially every clinical asset that comes through their doors these days, they genotype. So they have a thorough understanding of the genetic background and resistance mechanisms that exist in all the clinical assets that they can collect. So, we've certainly leveraged that data a lot to understand the spectrum of activity of the combinations that we've assessed, especially when they're on the gram-positive side in terms of the partner antibiotic, where it's very hard to get that information on your own without being able to deploy it as a potentiated therapeutic. So certainly I think that's very helpful. That's much more on the preclinical side, but it certainly helps guide us towards what our TPP could ultimately be for any one of the combinations.
I don't think there's any specific data per se. I think it has more to do with finding the sweet spot with respect to our combinations. Which products would be the best to pursue is hard to say. The data will drive it, obviously, but we've decided to choose cephalosporins since they're pretty benign, relatively speaking, and very widely prescribed. And so far we've had great success with them. The data that I would love to have is mechanistic. And we're working on it, but we're not putting a lot of time and effort in. I think, Troy, you had mentioned, don't spend a lot of time figuring something out that, might not be that important, right? It would be really meaningful to us to know that, but as long as we have efficacy, we're going to keep pushing forward and see where we can, find clinical utility. But clearly, since there's specificity and there's also a broad spectrum effect, it has to do with the target efflux pumps. And we know that we're hitting efflux, but we can't yet figure out which pumps, just because I don't think we have the right strains. So we're actually making them ourselves, and with the help of folks at Princeton. I believe the strains that have been floating around since Olga first created them are tainted. But but it's very expensive to do that kind of work, and it's very time-consuming. So, again, it comes back to economics and having money, or at least partners, who would be willing to help us with that.
Vance, go ahead.
Got a comment related to the clinical trials networks, and I think that precisely for the matter of economics, that it's an inevitability. I think that there's so much that's particularly, and John has written a really nice prototype that's been picked up by several funding agencies in terms of mechanism, a potential mechanism by which it would occur. But there's so much on the clinical end of getting patients into study. First of all, not all sites are good for all trials. If you want to do a skin and soft tissue, an ABSSSI trial, for instance, you're going to talk to a fundamentally different cadre of investigators than you would if you're going to do complicated UTI or if you're going to do HAP-VAP. And that's driven by the interest of the investigator. It's driven by the expertise. It's driven by the particular philosophy of the institution, and a whole bunch of intangibles that you have to ultimately capture. So a syndrome-specific clinical trials network is where we need to be, full stop. And this is not just sort of theoretical. I'll give you an example of what we just finished doing with the Antibacterial Resistance Leadership Group. It's in the diagnostic space, but it's a concept of fundamentally of employing, sort of dual, all of us are better than any of us kind of concept. So this is a thing called Mastermind, and the particular disease state was extragenital gonorrhea. So the specific, what we've completed doing, enrolling 2,600 patients into the study, is bringing, essentially allowing one patient to simultaneously inform more than one diagnostic platform, such that we brought multiple companies together, developed with an incredibly collaborative FDA, developed a gold standard by which all of those individual diagnostic platforms could be gauged, and then did the trial, and then generated the data that can then ultimately be used by each of those individual companies for the support of their own 510K submission. And so that study's done. How could this happen? Well, one way that it could happen is if the involvement in a clinical trials network was tied to funding through, for example, BARDA. In other words, if you want the money, you're going to have to use our clinical trials network. And my guess is that would enhance the participation in such a network considerably.
Absolutely.
I'll stop there.
Any other comments on that? Okay, so we have time left in the session for audience Q&A, and as well as those who are dialing in from the web. Any questions or comments? Okay, John.
So, good stuff again. Just quickly on the trial network, I'll just say that I am working with Wellcome Trust to build a business case to implement a standing trial network for complicated UTI, complicated intra-ab, and possibly nosocomial pneumonia that would do kind of what you've talked about. It would be a constantly running trial with a constant backbone of a good comparator like a carbapenem, where you could bring drugs in, and we believe that actually would reduce cost and time for any drug that wanted to be evaluated by approximately 40% if we can put the network together correctly. Not cheap, but cheaper than everybody doing it individually, and you could put diagnostics and all kinds of other stuff on top of it. So that's not my question. My question is really to Ed. You made a comment that I would like you to talk a little more about, which was that you said that even in the case of well-known trial designs with small molecules, we're still encounter surprises, right? And I've heard you say that before, and I think I heard it a little differently today. I think you're pointing at the notion that don't get too confident in your ability to work with variations on well-known pathways, because even there we don't, we still get surprised that there's a need still to generate something close to standard data sets for novel agents. But maybe I'm interested in having you talk about that.
Yeah. So there's at least a couple of comments in there. So yeah, Samadhi and I have this slide and we keep adding to it. And what this slide, it's titled something that says like clinical trials continue to teach us important lessons. So obviously nobody goes into phase 3 with the wrong dose or takes a molecule into phase 3 that they think is not going to be successful. Everybody goes into phase 3 with the most informed dose that they can possibly come up with and trying to get a molecule that they're most likely to be successful. And that's great, and that's what you should do, and you should do the PK/PD, and you should try and increase your chances of success, and we believe in those tools, and we think that it's the right thing to do. But what we've seen, and this is not surprising given what we're dealing with here as biology, is that sometimes, despite all those efforts, clinical trials teach us something that we didn't expect. And it's interesting, if you look at those trials that didn't work out, and then after the clinical trial has, shown a deficit in efficacy or, whatever the issue may be, sometimes you can go back and figure out, oh, here's what we missed. And you promise yourself you'll never miss it again. And sometimes you do, and sometimes you don't. But that's another issue. But yeah, so we try and learn, and sometimes you can figure out what went wrong. Sometimes it's interesting, you can't. And so there's almost sort of there are some instances where you can anticipate, or some of the things are avoidable, I guess, is what I'm thinking about, because maybe you could have maybe you could have learned something ahead of time, but other times, even despite your best learning and your best attempt, you can't quite figure out why things went wrong. So, I guess what I'm saying is that biology is complicated. Humans, sometimes are a little bit more complicated than animal models of infection, and so clinical trials remain a very important test to try and figure out what the effect of a product will be in humans who have an infection. So it's just important to keep that in mind and do all that you can with PK/PD to try and increase your chances of success. Do all that you can with animal models of infection to try and increase your chances of success. But sometimes, we're going to be surprised, and that's just the nature of biology. And so that's one. The other is that, yes, in areas of unmet need, if you look at the drugs that have come forward, that have, the sort of the basic properties, the capacities to be able to treat particular infections where we need new therapeutic options, where there's unmet need, we've been trying to find ways, and we have found ways to streamline those development programs. So you may see that we're willing to take on greater uncertainty. You may see wider non-inferiority margins, sample sizes that are smaller, safety databases that are less than what you'd expect for sort of a standard development program. And the idea is if you have an area of unmet medical need, that's an area where patients need therapies. Physicians, society in general will accept a greater degree of uncertainty around a compound that really addresses an area of unmet need. And this is actually described in our Subpart E regulations. So it's a concept that's been around for a while. So if there is a product that meets a particular unmet need, we are working with developers to try and find streamlined pathways to try and bring those products to market using a more streamlined development approach. That may be, depending upon the developer's intention, that may be sort of the first way, or the first indication that a product seeks, and then oftentimes there will be subsequent studies that follow to study the product in additional indications. So I think those are sort of the two major themes I was getting out of your question, John. Hopefully I got them both.
Great, thank you. Barry?
Yeah, hi, Barry Eisenstein, Carbex. Terrific discussion. I've got 3 quick questions that may take an hour to respond to. One of them dealing with PK and distribution mismatch or potential for that. The second is the potential effect of these very broad spectrum, potentially broad spectrum agents on the normal microbiome. And the third is the potential use of a potentiator in a more general way to match up with a whole bunch of different antimicrobials beyond just the one that was initially studied.
Yeah, so I can answer for potentiators. So I think certainly that's one of the advantages to the chemotype that we've actually come across. It's fair enough, it's a polymyxin base, but removing a lot of the positive charge and lipophilicity actually allows it to behave much more like a small molecule. So whereas polymyxin B and especially calistine methyl sulfate have highly heterogeneous PK and horrible distribution into the renal system and lungs, 741 is actually exceptional. So it's excreted about 50% intact in 24 hours in the urine, which, to get to your third point, overlaps very, very nicely with a lot of antimicrobials which are renally cleared. Its lung penetration is wonderful. We see excellent in vivo, obviously preclinical murine lung efficacy for a variety of different combination partners. So it behaves much more like a small molecule. Its clinical PK is perfectly linear and dose proportional. So we certainly went into that knowing that that was something we had to fix along with the toxicity. And certainly took a lot of work and a lot of optimization, but it's absolutely something that we think we can leverage, especially as a combination. And I think to your second point, that's a little bit tougher. I mean, certainly having renal clearance and having an overlapping compartment in terms of localization of drug is obviously essential for a combination. But I think certainly that varies quite disparately across the antimicrobials. Tetracyclines, beta-lactams, macrolides, fluoroquinolones all have different dispositions, whether it's respect to renal clearance or lung penetration. So I think, yeah, certainly you have to optimize that. Fortunately, there are multiple members within each class that have differing PK profiles that you can sort of tune in on. But that certainly is one of the preclinical challenges in identifying a partner. It's not just about an MIC. If it was about MIC, we'd have 50 drugs under our pockets right now. I think it's, MIC is the first part. Translation of that into efficacy, which requires PK and overlapping PD markers or drivers, is obviously essential.
Yeah, I can't add too much to that, just with respect to what we're trying to do to overcome these challenges. You saw in that one example the efficacy in vivo in that particular experiment. Clearly there was a PK issue because we got complete responses only after a second treatment. So we're still very early in that process of optimization. And my guess is that it's going to shake out and we'll find specific drugs within each one of the categories of the 10 classes of antibiotics that fit with the EPIs. It's going to be trial and error. It's a real long slog. From a technical standpoint, I'd say that's the biggest challenge that we have. If you're talking about in the IV, the ICU setting, you got a lot of flexibility, but ultimately we want oral meds, right? So, how do we co-formulate them? Do we build them in a kit? If you build them in a kit, good luck. If you have to take a blue pill 3 times and a red pill 5 times,
Yeah.
So it's not trivial at all. It's an excellent question, and it is the one thing that I believe is the biggest technical hurdle for us, is finding that sweet spot, particularly on the oral side. IV, we can probably figure it out. We'll get away with it.
Any other comments or thoughts on that?
Maybe I'll just echo some of the things that Greg's talked about. I mean, I think there's a scientific question. Where does the drug go? What levels does it achieve? In what tissues? How do you sort of deal with that? But it's interesting, Greg's also keyed into the issue of the practical issue. How is this going to work in the real world? If there's 2 components and they need to be given at various different intervals or simultaneously, is somebody going to forget to give one of the components and those sorts of things?
Yeah.
Or is it, so it's interesting that I think there are both scientific questions or scientific questions or experiments that can help to address this question, and then there's also sort of the practical or pragmatic issue that you need to think about as you're working through all this.
Yeah.
So appreciate your insights, Greg.
Ultimately, if they don't use it the right way, we're going to fail, right? Because even in a clinical trial setting,
Barry, do you have a follow-up? Oh, the third question, which...
Yeah, so I think, that's a question we've tried to understand internally and also through interactions with the FDA is what's the appetite for getting a potentiator? And it's a very similar conversation with beta-lactamase inhibitors that I'm sure companies have had of, clearly there's a broad applicability to both of those mechanisms. Most beta-lactamase inhibitors are probably going to work with every carbapenem, beta-lactam, cephalosporin that's out there. You're obviously going to want to optimize for PK, and I think that to sort of echo what I think we've heard mostly from regulators is that when there's much less control and much less data to support how those two things need to be combined to be optimized for effect, you're much more reluctant to just let one loose out there and say, go combine it this exactly the same way you did it with this one, and it should be fine. I think certainly when you just look at the different ways that even carbapenems are dosed, there's some that are Q6, some that are Q8, some that are QD, right? And so having a single beta-lactamase dosed a single way to perfectly optimize for that carbapenem, or those 3 or 4 carbapenems, is challenging, right? So I certainly think that you could do a lot of underlying PK, PK/PD, and in vivo efficacy to support broader use without a dedicated product. And there's certainly things, clearly things you could do, and you would have to have discussions with regulators on how to deploy that. But I think that would be some of the challenges in just trying to make sure. I mean, certainly from the way, gram-negative therapeutics are used now, they're empirically used as combinations, as layering of antibiotics throughout, someone's progression through the ICU when they enter, until we figure out what bug it is, till we figure out what resistance mechanisms they have. There's going to be multiple iterations of layering of antibiotics on top. None of them optimized necessarily to work with each other, how they're being dosed. So I think especially with the polymyxins, everyone thinks of them as being synergistic molecules, but they're never actually used in a way that would replicate that synergy. And I think coming into this is how we wanted to do that. We're looking at a polymyxin class, wanting to leverage that synergistic mechanism and do it in a way that's actually founded by science, so people can do it the right way, rather than just empirically. So I think it's a great question.
Yeah, I kid my drug discovery team. We've got great chemists, that have gotten us to this point. But I kid them. I say, "Can you just give me a single agent?" Why do we have to do all this combination stuff? All kidding aside, the complexity is, I think, sufficient enough, at least for our company. We'll focus on single combinations, if you will. Looking at cocktails, I think to Troy's point, would be more for the empirical researchers out there, or perhaps for a larger pharma company that would have the resources to dig into it. But I think we have to get the proof of concept, stay very focused, pick the first pathogen, which is Pseudomonas, get something that's clinically relevant, and get the ball rolling. So I'm not even close to that.
Yeah. And just thinking about it again, I think it comes back to really sort of, the scientific questions. If it's something that's going to potentiate, the degree of potentiation that you might need for one, parent antimicrobial may be different than what you'd need for another. Like for beta-lactamase inhibitors, I mean, the degree of stability of the beta-lactam itself may vary. So, the quantity of, a particular beta-lactamase inhibitor that you need might vary. So these can be some fairly complex questions to try and address. So how a potentiator or inhibitor is going to work with any one particular parent molecule could vary across the different parent molecules. And I haven't even started to mention, that if the parent molecule is given Q6 versus the inhibitor is intended to be given twice a day. I mean, how do you sort of manage all that? So, because the relative concentrations of the two, you would hope there's probably some ratio that you're trying to hit there. There may be ways to address all this, but again, I think it all comes back to the science and understanding the molecule, understanding the potentiator or the inhibitor, whatever the category of molecule may be, how those two things interact, and picking a reasonable way, again, trying to increase the likelihood of success, of the combination therapy.
Okay, great point to end on. So that'll round out this session. Thanks to Troy and Greg for your presentations, and Ed and Vance for all of your comments. We're going to go ahead and break for lunch now. Lunch is on your own. We have maps and everything out in the desk for local restaurants. You can always bring your food back into the room if you don't finish, and we'll see you promptly at 1:30 for the next session. Thank you. The next minute or so, if I could ask you all to take your seats and join us back in this room. Okay, welcome back. I hope you all enjoyed your lunch. During our third session, we'll focus on agents that are studied in combination with existing antimicrobials, just like the last session. But now we're going to talk about agents that can enhance the elimination of bacteria. Discussion will encompass scientific and developmental
challenges and concerns. And we have presentations from 3 different companies that are all working on innovative compounds that would fit nicely into this category, and then we're also going to have additional panelists. So let me kick off with our first presenter. Kara Casino is Executive Vice President of Research and Development and Chief Medical Officer at Contrafect Corp. Kara? Okay, there you are. Glad you came back from. I forgot to check ahead of time to make sure you were here.
Okay. Let me see. Here's my thing. Okay. Welcome back from lunch. Always challenging to be the first speaker back from lunch. Many thanks to the organizers for inviting me to talk a little bit about license. As was introduced, I'm at Contrafect. Contrafect is a publicly traded. That's the reason for the forward-looking statement. Our general counsel makes sure we show this wherever. I may make forward-looking statements. Don't trade stock based on them. But at any rate, we're a clinical stage biotechnology company in the greater New York area. We're focused on bringing protein-based therapeutics into the anti-infective arena. Our lead platform is really our Lysin platform, so that's what we're going to talk about today a little bit. I thought I'd introduce the concept of lysins. We heard about bacteriophage this morning. Lysins are a step along the bacteriophage continuum, basically. Lysins are cell wall hydrolase enzymes that in nature, during the phage life cycle, bacteriophage produce lysins to break through the peptidoglycan cell wall of the bacteria and release the progeny phage, which we heard about earlier today. And the insight here that underpins lysins as therapeutics is to isolate, clone, recombinantly produce those cell wall hydrolases, and then medicinalize them such that if administered externally to bacteria, one sees a pretty profound therapeutic effect, rapid lysis, and the potential for lysins to be used as a medicinal therapy for serious bacterial infections. In essence, taking one of the killing elements at the phage's disposal and turning it into a intravenously administered medicine. So we've learned a lot about lysins over the course of our time looking at CF301, which is our lead candidate. And more recently, we're working on anti-pseudomonal lysins. We have a CARB-X funded grant to support our discovery program in the gram-negative arena. Some of the features of lysins that underpin their potential utility as therapeutic agents are rapid targeted bactericidal activity, highly potent ability to eradicate biofilms, much beyond what one has normally, clearly conventional antibiotics don't do much with biofilms, and thus we think lysins have a potential place to work in a complementary manner with conventional antibiotics, partially because of the biofilm activity, but not just that, because we've also observed potent synergy with lysins in vitro and in vivo with conventional antibiotics. So CF301 is an anti-staphylococcal lysin. It's a 25 kilodalton enzyme, highly active against Staph aureus. Interestingly, lysins, because of their rapid killing and their behavior, if you will, in an antibiotic-like way, have enabled us to use conventional antibiotic tools, antimicrobial susceptibility testing, MICs, PK/PD in animals to help us understand efficacy, place in therapy, and use as a potential therapeutic. And I'll show you a little bit of data on why we've chosen to go forward with the Lysin 301 candidate in addition to conventional antibiotics. It is, of course, possible to have developed a lysin because of all these features I just shared that are would allow us to use MIC and PKPD to develop a lysine as a monotherapy. But these features, complementary to, synergistic with, and highly active against biofilms, caused the company to think about where the highest unmet medical need is in the Staph aureus arena. Think about Staph aureus, endocarditis, and complicated bacteremia as a potential indication, and bring us forward in that way for phase 2. Just to give you an idea of what RAPID looks like, I'm trying to advance. Oh, it didn't work. My slide is not working. Hold on, I'll try one more time. All right, that was supposed to be a movie. Can you make it work? There we go. So to give you an idea of what RAPID looks like, this is looking through a microscope at some bacteria. This is actually anthrax. Our team used that because it makes a better movie. They're bigger to see. Drop of lysine on top of the anthrax under a cover slip, and you saw the rapidity of the killing, not speeded up, real time. So some other interesting features, just to run through this briefly. Lysins have low propensity for resistance in vitro, 301, seropassage studies, 26-day seropassage studies, virtually no movement upwards in the MIC. In contrast to daptomycin in this experiment, where you see dapto readily demonstrates the emergence of resistance, add 301 to the daptomycin, and interestingly, you see that 301 is able to suppress the emergence of resistance to dapto. And we've repeated this, this study with other anti-staphylococcal antibiotics. We've also looked at this ex vivo, taking tissue from animals that have been treated with 301 and antibiotics, and we've been able to show a similar phenomenon, which we think is an interesting piece of the use of lysins as complementary adjunctive therapy to conventional antibiotics. I mentioned the biofilm thing. Biofilms obviously are a major challenge to the treatment of certain serious invasive staph infections, endocarditis, complicated bacteremia. You guys know them. Biofilms are not well addressed by conventional antibiotics, and they're one of the reasons why antibiotic therapy for staph fails and why surgery becomes the next last and treatment of last resort to fully eradicate infections. So down here at the bottom, on the side you see a slide with an MRSA strain, avidly formed biofilms on it, and 15 minutes after exposure to 301, you see complete eradication of the biofilm. And compared to daptomycin, the minimal biofilm eradication concentration is obviously highly sensitive to 301 and resistant or no effect from dapto. We published recently a complete review last year by Ray Shook, our VP of Research in AAC, on our biofilm activity. And lastly, I'll just show you this slide, which is one of a number of pivotal, I'll call them, animal studies that the company did to work out how would one bring an adjunctive lysin forward, and could one expect this to work, and at what dose in the setting of infectious endocarditis. So we've done a number of studies in rats and in rabbits. This is actually a rabbit, even though it says rats. Excuse me for that, something happens here. But at any rate, in this study, as in others, this was a rabbit model, we looked at daptomycin dosed for 4 days. Daptomycin alone results in a 3-log drop in colony-forming units. Dapto is the magenta color on the graph. With the addition of a single dose of 301 to that, and here you see in a number of doses, you see at least a 3-log drop in CFUs, and this is in heart valve vegetations, but we've seen the same thing in the kidneys and the spleen, so other target organs of interest. We see an additional 3-log drop on top of the daptomycin for a total of a 6-log reduction in CFUs in this described and well studied, standardized model. Actually, this work is done by Dr. Arnie Baer at Harbor UCLA, who's been working with the company for about 4 to 5 years on a variety of these models. So this has been replicated in various species and formed the basis of the approach into the clinic. We completed a phase 1 study, which was a healthy volunteer study. We chose a range of doses that we thought would be applicable in the clinic. The phase 1 study was a rather boring, as some people say. I don't think any study is boring, but this was a quiet phase 1 study. No serious adverse events reported, no clinical adverse safety signals, a couple of non-serious adverse events, which were more or less split between placebo and the 301 patients, and some evidence of anti-drug antibody developments, which is not surprising given that this is a protein in about 69% of the subjects, which we followed out to 180 days, at which point we found them to be resolved or nearly completely resolved. We noted that ADAs begin to appear at day 14 or 28 and weren't correlated with IgE or markers of allergic hypersensitivity. And phase 1 helped us with the well-behaved linear predictable PK profile, which we were able to use in consort with a large PK/PD model from a number of animal studies to help us figure out dosing and approach for our ongoing phase 2 clinical trial, which I'm happy to say is progressing well. We are beyond the 50% point in terms of enrollment. We're studying adult patients with complicated Staph aureus bacteremia and infective endocarditis. Patients are treated with conventional antibiotics per protocol, but essentially consistent with guidelines. So no surprises in terms of background antibiotics. And these patients have to have known or suspected Staph aureus, MRSA or methicillin-sensitive staph. One of the reasons I would say we chose to go in this direction is that there is an unmet medical need here. If you look at the last phase 3 study done for the last drug approved for this indication in the US, that was daptomycin. That was over 10 years ago. Vance, I don't know if Vance is still here. Vance was the lead author on that paper. And if you look at the clinical cure rates at test of cure, you're looking at about a 48%, depending on which numbers you look at, clinical cure rate with daptomycin and the comparator arm in the non-inferiority trial. So we felt that the features of the lysine, rapid ability to drop bacterial counts on its own, synergy with conventional antibiotics, and the antibiofilm activity might make it a useful partner to improve outcomes in this disease where there is a need to improve outcomes. So, so that's really the premise. Where our design, as you might infer from what I said, compares in a randomized, double-blinded, placebo-controlled manner 301 in addition to standard of care antibiotics compared to 301, compared to conventional antibiotics alone. And we're looking to randomize about 115 patients. We sized the study to be doable from a practical and financial perspective, but still to be able to detect what we would consider to be a substantial benefit. So somewheres in the 20%, maybe even a little less, 17% improvement in the treatment arm, in the active treatment arm should result in a statistically significant difference. And I have the rest of the information just high level, where our primary input is an exploratory early clinical response rate at day 14. That's based on what I showed you about the rapidity of the action of the lysine, but we are going to look at and collect and report the data at end of conventional treatment and test of cure, the traditional regulatory endpoint for this indication. And we are, I'm happy to say, pleased with our progress, and we are on track for top-line data by the end of the year. So we're moving forward. So in terms of challenges, that's not to say that any of this was easy. I could echo all of the comments made earlier about funding and resources for a small company. But I would also say, just maybe leveraging some of what John Rex was talking about earlier, in addition to trials, in general, are not easy to do, and companies stay away from them because it's difficult. You have a standard of care, and we're looking to show an improvement above and beyond what the standard of care is. As I said, the documented response rates, particularly in infectious endocarditis, are low enough to allow room to show a difference in this indication. But I think the trial design, the execution of the trial have challenges, and I think it'll be interesting to talk about, at the end of the day, what constitutes a clinically meaningful improvement above standard of care, because that's not something that's been worked out, discussed, agreed, but we're happy to be on the leading edge of the discussion with this trial, so thank you.
Thank you, Kara. Our next presenter is Wayne Denkner, Chief Medical Officer at Atox Bio.
Good afternoon, everybody. So as the old statement for people who know prior television shows, now for something completely different. So we're actually a late-stage company, and we'll talk about a little bit about that. And we don't directly attack the bacteria. We're actually looking to modulate or improve the dysfunctional host immune response so that the immune response will be more appropriate for the infectious product. So we're basically putting our drug on top of standard of care, which includes antibiotic therapy. So a little bit about Atox Bio. It's a late-stage company, as I mentioned. Our drug is called Rotessimod. We've moved from a number to an actual INN-approved generic name, so you can tell we've moved along pretty well. It's an immunomodulatory drug, and we're targeting critically ill patients. So these are going to be patients who have very serious infections, significant unmet medical need, who, despite other standard of care, including antibiotics, still have less than optimal outcomes. We actually, as I mentioned, we're late stage. We're already into an ongoing pivotal trial, a phase 3 study in patients with necrotizing soft tissue infections, what people seem to know as the flesh-eating bacteria. There hasn't been an improvement in care in a long time for this disease. And then recently, we've just started a trial in patients with acute kidney injury and those who have abdominal infection, sepsis. We're recognizing that acute kidney injury is becoming a fast known cause of significant morbidity and mortality in critical care patients. We're the first product to ever be developed specifically for NSTI. We completed a phase 2 trial, which I'll show a little bit about, where we actually were able to demonstrate no serious adverse events. So we have a very clean safety profile. We have a DSMB for the ongoing phase 3, and for the phase 2 we started. And I think importantly, as sort of mentioned throughout this conference, we've had the support of both the FDA and the EMA in terms of scientific advice. We have fast track status. We've worked with Ed Cox's group, and we've gotten a lot of important advice how to move the development of this product along. There is no guidance document for this disease. We'll talk a bit about the endpoint and the fact that it's quite unique and had to be developed specifically for this indication. And similarly for acute kidney injury, the endpoints are sort of just being developed. In terms of funding, I think the product was originally developed more toward biothreat. When it was first being developed, it was targeted for superantigen. I'll show you what the structure looks like. So there was already some connection to biothreat concept. We've been very fortunate to have the support of BARDA. They've been able to provide us funding for the phase 3 trial. I think without the BARDA funding, I'm not sure whether we would be able to progress as far as we have. And additionally, we have an excellent group of investors, so we're privately owned. We don't have the forward-looking statement at this point because we're not publicly owned. But our investor group and board of directors has been an excellent group of individuals. You can see some of the names of the companies there, which are quite active in the area of anti-infectives and others. The other thing that's important for a small biotech is to have the money to basically finish the trial. So we're well funded all the way through readout for both our studies that we have in development. So a little bit about the drug. So as I mentioned, we're somewhat novel and sort of in the sphere of this particular conference that we're basically trying to affect the immune system so that it takes the dysfunctional immune system and makes it more functional so that the patients can better fight their infection, and also to reduce the effect upon the infection on organ dysfunction. So our drug is a short peptide that basically binds to the CD28 homodimer region and then attenuates the formation of the immunologic synapse with B7-2, which actually other people call CD86, which is a receptor on the antigen-presenting cells. So there's an interaction between the antigen-presenting cells and the T cells, and it's the T cells that basically start to cause this interaction for cytokine production that can become out of control. So you see a little bit about the way the drug was developed. And as I mentioned, the drug was originally developed to counter superantigen, which was being looked at as a potential biothreat. We'll show you some of the early work that was done to demonstrate its activity against staph enterotoxin B. So this was some of the early non-clinical work that was performed to show a little bit of proof of concept in both in vitro and in vivo models. So in the in vitro model, PBMCs were stimulated with SEB, which is staph enterotoxin B, which there was concern about the ability to take that particular enterotoxin and weaponize it. And then if you're not familiar with superantigens, toxic shock syndrome is mediated by superantigens, and if you weaponize something and throw everybody into shock, you can pretty much have a significant impact upon the general population with mortality rates that probably approach 50% or more. So in this particular experiment, the PBMCs were stimulated with staph enterotoxin B and then either treated with a saline or a placebo, and then also treated with 3 different doses of reltestimod. And you can see that all 3 doses attenuated the IL-6 response significantly in those cells that were treated with the drug compared to the placebo. The drug was then taken and put into an in vivo model that used a common approach to sort of an abdominal or sepsis approach called the CLP model, which is used to basically just take the animal's colon, puncture it, and then cause them to have a peritonitis that then causes a generalized sepsis. You can see, again, the concept here is that we do not want to turn off the immune response. We actually just want to modulate it, again, allowing the immune response to behave properly so that the patient can overcome the infection along with their supportive care. So you can see in this in vivo model, not only did we see a significant improvement in mortality in the animals, but we saw this attenuation of, again, the IL-6 response, which was quite significant. So why are we interested sort of in this approach? Well, if you look at a lot of infections, antibiotics can go and do just so much. You still have the problem that patients develop acute inflammation that then can lead to organ dysfunction or organ failure. And the organ function or organ failure or organ dysfunction can account to as much as 50% of ICU deaths. So despite even infections that have treatable antibiotic profiles, you're still seeing patients dying from their overwhelming infections and just problems with their organ function that still needs supportive care. So you can see the variety of different organ dysfunctions that you see. Those who are familiar with trials that have been done in sepsis and so on see all these complications that arise despite the best antibiotics that are out there to basically overcome the ongoing infection itself. So our approach was basically, can we address this inflammatory response and improve the outcomes in these patients and reduce organ dysfunction where essentially the drug would have its best target. So the next question was, do we want to be like everybody else, or do we want to really focus more on something where we could look at a disease that had a significant unmet medical need, which necrotizing soft tissue infections have. For those who have been in antibiotic development, these infections are always excluded from just about every other antibiotic trial that is done because of outcomes associated with it. And these patients have very significant outcomes that can be very problematic for them. One is the morbidity because of the organ dysfunction that these patients suffer. They have poor quality of life due to loss of significant amount of tissue, risk for amputation, and mortality rates are still in the 10 to 20% range despite the best interventions. A lot of these patients wind up with becoming relatively non-functional even after they've gotten out of the intensive care unit into life. And the average age, unlike in other critical care illnesses, is in the 50s. So these people are still quite active in terms of family life, work, raising families, interaction in the community, and so on. And we've met with a lot of the survivors, and some of them just never get back into the workforce again. The other reason for choosing this disease is because source control is key to any intervention when you're looking at, again, antibiotic therapy. Also, when you're looking at an immunomodulatory drug, you want to establish source control. The advantage of NSCI is that these infections are recognized relatively quickly in an emergency room setting, and these patients are going to the operating room with probably an hour or two at the most of being seen in the emergency room. In trauma centers throughout the U.S., now acute care surgery centers, these patients sit right behind the trauma patients in terms of their access to the OR, knowing how quickly these infections can spread and how without intervention, without classic surgical intervention, it requires rapid debridement, the first debridement being pretty much the critical debridement for these patients. These patients would otherwise succumb to their infection. And then the vast majority of them wind up in ICU settings post-surgery. Our experience is they tend to get sicker before they get better after the initial debridement, taking away a lot of tissue and sort of stirring up again the immune response or hyperimmune response in these patients. So we embarked upon what really was the first well-controlled, randomized, placebo-controlled trial in necrotizing soft tissue infection in this patient population. We essentially did this at about 7 centers in the US. They were all major trauma centers, again, because they have experience in treating this infection. Interestingly, in talking to the surgeons, no one else wants to manage these patients. So the trauma group are pretty much experienced in seeing these patients, and because they can get these patients to the operating room very quickly. Additionally, the trauma centers have 24/7 coverage for all different surgical specialties. We looked at 2 different doses that was based upon allometric calculations from the animal studies, and we then had a true placebo group in the population, and this was layered upon standard of care therapy, which included surgical intervention, initial debridement, follow-up debridements, antibiotic therapy appropriate for the pathogens that they might be seeing, and then additionally for follow-up with that, we required only a single dose of the drug based upon our animal experience. What I failed to mention before about the drug is that because it's working at the host immune response, it essentially is pathogen agnostic. So there really is, we don't have to worry about the pathogen from that perspective. We look at the sites providing the appropriate standard of care in terms of antibiotic therapy, recognizing what's in the community, where the infection is occurring. And so we have less issues with the pathogen, and we don't have resistance developing, at least in terms of how our drug interacts with the host response. So we gave a single dose of the drug. It had to be delivered within 6 hours of the decision to go to surgery. Again, the surgical intervention sort of being a very finite point of time to basically base therapy on. We looked at organ function through a tool called SOFA at baseline, and then followed that throughout the patient's hospital course. And you can see in the 3 different dosing arms that actually the patients in the 0.5 milligram per kilogram dosing arm were a bit sicker than the patients in the other 2 arms. A higher percentage of them actually had evidence of shock vasopressor support at baseline. A number of these patients wound up developing shock after the surgery. We also then looked at other systemic and local manifestations, looked at the number of debridements these patients required. Our drug definitely has an impact upon cytokines in the human population. I'll show you a little bit of that data. And not only does it have an effect upon the systemic levels, but also at the local tissue level, which may be mediating some of the necrosis that's seen at the local level. We looked at how that impacts ICU days, days on the ventilator, so on. So this is just a brief look at how we impacted organ dysfunction in this population. So we, for those who may not be familiar with SOFA, SOFA is a sequential organ failure assessment tool. It looks at 6 different major sort of organ systems that have the risk of going into dysfunction that can impact patient lives at that point. So you have a respiratory component, a cardiovascular, renal, coagulation, CNS, liver, and these are based upon an integer system going from 0 to 4. 0 being no organ dysfunction whatsoever, 1, some physiologic perturbation of the organ, 2, 3, and 4 going from organ dysfunction to organ failure. So obviously the worst score you could have in this case would be 24, the best score would be 0. We decided to look at organ function at day 14 because the issue was that if you have a score of 0 or 1 at day 14, you pretty much don't have any organ dysfunction. You should be able to be moved out of the ICU. You may be able to be discharged to a rehab center depending upon how much intervention was required in terms of the debridement, possible amputation, and so on. But our goal was to basically look at how well these patients recovered from their initial insult and how much the drug may have impacted that. So when you look at, and then additionally, we also wanted to get past the point where the patients were having these multiple and significant debridements that might carry them out to as far as day 10 to day 14 in some cases. So when you look at the data, you can see that by looking at the 0.5 milligram per kilogram dosing group, we basically were able to demonstrate a significant improvement in terms of organ dysfunction or organ failure resolution. So we would consider these patients to be relatively well from an interventional perspective, from being no longer requiring medical intervention that would require intensive care support. And we'll show you how that translated into intensive care days. And you can see that basically then translated into a median day 14 SOFA score of less than 1 in the 0.5 group compared to the placebo group where far less patients had organ failure resolution, much more patients still having organ dysfunction even out as day 14. And this now also can be correlated with what's being seen in the ICU centers where you have this what they call critical care illness syndrome that's developing in some of these patients where they just don't get better and they linger in the ICU for weeks on end. Just to show you, again, previously I demonstrated to you the data on the cytokine production in both the in vitro and in the animals. This is the data actually taken from the clinical trial looking at how the cytokine measurements correlated with the SOFA score in this patient population. So those patients who had SOFA scores that were improved at 0 to 24 hours versus those at 0 to 48 hours. And you can see that clearly there is a good correlation between the levels of cytokine and the improvement in the cytokine and organ dysfunction as cytokines decline. Again, sort of providing sort of this concept of proofs of concept by reducing the cytokine levels, but not abrogating the cytokine response to allow the immune system to function more properly. You can see that patients would have improvements in organ function, and that then would lead to the patients should be having better recoveries. So this is just some more of the data just to show that we are able to demonstrate improvements across the board in ICU days, days on the ventilator, number of patients who only required 1 debridement, which is, again, interesting. The less debridements that you can perform in the patients, the better. Number of patients who had more than 3 debridements. The average number of debridements from our own review of the literature and doing a retrospective survey is about 3. So if you can keep patients to less than 3 debridements, that's ideal. And then mortality, again, smaller study, but we were seeing, early trends in improvement in mortality. So we took this data and, then decided that it was time to move into a phase 3 trial. Where a small company trying to do a large phase 2B trial would have, been difficult. And one of the challenges that we'll talk about is enrolling, into an orphan population such as this particular disease. There's probably about 25,000 cases per year of necrotizing soft tissue infection, includes Fournier's and so on, in the United States alone. And as pointed out earlier, you think that they might be easy to enroll, but they're distributed across multiple centers, and you got to find the patients where they are, and then be able to capture them, and at the same time enriching the population, as we've done in the phase 3 trial. So this required a significant amount of interaction, both with the FDA and the EMA, to kind of come to agreement about the endpoint. As I mentioned before, there's no guidance document for this. Essentially, we had to develop the endpoint based upon the data we collected from the phase 2 trial, from what we understood of NSTI, and looking at what the mechanism of action of the drug was, where it could have its greatest impact. So we developed a composite endpoint that looked at mortality, debridements, and essentially organ dysfunction. And any one of those fails, you basically fail the composite. As long as you're positive on all of them, you would have a success. Additionally, because we were dealing with an orphan disease, trying to do 2 well-controlled trials, as pointed out before, would be along a geological timescale. So we worked with actually both groups and the FDA to come to an agreement about the size for a single pivotal trial, recognizing that we had to show somewhat lower p-value to basically demonstrate efficacy of the drug. You can see that, and then, as I mentioned, we also then targeted a population of patients who already had organ dysfunction prior to the initial surgery, so we could demonstrate, clear-cut benefit in that population. We have probably engaged about 80 centers in the United States. We have active now about 65, and we're recently going to be engaging sites in France to help complete the enrollment of the trial. The target population is 290 patients. We're hoping to have results out in some part of 2019 to be able to read out the results of the study. And then just a brief introduction to the phase 2 trial that we're doing. This one, classic phase 2, we're looking here at 2 different doses. We're using the same dose that we are using in the phase 3 trial. We also have data, showing that we have activity against acute kidney injury, and we could also use data from the phase 3 trial to demonstrate the activity in those patients who come in with sepsis, with necrotizing soft tissue infection, who have acute kidney injury, and be able to merge that data later on for discussion with the agency around what a phase 3 trial or other approach would be to get approval in this indication. Again, only a single dose and targeting centers in the U.S. Again, these are surgical centers because we already have activity going on with the NSTI, and it's the same surgeons who manage the acute intra-abdominal infections are also the same surgeons who manage the NSTI infections. Yep. So lastly, I just want to point out some of the drug development challenges. So we're a small biotech. We talked about that, but being a small foreign biotech has some real impact. We had to transition from what was original biothreat development plan to then moving into the clinic. And then, as I pointed out, this, identifying a unique patient population is quite significant, and we want to avoid all the pitfalls associated with development programs in that particular area before, and dealing with heterogeneous patient populations in the sepsis area. We talked about the fact that we had to develop this novel clinical endpoint, and probably in the panel discussion, we'll talk a little bit more about the patient enrollment. Very challenging to go into an orphan disease that's sporadically occurring. We don't have the advantage and an often indication of finding, centers of excellence that see these patients in a registry, and finding investigative sites that can actually enroll patients and provide the clinical research support when patients are basically coming in about 2 o'clock in the morning is also limited the number of centers that we can deal with. And lastly, the whole regulatory approach to a single pivotal trial. Thank you.
Okay, thank you, Wayne. We'll go on to our next presenter, who's Vu Truong, CEO at Aridis. Okay, yeah.
Okay, so how long do I have? Well, 10 minutes. Okay, the last two speakers, 20 minutes. I'll try to do 10. Okay, thank you. Aridis is a privately held company founded by a bunch of futurists. We envision that there's going to be two paradigm shifts in infectious diseases. And we're going to discuss that today. One is that we predict that there's going to be a shift from empirical broad spectrum to more of these evidence-based, diagnostics-driven anti-infectives. And there are a number of reasons why we feel this way. I think one of them is that the proliferation of rapid diagnostic tests has been going on, and so far over half of the hospital centers in the U.S. and Europe are using these now. And when the saturation is complete, the physician is going to have the patient profile available to them within the hour of a patient visit. So when that happens, we believe that it just makes more sense to be using narrow spectrum and targeted therapy in terms of the problem of drug resistance and the problem of microbiome perturbation. I think the second paradigm shift that we think that's going to happen in this area is also the transition from non-inferiority trial design to superiority trial design. And this is going to have to be necessary in order for us to develop drugs that are well differentiated from what the available standard of care is. And so that is what we're working towards as a company. Now, specifically, what I'm talking about today will be a shift toward targeted therapy using monoclonal antibodies. There is actually a lot to like here when one look at as to why antibody of all the non-traditional anti-infectives. We know that in virtually every infectious diseases, you always find a subset of patient population that manages the infection better than the average population. And that's largely because we are generating random immune responses, every one of us, and this is how affinity maturation works. So you see these rare patient populations that seems to be highly protective. And so we have a way to screen the B-cell repertoire of these patients, and within a couple of weeks, we can identify the highest neutralizing antibody from that patient. And we put it back to the animal and ask the question, does that antibody directed against that specific target, does that lead to complete protection against morbidity and mortality in that animal? Then we could place a higher bet that it's likely that there is a high correlation in terms of protection. So our antibodies are fully human, essentially optimized by the human immune repertoire, and they are expected to have a very attractive safety profile. Very long durability action compared to traditional anti-infective, of course, 3 to 4 weeks half-life for an IgG1. Obviously, this approach also avoids the negative perturbations or impact on the microbiome. And so because of the safety profile and the durability of action of this class of anti-infectives, we have the flexibility of using it in a prophylactic mode as well as therapeutic mode. And some of us who are developing monoclonals in this space are adopting either or both approaches. Initially, we are focusing on therapeutic. I mean, there are advantages and disadvantages here in either approach. We're focusing on adjunctive therapeutic. This, we think, will provide for a clear demonstration of clinical benefits. It is more persuasive in terms of reimbursement rationale in a very difficult to reimburse therapeutic area. These tend to also be smaller clinical trial design and thereby costing less to do and quicker for readout. Now, there are a lot of risks here, of course. not only smaller addressable patient population. We'll focus on pneumonia now. This is actually close to the orphan type of indication in terms of patient population. But the bigger risk is, of course, the adjunctive mortality means that there is a much higher risk placed on how much of the incremental difference compared to standard of care can you demonstrate. And so it is more of a technical risk associated with the approach. Now, in terms of the benefits of a prophylactic mode of use, larger addressable patient population, obviously we're looking for at-risk patients that don't have disease. It avoids if it works, it avoids the expenditure of the entire disease episode, potentially also antibiotic sparing. But the disadvantage is, of course, that in the midst of, again, a very difficult to reimburse therapeutic area, you're going to have to demonstrate that the prevalence and the benefit of prevention is attractive enough that the payer is going to want to pay for this. And also, obviously, these will involve larger sample size. But either approach, these are target therapies. So both are going to require the companion diagnostic in order to demonstrate the potential benefit. We'll focus on infection in the ICU. Respiratory infections is what we're focusing on. To the pie chart here shows approximately the bacterial agent that are causing infection in this space. And we are now developing monoclonal antibodies that trying to address essentially most of the major pathogen. Pseudomonas, Staph aureus, A. baumannii, and we're currently working on Klebsiella pneumonia. A couple of our projects are now proceeding through the pivotal trial stage. In terms of the critical care medicine that we're focusing on, the right is, of course, therapeutic treatment. We're interested in pneumonia. Our next target indication for us is bacteremia. Compared to an ICU patient that, of course, don't have pneumonia symptoms, as you can see, the increase in the patient burden and the burden to the hospitalization cost is substantial when one develops the symptomology. The current standard of care is shown on the bottom here. It's either mono or combination therapy. This is an area where we're still seeing upwards of 50% mortality. So there's a need for safer, prolonged therapy, particularly ones that could effectively address resistance. Several of us are in clinical studies looking at prevention. So here in pneumonia, we'd be looking for at-risk bacterial colonized ICU patients that don't have symptomology. And here we're trying to prevent progression into pneumonia. And I think later on, Dave Mantis will give you an introduction to their exciting program looking at prevention in pneumonia of a monoclonal antibody therapy approach. Of course, currently, in terms of antibiotic, it's just not practical to be using this in a prophylactic mode. A clinical strategy for us is, again, added on top of standard of care. Any antibiotic, we don't have a preference. We actually allow the physician to choose that. The reason why we do this is that in the animal model, it tells us that typically when we use both of these molecules in combination, we actually get a synergistic effect, not just an additive effect, but synergistic effect. So this makes sense. Also, the adjunctive modality allows us to cover against polymicrobial infection condition where about 30 to 40% of the HAP-and-VAP patient will be more polymicrobial in terms of what's infected them. It allows us, of course, to use superiority trial design to show a clear benefit, and also, of course, to provide the opportunity to demonstrate outcomes-based and value-based pricing scheme. So the lead project for the company is an antibody study is neutralizing one of the toxins that Staph aureus makes, and that's called alpha toxin. This is the most well studied of all the toxins. Very stable sequence. Over about 30 years of sequence information, there's actually not a reported case of a mutation on this toxin. What a toxin does is it pokes holes and destroys host cells local to the infection site, but also to the cells that are coming in, the immune cells that are coming in to try to deal with infection. So what it does is, of course, neutralize the toxic effect of the toxin. We took the project through a randomized double-blind placebo-controlled study in HAP and VAP patients that are specifically caused by Staph aureus, and the inclusion is using a rapid diagnostic test. Not only do they have to have symptomology, but also be infected with Staph aureus. So the trial design is the standard of care is considered a placebo group, and we just add the antibodies on top. It's a single ascending dose. So again, a single shot of the antibody is all you need to cover that patient throughout the entire ICU stay. That's one of the benefits of monoclonal. Safety and PK were the primary endpoint. The drug was deemed to be well tolerated, no SAE all the way to 20 mg per kg. Some of the secondary outcome measures that we collected are shown at the bottom. Time to removal ventilation. Essentially, all of these patients are mechanically ventilated. Microbiological cure, time to cure, days in ICU and days in hospital. This is a small study. It's only 48 patients, but we're starting to see a very nice trend to a number of these outcome measures. We're going to need to do a larger sample size to capture outcome measures such as all-cause mortality or clinical cure rate. Of course, because clinical cure also captures mortality in that outcome measure. Where we are proceeding with this project is we're in the discussion with EMA and FDA on the study design and the primary endpoint. What we are proposing clinical cure of pneumonia as the primary endpoint, and how do we define cure is a subject of discussion. The other project, let me show you just quickly one of the data. And by the way, the clinical data is in press. It's going to be published in the Intensive Care Medicine Journal. So in a couple of weeks, it should be out. But here is one of the outcome measure, ventilation days. The treated group is 1, 3, 20, 10, and 20 mg per kg. Sample size, again, is small, but as you can see, there is a nice trend toward reduction in ventilation time in these patients. If you look at the Kaplan-Meier curve on the left side, there seems to be an effect here. And so what we, of course, aim to do is to increase the sample size significantly so that we could confirm this preliminary result. The other major project for the company is an antibody against Pseudomonas aeruginosa. Again, this is the most prevalent gram-negative in HAP and VAP. Staph aureus is the most prevalent in HAP and VAP. How the antibody works is it binds to a polysaccharide on the capsule of the bacteria called alginate. This is decorated on essentially all Pseudomonas. And when it does that, it stimulates complement deposition. And with the antibody and complement complex, this is what the circulating immune cells need to properly recognize the pathogen, leading to engulfment and clearance of the bacteria. Currently, the study is in a global phase 2 study. Right now, we power it to an 80% power level. We plan to expand to a 90% power study following discussion with the regulators. And when that happens, it's going to involve up to 240 patients. Placebo, which is the antibiotics alone, versus the antibiotic and antibody at 20 mg per kg. Clinical cure rate right now is the primary endpoint. We have so far activated about 9 clinical sites. We're going to expand that to about 130 clinical sites across about 22 or 23 countries. This is expected to have preliminary interim data readout sometime in the first half of next year. Both of these monoclonal antibody projects are as far as we can tell, the most advanced programs in monoclonal in pneumonia under development today. So that's the introduction of what we do.
Great, thank you. Okay, so now I'm going to ask the presenters to come back up here on stage, and also join us will be, let me see what my notes are, Sumanthi Nambiar, Director of Division of Anti-Infective Products in the Office of Antimicrobial Products at CDER, FDA, and Dennis Dixon, Chief of the Bacteriology and Mycology Branch at the National Institute of Allergy and Infectious Disease at NIH. So why don't we kick off
Sumanthi, you've had a chance to view these presentations, and you might have some thoughts or reaction to the range of technologies and the potential challenges that the presenters already highlighted.
Is this on? Okay.
Yeah.
Thanks, Gregory, and thank you for the opportunity to be part of this panel. Very interesting presentations this afternoon. And I think a couple of points that I would like to make is, one is regarding the trial design and the magnitude of the treatment effect. Now, some of these products and products like this, you know, we have interacted with companies as these products are being developed, and some products that have used this kind of an approach have already been approved. And one important criterion really is, what does this product offer over and above what the standard of care offers, and what is the magnitude of the treatment effect these products can bring? Because that really dictates what the size of the study would be. So that, I think, is a very critical element. And, you know, as we've heard during discussions this morning, most antibiotics that we currently have do a reasonably good job. So to be able to demonstrate a benefit over and above what the standard of care offers, I think your product really has to have some kind of tangible benefit. The second point that I wanted to raise is, and it did come across in the presentations we heard this afternoon, is the choice of endpoints. You know, we're certainly flexible and willing to consider novel endpoints. You heard discussions around endpoints that people are trying in these studies, which really haven't been evaluated before. But when one would consider using such endpoints, I think it's very important to remember that the endpoint we pick is clinically meaningful. It should translate into some benefit for patients. So that's very important. We wouldn't like an endpoint that just is a change on a lab parameter. The change on the lab parameter really has to translate into a clinical benefit. So I think that's very important. And then also, you know, what you've seen in your earlier studies, I think the example that Kara provided, where their hope is that the benefit that they saw in vitro would in fact translate into clinical benefit. I think that would be good if it does translate into clinical benefit, but trying to evaluate that in clinical trials is really based on some evidence that you got from your prior study. So I think that's sort of a logical way to do things. So I'd stop there and...
Okay, great. Dennis, any reactions from you?
You have the light. Oh, it is working. It's like,
Yeah,
guess whether it's on or not.
Don't touch.
So do you want to allow response time for somebody's questions before you move into my?
I'm sorry, what was that?
Do you want to allow time for discussion to somebody's questions before we launch into a different?
Yeah, we can if they're immediate, or we can sort of do it as a group. Why don't you go ahead, Dennis, with your thoughts, and then we'll
Okay.
do it as a group.
So thank you for the opportunity to participate. And we at the NIAID, National Institute of Allergy and Infectious Diseases, are champions of these approaches. We've listed alternatives to antibiotics in our strategic plans and our strategies that we posted online. And you exemplify some of the very things we hoped would come forward. We worked with some of you on these. So what I'm going to do is ask questions that are, and for the benefit of people who may not know, we provide a series of services to decrease the risk of companies moving to this space. And these are often done under contract for screening through MIC, and you're not going to be able to use those if you're developing an immunomodulator. So there's one question right there. What are we missing for you to be able to screen your stuff, what it's supposed to do? We basically provide the services for every step along the drug development pathway that are usually housed in big companies, broken up into individual service contracts. And that is aimed at helping you get past the biggest gap that you experience. So we're going to be asking questions, what are we missing in those suite of services that would have helped you as you were moving forward? And what are some of the experiences you learned with your product development that, well, wow, this was really different than the traditional pathway? What did I use as a surrogate for an MIC? What do I use as a surrogate for the traditional animal model moving forward? So if you can be thinking of something that comes to mind that you got around somehow, and how you did that, and what might be transferable to other products that follow you, or to ones that you're still stuck with. I don't know if any of you still have pockets in your IND submission or your NDA submission package from the preclinical side that you still need to fill or not. So a lot of questions there, but What sorts of services do you wish you had that you didn't have when you were moving into this area?
Great, thanks for that, Dennis. So let's go back to some of the questions that Sumanthi brought up, and then we'll take those. So I guess a suite of questions around clinical benefit and measuring that, particularly in the context of these more non-traditional products that are used in combination. And some of the presentations did sort of address this, but additional thoughts with respect to Sumanthi's specific question around demonstrating benefit over and above the standard of care, and challenges with that, and what you all have sort of tried to address.
Right now, one of the issue for us is how to define clinical cure of pneumonia. Clearly, the last guidance published in 2014 gave an idea of what the symptomology that would be defined as having a patient having pneumonia, but it did not cover what would constitute cure. It's clearly not at the inverse, right? And so one of the issue for us is how do you define cure of pneumonia? We've corralled about 7 KOLs in this space, and we got about 10 different answers in terms of how they would define cure of pneumonia. So that's one of the things that could help a lot of us who are focusing on treatment.
So as I mentioned, we had a number of discussions both with the FDA and the EMA around, how do you define an endpoint for immunomodulatory drug? I mean, these have always been kind of difficult areas to address. You're not just going to, just looking at a cytokine level is just at best a surrogate and a less than optimal surrogate. They help describe mechanism of action. But from our perspective, and doing a straight mortality study would have been a large number of patients, and the difficulty in enrolling patients in an orphan disease is difficult enough rather than trying to do a several hundred patient study to begin with. So the organ dysfunction, I think we would posit that it actually does translate into patient outcomes. I mean, it's less ICU days, which is important for patient populations. We can talk about the economics of that, which are critically important also. Patients spend less time in the ICU. They have less risk of then getting iatrogenic infections that can then lead to poor outcomes, the less time on the ventilator. And we're in the process now of looking at longer-term outcomes also through a large retrospective look to see how SOFA scores that late in the course correlate with outcomes such as not only mortality but rehospitalization rates and so on. So our feeling is, if your organ function is normal, you should do better than the patients who have continued organ dysfunction and linger in the ICUs for long periods of time.
I just make 2 comments. One around treatment benefit. We did what most people would do, is talk to treaters and experts, and kind of got to what we thought might be a reasonable number. On understanding we're in phase 2, we needed to keep it to a reasonable sized trial. So we did the best we could. What I'm pleased to say is that we have the study open in 14 countries now, finally. And our investigators globally, we didn't really make a global outreach, but I will say globally, we have a lot of enthusiasm for this idea. And it tells me that must be an unmet need that these people are seeing, because they're very enthusiastic about participating in the trial, enrolling patients. It's been a big effort, particularly getting through some of the hurdles outside of the U.S., actually. U.S. has been open since May. We just started opening up in Europe recently. So that's one thing. For our phase 2, we also chose what we consider, and we talked to Sumati about it, an exploratory endpoint, 14-day early response rate for endocarditis. But we thought that might be appropriate for the license because of the rapidity of action, and it might, if you think of learn and confirm as a drug development model, we're in the learn stage. So we thought that might teach us some things, but we kept to the traditional path of also measuring response at test of cure.
Sumanthe, any further
Yeah.
clarification or questions?
So, I mean, I don't want to go into discussion about a clinical cure endpoint. I mean, that's a very specific topic, but I think that's a particularly challenging area. Many people have tried to define what's clinical cure, and it's very difficult. And so we'll talk about that, maybe offline. That's a whole different discussion. But in terms of the endpoint, I think, especially when one is considering novel endpoint or a totally new indication, the example that Wayne gave, there is no guidance. We haven't had any product specifically developed for necrotizing soft tissue infection. I think there's a lot of merit to having discussions and coming to agreement on what could be a clinically meaningful endpoint. And I think Wayne would agree that we've had many discussions before we could arrive at an endpoint that we were all comfortable with, which we thought would be clinically meaningful. And I think it's also important to emphasize that there is a role to be played by with phase 2 trials. People try to expedite and rush through programs and don't necessarily spend the time to evaluate some of these newer endpoints in earlier phase trials, because that will then really help you design your phase 3 trials appropriately.
So Dennis, you brought up you mentioned the suite of services offered by NIAID and requested from the panelists your thoughts on, as you're going through these development programs, considering these more unique non-traditional antibiotics. What comes up that they may not be helping yet? Where can they help more? Things like that.
So for us, probably the biggest challenge was not understanding epidemiology of the necrotizing soft tissue infection. No one's really looked at it as, single center series and so on. The CDC reports, 1,500 cases a year. It's all based off of passive reporting on group A strep, which is, can be one of the more severe forms of the disease. So we basically had to develop all that on our own. And you had to have that data to be able to understand what the outcomes would be and be able to generate a sample size that would be, workable. And I would think, having more understanding of epidemiology of some of these orphan indications would really be helpful to know how you can then design your trial and design endpoints that would, again, as pointed out, that have some clinical meaningfulness. Not only to the regulatory agents, but more importantly, I think in the long run, to the patients and to the physicians and medical staff that are caring for those patients.
Well, that's more of a knowledge gap than it is a service.
Right.
Were there problems adapting animal models to assess the activity of your compound? And the models that would be directly applicable, transferable to the same mechanisms in humans?
There's always that concern about how do you take an animal model and, basically adapt it or correlate it to the human outcome, because you want to see, obviously, some effect in the in vivo. Clearly we can show in PBMCs that we can, suppress or attenuate the response, but we definitely had to. So finding animal models, I think, was, going down the traditional route of looking at CLP and so on, has its advantages and has its disadvantages in terms of looking at that. And cytokine, again, trying to correlate cytokine production to outcomes was critical. I think so having access to, good animal model services, I think would be important, so people don't have to go hunting around for standardization, because standardization really comes key in some of these animal models.
And for your example, which model worked best?
We've seen the CLP model work pretty well. We had a model for actually sort of a necrotizing infection of the pore of animals and saw some interesting results with that. So I think, I'm not sure if there would be another model that we would want to look at, because again, you can learn just so much from the animals, and we saw the improvement in mortality and changes in cytokine production that we could measure. I think for acute kidney injury, there's also sort of a dearth of animal models that really can identify how well you're improving kidney function, and that would, without straight going into a human population.
To prompt questions, one example might be from Kara. And did you run into any issues as you were looking at MIC impact could you just take a standard MIC assay and apply your product to that, or did you have to?
That's a good question. We did considerable work on the MIC methodology. We are fortunate because my head of research followed this compound from Rockefeller University, where he identified it, into Contrafect, where he's been for more years than I have for the past 6 years. He spent a good year and a half working out an MIC methodology that is now, happy to say, endorsed by CLSI and is standardizable for use in a clinical setting.
And works for FDA?
What's that?
And is recognizable and
Well, I think
comparable to what?
It's pretty rigorous review at CLSI. So, what I would say though is, for a small company, we're lucky that we had some microbiologist leader who could take that on. I think it could be challenging for companies to do so. I think that's interesting thing. We had a particularly unique set of challenges with our MIC methodology relative to serum and MHB effects and a whole kind of bunch of technical issues that are not worth getting into now. But yeah.
Yeah, I think for biologics, the two areas of great need in terms of services, I think, would be clinical manufacturing and a phase one unit that the company could access to. Typically, clinical manufacturing costs several times more than the phase one costs, and so that's usually is biggie.
I think we've been more
antibodies, for example, it's $3 to $5 million just for the phase one clinical supply.
And I think we've been more successful in helping you with the manufacturing than with the clinical trial, but I think we're ready to run back at that a little bit swifter now.
Great.
And I'd like to point out, that was one of the things that really was important to us in terms of the BARDA funding, because it allowed us to take clinical trial supply and develop a scale-up, because at the end of the day, you can have a great phase 3 trial, but if you're not ready for the clinic, the drug is not going to get out there for the patient population. So the BARDA funding was really important from that perspective, and then getting guidance from BARDA on an ongoing basis as to how the scale-up was going, and ensuring that we were going to meet all the FDA standards that would be required also. So I think having that, and we basically are traditional peptide, so the advantage is that at least there's companies out there that have that experience sort of developing peptides.
And if I could ask Vu, what rapid diagnostic did you use for the staphylococcal screening?
We use a Cepheid Gene Expert system, but we're going to expand it into several others. For example, the bioMérieux FilmArray.
Did you explore any partnerships with diagnostics companies for merging your efforts for mutual gain?
Yes, yeah, we're in discussion with several.
Don't ask you to call any names, but how has that gone in general in trying to
use
that partnership?
Yeah, that too has been challenging, because I think these rapid diagnostic companies recognize that there is actually more profit to be gained developing tests for oncology and other therapeutic areas. And so infectious disease, that rapid diagnostic has not gotten the attention that it really deserves.
Okay, that's helpful.
So quick time check here. We're exactly out of time for this particular session, but I'm going to take the liberty to extend it by virtue of my powers as the moderator to extend at least another 10 minutes or so to accommodate any questions from the audience or folks online. Go ahead, and as a reminder, there are microphones about midway back. Yes, go ahead.
My question is for Kara. I think that was a very interesting approach, and if you're successful, I think you can replicate it in for many other pathogens, because there's no shortage of bacterial lysins for different pathogens now. Whether they're going to be druggable or not, I don't know. But one potential challenge I was thinking, because we were working with some C. diff lysins, it was the broad seroprevalence in the population. So we tested about 130 serum samples from healthy individuals, and it was difficult to find one who doesn't have antibodies against the lysin. So how is it going to play out? Have you looked at whether your product might be neutralized?
That's been looked at in the animal studies. There are anti-drug antibodies that form and that are formed that exist ambiently in nature because we're deriving license from natural products. But in the animal work that we've done, the anti-drug antibodies are not neutralizing, non-neutralizing. We have our ongoing phase 2 study. Happy to say from an immunogenicity hypersensitivity type perspective in our study, I think, although we're blinded, I can say that we have seen no reports of hypersensitivity events considered related to study drug by ourselves or by investigators. So from a hypersensitivity perspective, we haven't seen anything, knock on plastic here. And we're pleased with that. We're pleased with the progress from a safety perspective. And in terms of the emergence, drug-emergent anti-drug antibodies, yes, as I said in phase 1, healthy volunteers, maybe 60% or so, 69%, developed ADAs post-dose. They emerge at day 14 to 28. So for the current treatment paradigm, single dose, early in the course of therapy, treatment emergent ADAs are not expected to be an issue, and we're going to work on what that all means for redosing, extended dosing, et cetera.
But pre-dosing, they were seronegative?
In phase 1, the patients were seronegative. In phase 2, because of the practicalities of treating patients, we're not excluding anybody on that basis. We're collecting the information, though.
Yeah. Next question.
Menachem Shoam, Q2 Pharma. I have 2 questions. One is specific for Kara. What was the standard of care in your phase 2 clinical trial?
Basically, similar to the daptomycin phase 3 study, basically for methicillin-sensitive staph, a range of semisynthetic penicillins or first-generation cephalosporins are permitted, and to treat MRSA, vancomycin or daptomycin are permitted. We needed to leave sufficient flexibility to allow prescribers in 14 countries to find something that they could use, because not all these agents are approved everywhere or available. But we wanted to keep it within a range of what's generally guidelines.
Okay, and then a more general question. We heard a lot about synergy with antibiotics this afternoon. What is known about the mechanism of action of potentiation of antibiotic efficacy?
It depends on the mechanism of action of a particular biologic. I mean, for antibodies, there's about a half a dozen of mechanism of action that it could work on, and a combination of it, right? Antibodies could prevent host entry, could block toxic pump, could neutralize toxins, could stimulate opsonization, or a combination of those. And so it depends on the antibody that is generated. You could imagine a scenario where clearly it helps to prime the antibiotic to work better, right?
Yeah.
It could be related to biofilm inhibition, which would make it easier for the antibiotic to penetrate the cell.
That's right.
Yeah. So for lysins, because it's an enzyme, it acts on the cell wall. We think even at sub-MIC levels that the lysins may not kill, but may in one way or another damage the cell wall, render it more susceptible, just broad strokes in general. And we're looking at, Ray is our head of micro and research is looking at ways to further elucidate that.
Maybe last question.
Yeah, maybe this one's for Wayne. So when you have the supportive care for AKI or multi-organ failure where you tend to have late mortality, how good was the epidemiology when you're looking at that association with SOFA and when you would tend to see mortality, and how did you end up at 28 days? It's kind of a standard endpoint. Is that really ideal?
It probably is not, but it was a way to incorporate. So for the composite endpoint for NSTI, it was a way to incorporate mortality but not make it the definitive endpoint, again, because of the difficulty in sample size. But mortality obviously is important. You also want to make sure that you're not going in the wrong direction from a safety perspective. We do actually have a 3-month follow-up for all the patients in the NSTI, and the follow-up has been unbelievably great. I've done a lot of clinical trials, and I've never seen this percent of follow-up except in vaccine trials. So for AKI, we also have a 3-month follow-up. But as I mentioned, we're starting to look at a large retrospective database, working with Optum Life Sciences, which has got the huge database from United Healthcare, and trying to look at mortality further out, 6 months, 9 months, and then also look at other endpoints such as number of hospitalizations, number of readmissions, things that impact a number of outpatient sort of use, to sort of get a better feel of how the endpoint can really be correlated with not only short-term outcomes in hospital mortality and so on.
So in that 3-month study, you are seeing more differentiation between the control groups. So, I mean, in the
not as much as I anticipated. A lot of the mortality is definitely occurring before day 28, and less so. And I think it may have to do with the fact that patients with underlying comorbidities, they have NSTI, their outcomes, if they're not doing well, the families seem to withdraw care, I think, and not let the individual linger, as opposed to sort of more general sepsis protocols where the patients kind of linger and linger and linger, and then finally, the parent, 2 months later, they withdraw care. So I think part of what we're seeing, however, there's data from Denmark where they did sort of their own clinical trial, and they also have this large database that they've been developing that looks at mortality rates, and they see definite differences between day 28 and month 6. You're picking up about another 10, 15% mortality in those patients.
Okay. Okay, great. Thanks to the 3 presenters and our reactors as well. We're going to go ahead and take a quick break. Let's come back for the penultimate session at 3:05. Thank you.
Thanks.
Fourth session. This session, we're going to take a bit of a turn and talk about products that are intended to prevent infection, so no more in combination with antibiotics, I think. So we're talking about prevention. Again, we'll hear from 3 companies who are developing or in the process of developing really interesting compounds, and then we'll have a full panel to react and discuss. So joining me for our first presentation is Dave Mantis, Chief Development Officer at Arsanis Biosciences. Stuff. Okay.
Thank you, and thank you for the opportunity to speak today. Some of it will be familiar. I'll try where there's overlap with previous speakers. Vu Trong did a great job giving us an overview of monoclonal antibodies for infectious disease. I will put in just a couple of my own editorial comments on that. And I'll focus on our lead compound. So many of us at Arsanis were at Cubist Pharmaceuticals working on daptomycin in the early 2000s through its purchase by Merck. And if we think about an ideal anti-infective, and of course this is somewhat jaded because we're focusing on antibodies, we think of something that only targets the pathogen of interest, has minimal potential for resistance development, no off-target effects, for example, typical toxicity you would see with a small molecule, well-established manufacturing and quality, single-dose PK. Give a single dose, protect for 21 days, or treat for 21 days, or even a month. What's interesting is nowadays, if you bring something, at least to investors and even maybe to the regulators, although they wouldn't say this, that isn't an antibiotic as a solution, makes it attractive. And a safety profile, and this is why we're talking about prevention in this session, a safety profile that allows preemptive therapy, assuming at-risk subjects can be identified. So you can treat and put people at risk, but the risk is so low, it's an acceptable risk to protect from, for these patients, what is a high-risk, high-mortality, morbidity event. I've summarized and taken a slightly different approach from others. I've listed some of the challenges up front. This is a novel approach and has an appearance of novelty. Some of us have novel mechanisms of actions. We are an anti-toxin monoclonal antibody, or at least our lead is, although some of our antibodies actually directly impact and are directly cidal to bacteria. I would point out that that makes it difficult to fit it in the 4 boxes that John elucidated earlier. It doesn't mean that's not useful for us to discuss these products, but some antibodies may fit into multiple boxes. How much targeting is too much targeting? There's this balance. We have antibodies that will hit single clones of bacteria, which means it would be difficult or very impossible, potentially impossible to develop for certain indications, certainly as monotherapy. People are always worried about the cost of goods of biologics, particularly monoclonal antibodies. But with more than 70 monoclonal antibodies on the market now, the cost of goods is averaging around $200 to $300 a gram. It wouldn't be shocking to imagine in 10 years that drops yet another order of magnitude. And then, of course, all the normal challenges people have talked about today of anti-infective development. And for us, some of the challenges of prevention development. How do you figure out who's at risk for the disease? Can you enrich for high risk? And what are the real costs of the disease? Vu Trong mentioned that marketing a drug for prevention might be a challenge. It would be if the numbers needed to treat is 40 or 50. But we believe by enriching that at-risk population, understanding who's truly at risk for, in this case, pneumonia, we will be able to get to numbers needed to treat that is acceptable economically. There's been discussion of cocktails earlier. This product I'll talk about, ASN 100, is the simplest cocktail you can make, or maybe that would be 2 antibodies together. And I primed ourselves for the question. My first question of the day was about the combination product guidance that FDA uses. And that is one of our, and I think one of our key regulatory challenges moving forward. If you've ever seen me speak before, you could know I could easily do 10 or 15 minutes on the question of who you call in a non-traditional antibiotic. Because antitoxins have a 130, 140-year history. The first Nobel Prize in Medicine was given to Emil von Behring for his antitoxin against diphtheria and tetanus. So developing antibodies to neutralize toxins to prevent and treat bacterial diseases is quite traditional. Monoclonal antibodies are a bit newer. Today I mentioned, I think this number has risen considerably, 70 billion in sales. 4 approved for infectious disease indications. 1 in 1998, palivizumab for RSV, 2 for anthrax, and then bezlotoxumab for C. diff. These latter 3, all antitoxin approaches. Neutralizing the antitoxin helps either treat or helps prevent the recurrence of disease. Which indication are we looking at? We're looking at mechanically ventilated patients. Already, we're talking a high-risk, high morbidity, and of course, high-cost population. And we're looking at Staph aureus pneumonia. Right now, we take a number of physical actions to prevent pneumonia in mechanically ventilated patients. We suction the endotracheal tube and remove the aspirate. We raise the head of the bed, for example. And these VAP prevention bundles I've seen on doors of ICUs around the world, India, the US, Europe. However, despite these efforts, Staph aureus colonization happens in the endotracheal tube, the so-called superhighway for infection into the lungs. That plastic tube that helps the patient with air or oxygen, but also allows access for bacteria. This colonization of the endotracheal aspirate usually happens for Staph aureus in the first 7 days of mechanical ventilation. And we now know from epidemiology studies we've done, 30 to 40% of these patients who become heavily colonized will progress to pneumonia. Now, Staph aureus pneumonia, there are a number of treatment options for VAP, but none of them have reasonably good outcomes. And I quoted Sumati, unfortunately, on that, reasonably good outcomes, because in this particular space, just like bacteremia and endocarditis, current antibiotics, even the latest antibiotics, still have unacceptable mortalities. Mortalities as high as 30 to 40%. Patients who are successfully treated and survive VAP are often back in the hospital within the month and cost the hospital upwards of $50,000 or more. Our goal is to come here where there are no approved therapies, identify at-risk individuals who are heavily colonized in their endotracheal aspirate with Staph aureus. We defined heavy colonization as 10 to the 5th CFUs per ml of ETA, and prevent this progression. How are we going to do that? And how are we trying to do that? I'll spend a lot of time on this cartoon. I learned most of my science watching cartoons, also most of my knowledge of classical music and opera. So pathogenesis of Staph aureus pneumonia, we see the epithelial cells of the lung here in pink, in gray, and I can go over here too, although this doesn't work as well over there. Those are Staph aureus cells. Staph aureus brings an arsenal of toxins to the pathogenesis of pneumonia. Vu Trong mentioned one, HLA, or alpha-hemolysin. The goal of this pore-forming toxin is to break open the epithelial cells, release nutrients to feed the colony. That would be bad enough. Staph aureus makes 5 additional toxins to cause pneumonia. These are the so-called leukocidins. The role of the leukocidins is to destroy immune cells that come to the aid of the lung to try to remove this Staph aureus. Some of you will know PVL. That's a well-known toxin that causes a particularly nasty necrotizing pneumonia in many cases. It's only actually encoded in 5 to 10% of staph strains. This last leukocidin, LeuKGH, is actually encoded in all known strains of Staph aureus. LeuKGH is particularly potent at destroying phagocytes. In fact, the target for that toxin is upregulated during infection. Took me a while to think about that, to imagine that Staph aureus, a uniquely human pathogen, has evolved with us such that it's created a toxin that gets more potent when we try to fight Staph aureus. That's a pretty elegant and nasty set of toxins. The mechanism, so this is our mechanism of disease. Our mechanism of action for ASN 100 is to neutralize these 6 toxins. So there may be colonization, but colonization cannot progress to infection. HLA is neutralized such that the epithelial layers are protected, and the leukocidins are neutralized such that the immune cells can do their job. I won't go through all of our preclinical data, but we've studied the whole rabbit model. In rabbits, you can cause a necrotizing pneumonia where all the rabbits die within 16 hours of infection. If you pre-treat them with an HLA-only mAb, you can get full protection with some strains, partial protection with others. If you neutralize all 6 of these with the 2 antibodies that make up ASN 100, you get 100% protection against all strains we've tested. And not only that, 96 hours after this experiment, you can look inside the lungs and there are no bacteria. There are no bacteria in the blood. There are no bacteria in the peripheral organs. Simply disarming Staph aureus from all of these toxins allows the rabbit immune system to remove the Staph aureus and remove the infection and remove the bacteria from the whole animal. I will focus on one set of in vitro experiments because they do talk about why we need 2 antibodies to neutralize all 5 of the leukocidins. This experiment ignores HLA. It's only looking at the protection of phagocytes in an in vitro assay. You grow strains of Staph aureus in a physiologically relevant medium, and you take the supernatant, which is enriched in these leukocidins, in these toxins, and you see how much antitoxin, how much antibody does it take to neutralize that toxin to protect the phagocytes. Each one of these experiments is a different strain of Staph aureus. This is 12 experiments out of almost 50 we've done. You'll see in red ASN1, an antibody that neutralizes HLA and 4 of the 5 leukocidins. You can see the protective effect it has. In aqua, you see ASN2, an antibody that neutralizes LeuKGH, as I mentioned, the most potent of the leukocidins. And then in blue, we see the combination. The 2 antibodies combined is ASN 100. And you can see that depending on the strain, you might need more of ASN2, or actually this is a 1-to-1 ratio. ASN1 or ASN2 may do more of the heavy lifting in terms of neutralizing toxin. What's interesting is there are several examples that look like classical synergy. And it is not really synergy, it is evidence of how potent these toxins are. Staph aureus makes more toxin than it needs to. So for example, in this lower left here against this USA 400 strain, if you neutralize all the 4 leukocidins covered by ASN1, There's enough Luc-GH to destroy all the phagocytes. If you just neutralize Luc-GH, there's enough of the other 4 leukocidins to destroy all the phagocytes. You have to combine them to protect all the phagocytes. We took this in vitro data, the rabbit data I mentioned. We did a phase 1 study. These are 2 fully human monoclonal antibodies. You would expect about a 3-week half-life. We confirmed it. You would expect, given that they're fully human and targeting a bacterial toxin target, that they would be safe and well tolerated. They are. We found no dose-limiting toxicities. In fact, no related toxicities at all. We've now taken this product into a phase 2 study for the prevention of Staph aureus pneumonia in mechanically ventilated patients at high risk. The way the study works is you're enrolled. If you're mechanically ventilated without pneumonia, we allow concomitant antibiotics if they're not being used to treat pneumonia, if they're just on board. And someone said earlier, just about everybody in a US ICU, and this covers other countries as well, is on one antibiotic or another, maybe not for infection, but perhaps to protect the line or for fever of unknown origin. We screen them for up to 14 days, look at their endotracheal aspirate using standard microbiological methods to see are they heavily colonized with staph. If they become heavily colonized with staph, and most of that happens in the first week, they become eligible for randomization. You're heavily colonized, you cannot have pneumonia, so we double check diagnostically for that, and you're randomized 1-to-1 ASN 100, a single IV dose, versus placebo. We then follow you for 21 days, and we're essentially counting pneumonia. We don't have to worry about pneumonia cure, we simply have to worry about the diagnosis of pneumonia by the investigator at the site. We have powered this study to detect a 50% reduction in the rate of Staph aureus pneumonia in these patients. We expect an interim readout by the end of the first half of this year. If you look at the calendar, you know that must be forthcoming. And a final readout on this study at the end of the year. So the efficacy assessment is Staph aureus pneumonia, but we are very interested in many of the secondaries, which will include things like duration of ventilation, duration of hospital stay, but also all-cause pneumonia. There's been some publications to show that if you neutralize HLA, you protect the epithelial cell and make it more difficult for gram-negative infections to happen. We would position this product not in all patients ventilated, but only those at high risk. We expect that to be about 20%. Our data suggests that up to a third of them will progress to pneumonia. We're planning on at least halving them. You can do the math on the numbers needed to treat and realize that this makes a lot of economic sense for hospitals and payers. Of course, it makes sense for patients because you never want to end up with pneumonia because even well treated, it has massive issues. And this complication, as I mentioned, can cost the hospital up to $50,000 of incremental costs. That concludes my presentation. Again, I hope I made a case for why monoclonal antibodies made sense 130 years ago as antibodies, and mAbs make sense now. Thank you.
You made an excellent case. A little over 10 minutes, but you made an excellent case. As we near the end of the day, I think we all want to leave at least by 5, if not a little bit before. So I'll ask the next 3 presenters to try to stick to the 10-minute allotment that we talked about before. Okay, so for the next session or next presentation, we have Lee Jones as founder, president, and CEO of Rebiotics.
So hello, everyone. Thank you for sticking with me this late in the day. Today, I'm going to tell you a little bit of background about Rebiotics, who we are, and how we got into this conference. We are revolutionizing the treatment of debilitating diseases by harnessing the power of the gut microbiome to greatly improve lives. I founded the company in 2011 with the idea that I wanted to bring a microbiome-based therapy to the marketplace to have wide access to patients. Since then, we've developed a proprietary microbiome-based drug product platform to rehabilitate the human gut microbiome. We've demonstrated efficacy in 3 phase 2 clinical trials to prevent recurrent C. diff and are currently in our phase 3 clinical study. C. diff is an interesting disease because a patient can get that disease typically because they've been on some form of antibiotics or have some other illness that they were treated for. They get the disease and the disease is treated by more antibiotic therapy. When they find out that they can get the disease again and again and again, these people start to panic, and traditional antibiotic therapy really doesn't help to cure them. It just knocks back symptoms till the next time they get it again. So it's gotten to be a real issue, and it's continuing to grow. Today, we are the most clinically successful microbiome company looking at the prevention of recurrent C. diff. So what is the human gut microbiome? Many of you know trillions of microorganisms live on our surfaces, and they work in conjunction with each other and the host to keep us healthy. And that whole community, the most diverse and dense community of microbes, are found in the human intestinal tract. That community plays a big role in the immune system, and as the research has gone on, there's more and more evidence to show that it has a big interaction with the host. That community can be damaged or be dysbiotic via antibiotics or viruses or environmental factors, et cetera. When that happens, that dysbiosis can lead to C. diff infections, which is a #1 healthcare acquired or hospital-acquired infection in the U.S. We have learned from our own research and others that restoring a healthy microbiome can actually prevent recurrent C. diff. And the question we have, and we can ask today, is can the Microbiome Restoration Act to prevent other infections? So there's a predicate procedure that we learned about called fecal transplants, where healthy human donor stool is injected or infused into sick patients' intestinal tract, and it's been shown to treat disease. So when we got started with this, we thought, oh, here's a perfect example of something that already works. Our goal was to create a product that mimicked that healthy human gut microbiome. We spent quite actually months and years trying to figure out how to produce this product so that it did mimic the gut microbiome, and spent quite a bit of time understanding how to make it, how to preserve it, and then how to deliver it to the patient. We've been successful in that. We have a high diversity and high microbial population per dose. We preserve both the spore and non-spore forming organisms, and we have consistent bacteroides per dose, which we found was missing in the C. diff patients. One of the challenges of this product is it's based on live organisms. So the question is, what is a dose? Gets to be really interesting, because unlike normal drugs, when you put in one microbe, you get billions. So how do you even start out thinking about what a dose is in this kind of candidate? So what we've done since the beginning of our clinical program is to gather samples of the product, then the patient fecal material before they're treated, and then post-treatment. And what we've seen is that our product has a significant impact on dysbiosis. What you see here is on the left is what a typical product profile is. It's relative abundance of 4 different taxa. We have what you see in the sick patient is a typical profile of a sick patient, very dysbiotic, a different relative proportion of organisms. And then after treatment, what we see is that person who's successful progresses to look much more like the product and much more like a normal, healthy person. We can have a pretty robust clinical program. I bring this up again because this is the basis for a lot of the data I'll be showing you. The important thing to know about this is that we have a consistent product, the same manufacturing process and same release criteria throughout all of our studies, and have looked at a consistent patient population. The first study that we did was the first multi-center randomized, or multi-center, in this case, open-label, non-controlled study that the FDA asked us to do, really just to look at safety, because this was a new therapy. We advanced onto a multi-center randomized double-blind placebo-controlled trial, and as we've gone forward in our clinical program, we now have really good understanding of who that patient population is and what the results we can expect. So we expect to have a successful phase 3 program. So the randomized double-blind placebo-controlled trial was the first one of its kind. Going through the FDA, we conduct this trial both in the US and Canada. It was a 3-arm study. Our goal here was to understand whether 1 dose or 2 of our product made a difference against placebo. And placebo in this case was every one of these patients had been on some kind of antibiotic treatment, were removed from their antibiotic treatment, and then given either an active product or a placebo. The placebo was our product minus the microbes. Anyone who failed the blinded portion was allowed to advance into open label treatment. It's very difficult to enroll placebo-controlled patients in these types of studies where the patients are desperate for care. So by allowing randomized patients to remain blinded and go into open label treatment if they failed was an easy way to improve our enrollment. What we found was a little bit of a surprise that 2 doses of our product was not better than 1 dose for all populations, so there was no escalating dose effect. And we found that a single treatment of RBX 2660 was significant over placebo. We also found out that the placebo rate was much higher than anybody anticipated. And so we've now verified that this is similar to what you would expect in a standard of care population. But more importantly, this leads me to this analysis, is that we looked at the microbiomes of all the patients and then treatment over time. What you see in the dots on the left-hand side, each dot represents a microbiome from a patient in the Human Microbiome Project. These are a number of healthy humans, and their microbiomes were profiled. Our patient at baseline are the open dots on the right-hand side, and the triangle is kind of the center of mass of that patient cloud. What you can see is the farther apart they are, the more dissimilar they are, and so our patients at baseline are very different than the healthy human population. At 7 days post-treatment, you see that the microbiomes of the patients start moving quite closer to the healthy population, and that trend carries on through 30 and 60 days. So after treatment, the microbiomes of the patients more closely resemble a normal population. So then the next question became, can the modulating the microbiome have an effect on resistant organisms or resistance genes? So we know that antibiotics facilitate antimicrobial resistance in the microbiome, and that the microbiome functions as a reservoir of antibiotic resistance because the AR genes can be transferred from benign microbes horizontally into pathogens. So we started looking at this community resistance, and what you see here is that baseline, again, that first box, it's about 10 patients, has a high level of resistance genes. And then 7 days post-treatment, those resistance genes greatly go away, and the same thing you see at 30 days. We have followed these patient populations out, in some cases, to over 2 years, so that data is being analyzed, and I don't have it to share with you here. But what we have found is that the closer that the recipient taxonomy is to the donor, the fewer resistance genes were detected, and this is actually correlated with statistical significance, and it follows a pattern so that as we see as time goes on and that patient moves closer towards what the normal patient would look like, the resistance genes go down. We've validated that with other clinical study data. Today we're developing a prototype, what we call Microbiome Health Index, to measure response. So what we've done is taken our data and looked at, it's a single measure of collective changes in the key taxa for these different patient groups. You can see on the left, baseline patients, everything's really low on the MHI. Our product is a purple, and then the green is the Human Microbiome Project. What we've seen from this is that even as early as 7 days, we can start differentiating the successful patients from the people who are not going to be successful. We've actually looked at this on the placebo patients as well and find that even though there's some placebo patients that are successful, they never get back to a healthy microbiome index. So now we're starting to look at this in relationship to antibiotic resistance and whether or not we can take this into other indications. So what kind of conclusions and next steps? When treated with RBX 2660, significant reductions in CDI infections were shown in recurrent patients over standard of care antibiotic therapies. In the context of preventing CDI recurrence, this may be the first therapy with potential to reverse the enrichment of antibiotic-resistant genes. The distance from the 2660 composition can measure the treatment effect. So the closer the donor, the better the treatment success, and has fewer AR genes. And this correlation was repeated with additional clinical data sets, and we are also using outside, not our own clinical data, but other people's clinical data to validate these concepts. So we are now entering a new proof of concept clinical trials to test our product and other infections. Thank you.
Great, thank you. Our next speaker is Michael Bevilacqua, CEO. I had that perfect when I was practicing. CEO, CSO, and co-founder of Amicrobe. Got that one right, not A Microbe.
Thank you very much. Thank you. The World Health Organization estimates that there's more than 300 million major surgical procedures performed each and every year. Add to this the fact that there are tens of millions of traumatic injuries. Maybe divide by 365, and you come out with about 1 million times each and every day that our skin, our natural barrier of defense against microbes, is broken by the wounds of surgery and trauma. Each one of these opportunities allows microbes to get into those tissues, start to set up house, and maybe begin the progression toward infection. At Amicrobe, we are focused on the development of antimicrobials that are purpose-built to go into these wounds of surgery and trauma and help stem the tide, block the progression toward infection. We work at an interface between biotechnology and material science. Like biotech companies, we make proteins. Like material science companies, we design these proteins to have physical or materials properties. We have 2 lead product programs, Amicidin Alpha Surgical Gel and Amicidin Beta Solution. Both are designed to have broad microbicidal activity. Both are designed with different optimized physical properties for performance in wounds. And of course, they're both designed to try to achieve safety. We're very thankful and grateful to both the Department of Defense and to CARB-X for their support of these programs. So why are we doing this? Well, we're doing it because microbes really like wound environments. They get in there, they find their food sources, they find moisture, and they don't find a competing microbiome, so they can start to set up their homes. All the way back in World War I, a French surgeon and Nobel laureate, Alexis Carrel, was working with the great British biochemist Henry Dakin, which led to the development of Dakin's solution. But in those surgical procedures, he said, surgical infection at the outset is always local, since the microbes are, so to speak, within reach of the hand. The question then is simply how to destroy them without harming the tissues. Well, we think Carrel's question is as important today as it was in World War I, and we think a new technology can help answer his question. So let's look a little bit further at the progression of infection that can occur in surgery and trauma. Of course, it begins on the left with a surgical or traumatic wound. Then these wounds can become, and usually do become, contaminated at some level with microbes. If things go poorly, you wind up with a local infection. If you're thinking about limbs, it would be things like skin and soft tissue infections or osteomyelitis. And if they continue to go poorly, you can wind up with the very bad situation of systemic invasion and sepsis. So what determines whether these things go well or go poorly? There's several factors, but some of the top factors include the number and virulence, as you've heard about here today, the number and virulence of microbes that get into those tissues, the overall health of the patient, and the interventions that the healthcare providers can bring. How good are they? I'm sure there can be a lot of debate about exactly how good are antiseptics and antibiotics in different settings, but I think there's general agreement that today's antiseptics, alcohols and chlorhexidines, perform quite well on the far left. Certainly today, we still have as our only major choice, when you get toward the right, the antibiotics. You're hearing today about some great new technologies that could add some additional weapons on the right side. We at Amicrobe very much believe that there is a therapeutic gap in the middle. And we're designing our amicidins, again, to go directly into those wounds and hopefully to help block the progression of infection, particularly steps 2 and 3 for our alpha program and step 3 and 4 for our beta program. Okay, so amicidins are synthetic proteins. They're relatively large. They're diblock architecture. So both amicidin alpha and amicidin beta have a cationic or positively charged block, shown in blue, and a hydrophobic block shown in red and pink. Amicidin alpha is the bigger of the molecules. It's about 170 amino acids, about 21 kilodaltons, and both the molecules are designed to self-assemble in aqueous environments into multimeric structures. So alpha self-assembles into fibrillar structures and forms a natural barrier gel at 2-weight%. It doesn't need any excipients to gel up. It's just a fibrillar gel, hydrogel that is antimicrobial. Amicidin beta, the smaller molecule, around 100 amino acids, forms micellar structures, but very importantly, it was designed for and has substantial surfactant property. It's this combination of microbicidal activity and the aphysical property that we believe works to enhance intra-wound performance. A side note, but I think an important one, is they're manufactured with robust polymer methods, much more like the methods used to make nylon than the methods to use to make other biologics in the biotech industry or synthetic peptides. Big one-pot polymerization. The good news of this is that it seems to be, and we're working right now with contract manufacturers, very scalable, very cost-effective, and should enable metric ton production. Of course, they have to be antimicrobial, and they are. They're good antimicrobials. They're cationic amphiphiles for people who follow that literature. They're quite robust against a number of microbes, including the key wound pathogens. A partial list includes MRSA, vancomycin-resistant enterococci, and some of the worst-acting gram-negatives, including Acinetobacter and P. aeruginosa, including their multi-drug-resistant strains. In the middle, you see a simple live dead stain with fluorescence. On top, all the P. aeruginosa all alive. On the bottom, they're all dead. They've been permeabilized, and green dye is coming in at 100 micrograms per ml of amicidin beta. A little cartoon for mechanism is shown on the right. Okay, so what about the products? The amicidin alpha surgical gel, designed to be an active microbicidal barrier for intraoperative use to reduce the incidence of surgical site infection. We anticipate that the surgeons immediately upon the first incision will start to apply to the tissues, maybe once or twice during surgery, and right at the end, rinse things out, do a last coat, and then close up and move on. In addition, on the lower left, in addition to the microbicidal activity and tissue coating properties, this product is designed to be shear thinning, like paint, for easy application and spreading on the tissues, transparent for visualization of the coated tissues. It is easy to remove by irrigation, but it is also, we believe, fully bioresorbable. It's made out of 2 amino acids, fully bioresorbable when left in place. On the right shows a simple ex vivo assay on porcine tissues where at 2-weight%, our clinically anticipated concentration, the amicidin alpha barrier completely blocks deliberate contamination with Pseudomonas. As we go to amicidin beta solution, it forms the even more potent microbicidal solution. It's our best antimicrobial and designed with the surfactant properties. In this case, we're looking for use in intraoperative and postoperative treatment of tissues that are already contaminated or infected. Think things that are heavily contaminated or have biofilms, or maybe even early infections, focal infections, depending on whether surgeons are going in or not. Shown on the right is the fact that it works very well on biofilms. This is a simple Pseudomonas in vitro biofilm study. Okay, let's go back to this notion, though, of interrupting the progression of infection. The progression of infection was talked about at several sessions today. We think it's very important. Infection doesn't happen instantaneously. It happens over a little bit of time, especially here in surgery and trauma. We've worked with the University of Cincinnati and been supported in this work by the Department of Defense on a model that was originally published by Kavanaugh in 2009. It's a good model, and we've pushed it a little bit. We like it very much because it reliably demonstrates progression based on microbial number and virulence. We get to test the thing that I think is so hard, or not so hard, but so important right now, is how much and how often. How much material should we put in there? How often should we do it? And that's what we're using this for, and we're making progress. When amicidin alpha surgical gel is used as we expect to use it clinically, it seems to work quite well. It limits the initial contamination. Again, this is a deliberate contamination. We do it low inoculum and high inoculum. This happens to be a high inoculum, 10 to the 7th Pseudomonas. It limits initial contamination, but very effectively blocks progression. So if we look after 24 hours in control animals, that's the gray bars, after 24 hours, there's a bad local infection. So the muscle is completely infiltrated, tremendous inflammation, and quite frankly, a lot of necrosis. Over 100 million microbes recovered per gram of tissue. In addition, the animals are septic, and we can recover microbes from spleen, liver, kidney, and lung. On the other hand, when we treat with our amicidin alpha surgical gel, we get a dramatically reduced number in the muscle or on the muscle. In fact, we're now down to 10,000 to 100,000, greater than a 3-log or 99.9% reduction. And I think as importantly, or more importantly, we find no progression to sepsis in these animals. They look healthy, and we're not finding any of the microbes systemically. That's what we're hoping to do with the product. That's where we're going. We get also terrific results with amicidin beta. This is, again, going in after we contaminate. So we do, again, low contaminations, high contaminations. This is a high contamination with P. aeruginosa. And by 48 hours, these animals are all septic. The control animals have a 50% mortality. In the amicidin beta treated animals, we, again, have a much better story. So in amicidin beta treated animals, the preclinical scientists doing the studies said they did not appear septic at all. All did survive. When we look in the wounds at 48 hours, we see a very different picture. On the left, the control wounds show that dusky red of hemorrhage and inflammation. On the right shows a much cleaner looking pink wound in the amicidin beta treated animals. Okay, that's what we are about. We are making purpose-built antimicrobials, a little biotech, a little material science, and we're doing them for the wounds of surgery and trauma. We hope that these two complementary products, we're dedicated to developing them. We hope that the two complementary products will find important uses. The amicidin alpha surgical gel, we think, is going to be used primarily in situations where the surgeons feel they can beat the microbes to the tissues. So class 1, class 2 procedures, clean or clean contaminated procedures, things all to prevent surgical site infection. Amicidin beta, on the other hand, we anticipate is going to be used when the surgeons know the microbes have already beaten them to the tissues, or maybe heavily contaminated or already set up focal infection or biofilms. Where do we think? Treatment, surgical treatment of periprosthetic joint infection, treatment of superficial and deep incisional SSIs, and some very dirty traumatic wounds. We are committed to doing this, and we hope that over time these two products will end up helping to prevent and treat life-threatening infections of surgery and trauma. This is the Amicrobe senior team. I want to also thank again both DoD and Carbex for supporting our work. And finally, I'd like to thank all of you for taking time to listen to the work we're doing at Amicrobe.
Okay, you can stay up here.
Okay. Great, thank you. I'll invite our speakers back up to join me here on stage, and then our additional two panelists, including Devrick Anderson, Associate Professor in the Division of Infectious Diseases and Department of Medicine at Duke University, and Director of the Duke Center for Antimicrobial Stewardship and Infection Prevention. Also, Joe Larson is Acting Director of the Division of Chemical, Biological, and Radiological and Nuclear Medical Countermeasures within BARDA, the Biomedical Advanced Research Development Authority. So let me ask Devrick maybe to give some reaction or comments based on what you saw from the presentations.
Sure, and first, thanks again for having me here. I guess my role in this panel is from the more clinically oriented stewardship and infection prevention side, and again, hearing the presentation I think is really exciting. I drink the Kool-Aid of prevention, and I'm really excited to hear about new and exciting strategies. You know, again, we can think of new strategies for treatment, but if we can just prevent the infections to begin with, then we're making some major progress. Again, as the representative of clinical infection prevention and stewardship, it's probably worth mentioning there, you know, a few items about that field that I think are relevant for this conversation. And again, like any field, we've got our own kind of, you know, hot topics for debate and controversy and differing philosophies. And you can go to various meetings and hear pro-con, you know, debates about some of these exact things. One of the things that comes to mind is, you know, this debate between is prevention, do we need new technology for prevention, or do we need just better implementation of current strategies? And again, my strong suspicion is that we need both of those things, but I think in the field you can get some pretty strong advocates for both of those. Again, it's all technology, and I probably would say that the discussions we're having today are certainly on that side of the spectrum. versus that bundle that we know about, for example, for ventilator-associated pneumonia. And frankly, we just don't do a good job of always implementing what we consider to be best practices. And so where should our efforts lie, I think, is an issue that gets debated. And then the other topic, again, regularly debated, is the notion of vertical interventions or horizontal interventions. Do we find a specific pathogen or a specific infection and vertically try and prevent that from occurring, or can we develop a horizontal prevention strategy that really covers multiple organisms and or multiple types of infections? And I think we heard a nice mix of those different strategies today, which I was really, again, excited to hear. Some examples outside of this kind of highly technological discussion today are hand hygiene is probably the ultimate horizontal transmission intervention, whereas, again, hotly debated the notion of MRSA, identification, screening, and decolonization as a very vertical intervention, and how we, which one is right? Again, probably there are scenarios where both are going to be important for us to try and pursue, which really leads me to my last point before I kind of relinquish the microphone here, that my, again, strong suspicion with all of these technologies, as someone who has to be in the hospital and is confronted with mostly new drugs at this point, but how we decide whether they are ultimately used in the hospital is a very tricky issue to deal with, and unfortunately is colored by the siloed budgetary approach that most hospitals take. And so, again, let's use a recent example on the drug space, but C. difficile and fidaxomicin, it has really good data to suggest it's probably better for recurrence than vancomycin, yet it's so extraordinarily expensive compared to the other drugs that just does not get used frequently. The pharmacies at our hospitals, that's who gets the hit for these costs, because it's completely siloed away from the outcomes and safety and length of stay. Those people don't talk very regularly within hospitals. So it's a difficult discussion to have, difficult decisions that are not always terribly well informed, because you just don't always have the right people at the table to make those. So my strong suspicion is that what we need is what was alluded to at the beginning by Dave. When we start to think of these newer technologies, I think we're going to be in much better position if we have an ability to find those high-risk or at-risk patients and first start to use them in those patients, getting buy-in within the hospital setting, and I think inevitably we see expansion from there. Thank you.
Great. Joe? Yeah, thanks for having me. And I think I'm going to kind of start broad and then go narrow, in the sense of starting with all of my comments on everything that I've heard today. So for most of you know, BARDA makes investments in antibiotic development. We've been doing that since 2010, and most of our investments are in kind of precedented existing classes. None of the stuff that we're talking about today, with the exception of ATOX, which we invested in through one of our innovation programs a number of years ago. And so, we've kind of sat on the sidelines of this non-traditional space for, 3, 4, 5 years now, wondering what role we were going to play, and really thinking about the role a public funder should play in this, in terms of trying to de-risk and carve a development and help define a regulatory path for some of these approaches, because we think that that's ultimately in the public interest. For some of these things, and I'll get narrow when I start talking about these technologies in a minute, I think there is a definable path forward that can certainly allow a clinical trial to be done. Now, then you get into the economic barriers of whether or not people are going to reimburse for a preventative therapy, which is a whole other thing to contend with, and is something that we are concerned about in terms of sustaining these products after we make our initial investment. But for some of these others that we've heard, a lot of what I heard today gives me a great deal of pause about whether or not we would be able to invest, because, or get over kind of that activation energy that would allow us to invest, because of just what we see as some of the challenges in being able to do clinical trials. I hear things like antibiotics, and then plus something else, you're going to have to show a substantial benefit, and that gives me, I start having a reaction to that, akin to when we had to develop raxibacumab for inhalational anthrax, and we had to basically show that raxibacumab provided an additional benefit to existing antibiotic therapy, and this was using the animal rule, so it's not patients. But we had to basically wait till the animals were so sick that we knew the antibiotics weren't going to be effective and then provide the antibody. And you know, we did that. A lot of animals were sacrificed in that process, and we still weren't able to demonstrate a statistically significant benefit that the antitoxin therapy in conjunction with antibiotics provided a superior outcome. There was a trend, and that ultimately was sufficient to go forward with approval. So I think about things like that, and then think about having to do that clinically, and think about the cost. And I also think about the timelines that we would have to do for some of these, of enrolling hundreds, if not thousands, of patients to get a difference that's observable and statistically controlled. And then when we start thinking about that, I start thinking about my bosses yelling at me for not, you know, for using taxpayer money in a way that's not going to result in any sort of productive outcome. So I think collectively, there's a lot more work to be done. I'm pleased to see that FDA is arranging a workshop on the 21st and 22nd of August to continue this conversation, because we're ultimately, if we don't figure this out, there's going to be a whole pipeline or you know, a bunch of things, a bunch of products that we are developed, whether through CARB-X or through NIH, are just going to die on the vine. And for some of these preventative therapies getting more narrow, I think, yeah, certainly there's a path forward for a microbiome C. diff therapy. I think that's great. I think that's something that BARDA would be interested in taking a look at. For these monoclonals and doing preventative, I mean, I think it's really, I would be really interested in hearing, you know, what the estimated patient sizes are going to be required for your phase 3 and what your estimated cost you think it's going to take to do that and how long it would take. That doesn't mean it's not feasible. It just may not be commercially attractive. And then for these wound infections, obviously we have an interest in wound infections from burns or, you know, exposure to cutaneous mustard gas and things like that. And I think from everything we've seen in our burn program, those endpoints are quite reproducible and definable. So I think you're there. I did want to add on, people were talking about a clinical trial network earlier, and we did a lot of work on that and looking at, you know, utilizing a common clinical or control arm. And we actually did a cost analysis of what it would cost and went out to industry and got a bunch of quotes back. And it was basically to maintain the infrastructure, it was going to be about $50 to $75 million a year. And when we engaged industry about whether or not they would be interested in participating in this network, we got a, maybe, you know, maybe you guys can go do 2 or 3 trials and we'll see how that works. And then if it works out okay, we'll come back. And so from a funder or a government perspective, what worries us is that, you know, $75 million a year is an expensive white elephant. And if people aren't going to use it, then we're in a situation where we're married to that infrastructure and we've got to deal with it. And the comment I think Vance made was, you know, well, we can just make people that if they want BARDA money, they got to use the network. Well, there's intention and reality, and that's nobly intended, and I see that point. But in reality, I would have every industry organization, congressmen and lobbyists lined up around the block demanding why BARDA was holding industry hostage, you know, to making them use their network that was government supported and maybe less efficient than the private sector. And that's just the reality. So, so I think the network is a good idea. We of course went down that path because we see that there's a lot of cost savings that potentially could be generated from such a network. But again, I think it's going to involve regulators, industry, and funders coming together to decide what makes sense in terms of a network like that to facilitate these type of trials, and then also coming together to really figure out a pathway that's commercially viable and feasible and doesn't, you know, make even the public funders reluctant to jump all in and support going forward, or I think we're going to be in a world of hurt.
Yeah, great. Any reaction from the presenters or follow-ups to the comments you just heard?
I'll comment on one thing Joe said, which is if you do identify high-risk patients, right, you can get an order of magnitude up from the 2.4% infection rate that John Rex pointed out was the Pfizer for the staph vaccine. And if you get there, right, into the, let's say, 25 to 30% risk of progressing to pneumonia, then the total size for our phase 2 is 354 patients, 175 more or less in each group. You wouldn't expect if, in fact, that demonstrates a 50% more reduction in the occurrence of Staph aureus pneumonia, the phase 3 to be a much different size, unless we see, and I'm looking at Ed while I'm saying this, unless we see a safety signal that would require a larger one. Because again, it's a single pathogen, it's a targeted therapy. We wouldn't imagine having to go up an order of magnitude in terms of the sample size for the study if, in fact, 300, 400 patients can demonstrate efficacy.
And I would agree with that because on the recurrent C. diff side, the people are self-selecting as far as who's going to be highly likely to get another infection. So what we see is once they've had a first recurrence, they're 50% likely to have another one. So that really narrows down from all C. diff patients to those that are really at significant risk. Those people are really expensive to treat. It affects their lives. They're many times homebound, many times on chronic antibiotic therapy. They can't go to school, they can't work anymore. And a therapy such as we're investigating, we've shown can have a 55% reduction in that recurrence rate. And those people are so grateful. I mean, they're thrilled to have their lives back. So not only can we save money, but we can really, and I agree with you, our phase 2 was small. It was 127 patients. The second one was 250. And our phase 3 trial is right around that same size. So we've been collecting safety data throughout because the question is, how do you gauge safety? And that's been really more the driving factor than the efficacy rates.
A lot was discussed in previous sessions around measures of clinical benefit and meaningful endpoints. Curious comments on, particularly for these products, are a little bit more preventive in nature. Enough evidence for providers to use and to adopt their use, and any concerns or comments on that?
Well, I can tell you something kind of odd that's happened, just kind of an ironic twist. The FDA has been very helpful to us in defining our program. And so helpful, as a matter of fact, physicians said, no, wait a minute, we don't think this should be a drug. We think it should be something we could use for our patients. So the FDA said, that's okay, we won't enforce it.
Yeah.
So we were marching forward on an IND pathway, and as an unintended consequence, another competitive company came forward, and they're selling tens of thousands of units that compete directly with the clinical trials we're trying to do. And so what we found over the last several years is that the enrollment rates have really gone down. We're struggling to even find patients, even though our sample size is small. So I think, in answer to your question, what we found is that there is an absolute demand for the type of product we're putting out. It's just that we're having a hard time getting the endpoint because of competitive forces. So I think once they're available, patients will drive a lot of it.
And I will say for our product, right, preventing a ventilator-associated pneumonia caused by any pathogen, it's clearly a good thing. It's hard to argue that, well, it might not be worth the cost because we can treat them successfully. We don't treat them successfully. 30% of patients still die. So you always want to prevent. That being said, we have noticed in terms of if you talk to KOLs or talk to investors, they do always ask the question, well, will we ever get to that paradigm where we prevent? Now, I will say, if you talk to intensivists, intensive care docs, they are thinking about preventing complications. They want their patients out of their ICUs, usually within 2 or 3 days, right? That is both for their health, and there's huge economics as well. It turns out those first 2 or 3 days are the most billable days. They don't want someone who spends an extra week on the ventilator because they get pneumonia. So they are wired a bit more for prevention. For us, it'll be about educating them to look for high-risk patients on a regular basis, treat them preemptively, and prevent the pneumonia, and then reap the benefits in addition to the patient reaping the benefit.
I might add on to that, whether intentionally or not, by choosing the disease processes that you have, ventilator-associated pneumonia and surgical site infection in particular, you've targeted some of the most aggressive kind of prevention-minded clinicians that are out there, and it's the surgeons that really push I think it's the ICU physicians that really pushed it when a cutting-edge technology comes around. They're the ones who are indeed advocating it, because I think you're right. I think they're the ones that have more of an eye towards prevention.
Well, I think-
It's not really a coincidence.
Yeah.
No.
Well done.
And I think a lot of the physicians in our case, they feel badly for the C. diff patients, but they run out of options, and all they want them to do is to go away. So what you find out is that those patients go from doctor to doctor to doctor to doctor because the doctor says, you know, I really don't want to treat you anymore because I have nothing else to give you. So we ended up starting as a treatment of last resort, and now are working our way up into more normal treatment regimes. But I think that's part of it, is the physicians themselves want to do something. They want to help people, and they don't always have the tools.
How are you feeling about payers as it relates to coverage and reimbursement? And does that rationale sort of extend to payers, you know, for the providers, maybe that because of the clinical areas that you've targeted are more of a slam dunk? How are you thinking about for the evidence needed for payers?
I'll give just a brief answer. I'm certainly no expert on that, and that's still a little bit out in our future. But again, we recognize some of the cost issues in terms of how expensive a surgical site infection can be. And so we're fortunate that the products we're making, we think we can produce at a fairly low cost. And so at a preliminary analysis, we think the numbers should work. But again, it's a little early for us to say.
We've already been talking to the payers and have done a number of pricing surveys as well as the benefits people. So we're pretty comfortable that this is a product that they'd be actually happy to cover because it really reduces their costs over the long run.
Yeah. Okay. I also want to, we touched on this a little bit, Deverick, but maybe expand a little bit more. The potential for these kinds of products to have a significant impact or enabling factor for more, stewardship of traditional antibiotics. Have you sort of engaged in conversations around these products specifically enabling that, or tell me your thoughts? Anyone?
Yes, you.
Anyone?
For me, well, one of the thoughts that occurred during some of the presentations, and maybe it's, to one of the themes that we've been trying to address today are, the measurable outcomes we're considering. It seems to me that thinking about not only preventing infections, but preventing the exposures to antibiotics are reasonable things to include in those assessments of these kinds of technologies. Now, again, I don't know how well that would be received, but it strikes me as something that would be an important part of preventing these infections.
And it certainly resonates with investors who, again, know about antimicrobial resistance and the desire and the spread of stewardship. I think numerically, it all depends on how widely used some of these products would be if it actually ended up being globally antibiotic sparing. But it can't be a bad thing to prevent infections that would require not just one sometimes, but multiple antibiotics in the hospital setting or the outpatient setting.
Okay. So let me pause here to see if we have any questions from the audience at all. Go ahead.
This is Jawad Aman from Integrated Biotherapeutics. So I have to say this is the first time I'm experiencing a prevention panel without the word V vaccine being even mentioned. So, I know there's a lot of, issues associated with testing a vaccine against an infectious disease. We've heard about all these, problems with Pfizer's clinical trial and difficulty of recruiting patients and getting enough power. But in a context like an ICU, and it's specifically the questions Dr. Anderson, because most of these vaccines really work by an anamnestic response, because people are exposed to these type of pathogens. If you have a vaccine where you can expect a response within about 10 days, do you think it has a place in an ICU to prevent same type of diseases like ventilator-associated pneumonia that now being targeted by Arsenis, for example? So what is your take on that?
Right. I think, again, part of the thought process would be if the end point is 10 days, it really strikes me as more relevant as to how quickly some form of protection is provided. If you look at the average length of stay, for example, in ICUs, my sense is it's shorter than 10 days. And so if you have to get to 10 days to have your effect, then I don't see that there's going to be value in that treatment. I don't know that, of course, I don't think that's how vaccines work. I think you're going to have some form of reaction and antibody production pretty quickly. And so does that lead to some form of protective effect even in the short term? I think I'd want to know more about that before I said that, well, maybe full effect is only at 10 days, but some effect before then, potentially there's value then.
I would just point out many of these patients have some level of immune compromise, which also would be a challenge for an active vaccine.
Let me address some of the folks that are dialed in online. Question, this is going to be to Dave. How would high Staph aureus burden be determined in practice in hospitals for the approach that you outlined?
Well, today, you can already do it with either semi-quantitative or quantitative methods in the micro lab. So all of the labs in our study, and we're doing our study at 65 sites around the world, they use the semi-quantitative plating method where you can look at essentially low, medium, high, and medium high counts as heavy colonization, and there's good correlation with quantitative methods. Now, we've talked about diagnostics, and certainly we would be interested in earlier diagnostics. And in fact, MedImmune, AstraZeneca is doing a study where they're taking the endotracheal aspirate and just looking for any level of colonization. And if it's Pseudomonas, they give a Pseudomonas antibody. If it's Staph aureus, they give an HLA-only antibody. And you can envision the field and progressing to a point where you do that kind of screening at ventilation or at entry to the ICU, and then you say, you know, this patient needs X, Y, and Z in order to protect them for their stay in the ICU.
Great. One more question from the web. This is to Lee. Very specific about your study when you mentioned that patients enrolled in your trial experienced recurrence with C. diff. The question was, what particular strain of C. diff? Do you have information on that?
We actually looked, and it was difficult to tell. Typically what happens when people get C. diff is the first thing that happens is they're slapped onto an antibiotic therapy. So by the time we see them in our clinical trial, we know they've had multiple recurrences. We remove them from their antibiotic therapy and then treat them a couple of days later, so that from the time they're tested till the time we treat them, generally they have very little C. diff left because they've been pre-treated in some ways with antibiotic therapy. So we haven't been able to identify any strains. Those that we have identified were all different kinds, and so there wasn't any pattern as to what I think the question might be getting at. Were there certain types of C. diff that this would work better against or not? And we weren't able to determine that from the lack of C. diff when we treated them.
Okay. Question over here.
My question is directed to Dave. Your approach is to prevent pneumonia in IOC patients. Have you thought about using this approach for prophylaxis in dialysis patients who also have a high risk of developing a MRSA infection?
We've certainly looked at other indications. What we tried to do for pneumonia is determine what toxins were staph producing for pneumonia. And these are the 6 that are relevant. As you know, staph produces a whole host of other toxins and virulence factors. And for bloodstream infections, which I think we'd be talking about, or even skin infections, we're looking at other models and whether these these toxins are the critical toxins and those would neutralize to prevent, or whether we'd have to look at an additional cocktail, additional antibodies. But we're certainly looking at that. And we'd be interested in expanding, as we progress with pneumonia, into indications where these 2 antibodies make sense. Right now, dialysis patients are not necessarily on our list, but other indications might be, for example, CF patients, where Staph aureus can lead as a leading cause of CF exacerbations. So there may be a role to play here, again, because these are the 6 toxins that play a role in the lung. But for dialysis patients, it might be a different set.
Great. Any other questions? Okay. We'll call that the last question, and then we'll move on.
Vu Trong, I have a question for Joe. It sounds like BARDA is expanding from biodefense type of pathogen into public health type of pathogen in recent years. Can you outline the thinking process? How do you define, what are some of the parameters you go through to decide whether or not you want to invest outside of biodefense type of pathogen?
Right. So, the current administration is very focused on national security, but we've framed antimicrobial resistance basically, as a consequence that can occur from being attacked with any of the threat agents that we deal with. If you're exposed to radiation and you lose your immune system, then your secondary bacterial infection is a probable thing. And if that's resistant, then we don't have antibiotics to treat it, then that's a problem. If we give 2 million people anthrax, or get exposed to anthrax and we have to give them Cipro for 60 days, then Clostridium difficile is a major issue. And so we'd like to have things to, if you can't survive the event, then you didn't, whether it's from a secondary thing or the primary thing, you didn't survive the event. So that's kind of the first point. The second point, when we go through and examine programs, I mean, we look comprehensively at their entire development program. We look at the company. We look at the people that they have on board. We look at, how realistic and feasible and cost kind of effective their development plan is. And we kind of evaluate that also against the programmatic priorities that we have. Those have tended to be recently, new antibiotics for gram-negative agents. That's where we thought the greatest unmet need is. Since we started the program, we've now advanced, 8 programs into phase 3 clinical development. Our first approval was this last year with Vabomere. We've got a couple more, we hope, pending. And so I think in, 2019, 2020, we're going to have a bunch of stuff to graduate out of the portfolio that were all precedented classes that kind of overcame normal mechanisms of resistance. And where I have imparted upon the team to try to go was, I've seen us become less risk tolerant over time and more conservative. And I don't know if that's because we had a luxury of things that we already were investing in versus when I was kind of building it and populating it, we were taking a little bit more chances. But this entire field of, collectively non-traditional stuff is an area where I think we need to be taking risks. And what, I think every time we get a proposal, that people come in and they are very critical of it, and especially if there's a lack of clarity on how to take the product from where it is and get it to market. And that's where I think collectively all of us need to work to try to obtain that clarity. So, we as public funders can adopt that kind of posture of additional risk tolerance.
Okay, go ahead.
Yeah, I just wanted to follow on a topic that's been brought up a couple of times today, and that's the diagnostics. I think these are very important for, of course, everyone in medicine, but for those of us who are concerned about prevention or interrupting an infection at an early stage, having surrogate markers that could help us do that, I think, is really quite important. And I'm reminded also, once a bit ago in my career, I was a vice president at Amgen, and work that had nothing to do with me. It was done by others before I got there. I'm reminded of Neupogen. Neupogen is one of biotech's greatest products. And what does it do? It really prevents infections. It's designed to grow your white cells back in cancer patients. Why do you want to grow back your white cells? To prevent infections. But it was approved by growing back the white cells, getting the neutrophils to come back. That was a product there where the surrogate marker took it the whole way. Not saying that it should all the time. I'm just saying that they can be very helpful in developing products. Thank you.
Okay, so that concludes this session. I want to thank the presenters and our panelists for a great discussion. Okay, we're going to roll right into our final session of the day. During this session, we'll ask our panelists to join me up on the stage here to tell us all what we learned. What were your major takeaways? What were important things that you all heard throughout the day that would be important to take forward? So joining me are Jonathan Darrow, faculty member at Harvard Medical School in the program on regulation, therapeutics, and law.
Scott Evans, Professor of Epidemiology and Biostatistics at the Milken Institute School of Public Health at George Washington University and the Director of the Biostatistics Center over there. Mark Gitzinger, CEO and founder of BioVeris, and John Rex is joining us again, too. Thank you.
Well, good afternoon. Jonathan Darrow. I came to this meeting this morning having substantial concerns about antimicrobial resistance, and following some of these fantastic presentations, I can say that I'm feeling a lot better. It's important to not become complacent, however, and remember that many products that are very promising in early stage development don't end up becoming approved products. And that's because of all of the challenges that they encounter along the way, some of which we've heard about today. The ones that stood out for me, number one, economics. That wasn't the topic today. We could easily spend a whole day talking about the economic challenges of developing antibiotic drugs. Instead, we're focusing on the scientific challenges, developmental challenges. There, I would say, one of the number one challenges is the small number of patients. It's hard to find patients, it's hard to identify patients, and that can have an impact on the financials as well once you get an approved drug. Rapid diagnostics, we're told, can help, but won't solve the problem. With respect to rapid diagnostics, one item to consider moving forward is the extent to which these should be considered as companion diagnostics, something the FDA defines as essential to the safe and effective use of a treatment. And that definition would seem to apply quite well in the area of antimicrobials. I know there's some disagreement here on the panel, but you'll hear more about that later. Second, is there was some concern expressed about superiority studies with respect to standard of care changing. If you're comparing your drug to the current standard of care, a new drug is approved, that changes that standard of care, that could upset your trial. That struck me as an interesting concern because if the chance is sufficiently high that the standard of care will change, that suggests that the pipeline is perhaps more robust than some of the discussions would lead you to believe. There are a number of other challenges I could mention, but I think I'd rather spend the time mentioning something else, which is I think it's important to recognize the level of flexibility and cooperativeness that the FDA has exhibited toward the development of antimicrobial products for decades. There are a number of programs that Congress or the FDA have initiated that have been used in this context. I happen to have a database that's based on FDA data. The Fast Track program, this is the Subpart E program that was mentioned earlier today, can allow approval on the basis of phase 2 trials without the need for phase 3 trials. Between 1987 and 2016, 38% of antimicrobials were approved under that program. Accelerated approval, introduced in 1992, allows approval on the basis of surrogate endpoints. Again, during that same period, 1987 to 2016, 18% of products were approved under that program in the antimicrobial space. Faster FDA review can be important. 61% of antimicrobial products received priority review by the FDA. Breakthrough therapy designation is newer. This was introduced in 2012, but there have already been at least 5 antimicrobials approved under that program. With respect to the rarity of drugs, that being a concern, it may have unique characteristics in the antimicrobial space, but this is not, small numbers of patients is not unique to that space. And 13% of antimicrobials have been approved with an orphan designation, again, between 1987 and 2016. Last thing I'll mention, the animal rule. Again, we're talking about significant flexibility that the FDA has applied in this context. We've had 7 animal rule approvals in the antimicrobial space. So with that, I think I'm probably at the end of my 5 minutes.
Oh, okay. Scott?
Thank you. So I'm a biostatistician, and
in statistics, we have a saying that there are lies, damn lies, and antibiotic development. You may have heard a different version of that, but actually, of course, I'm kidding, and I thank everyone for all their presentations, informative presentations today, and really fascinating discussion. I think the challenge facing us moving forward is trying to find the balance between acknowledging all of the practical challenges associated with this area in terms of diagnostics and enrolling patients and so forth, but not losing sort of the fundamentals and the rigors of randomized trials so that we can come out with valid conclusions as we move forward. So I'm going to make a few comments about various things we heard today. My first comment is more of an ethical dilemma. Early on, Dr. Rex mentioned that some of these trials, or many of these trials, are for thinking about future patients, and that's really a very different paradigm in many ways than we're used to dealing with. And I think that's very admirable in forward thinking to sort of think in those terms. But because it's very different, I think we also have a lot of work to do in thinking carefully about how to align this with patients that will participate in the trial. So you come to me and say, Scott, you've just been diagnosed with an infection. And we have this drug, it's a great drug, vancomycin. We think it can treat you fairly well. We could also enroll you in this trial, which might randomize you to vancomycin versus a new drug. Now, we don't believe the new drug necessarily will be any better. In matter of fact, the most optimistic people involved, the sponsors of this new drug, are hoping that it will be similar. So what is my motivation to participate in this trial? It seems like high risk and little reward. Now, if that's balanced with some other benefit for me, then it seems to be worth the risk. Now, whether that other aspect is safety or quality of life, or maybe it's something else, and maybe it's resistance. There was a discussion earlier about, could we start to figure out how do we measure an outcome of resistance, and that may be beneficial if resistance could be measured as an outcome. And we have a few surrogate-like things that we use for resistance, but it's a very difficult thing to measure, and I think it's worth us thinking about how would we measure resistance as an outcome, a resistance profile as an outcome. It's really a multidimensional scenario. But maybe with sort of building on what sort of resistant profiles are really threatening and valuing that as an outcome could be done. We've thought about doing this in sort of two-dimensional space. I have an outcome for a patient, and I look at a contrast between treatments, but then I have an outcome in terms of resistance, and I can look at the differences between two treatments in two-dimensional space. I can put a confidence region around it analogous to a confidence interval and try to give me a sense of whether I'm in a positive zone in sort of a global sense. So that's an interesting thing to think about, and one potential way forward. Now, for there was a theme today talking about what sort of outcomes should we be measuring. And of course, there's a lot of specificity with respect to certain trials and so forth. And one of the prep calls for this meeting, we discussed this on one of our calls. And now, typical outcomes and scenarios, we have the clinical cure, and you said, well, you investigated this 12, every trial uses a different definition. What is the definition of this? We've been sort of thinking critically about how to measure outcomes on patients. And we're thinking from sort of the clinical decision-making point of view that I want to know how a patient is doing. Now, if you measure a clinical cure, we talked a little bit about composites. Well, you're already using composites. Your clinical cure is a composite. It's got survival wrapped up in it. It's got some manifestation of whether symptoms are disappearing. It may have a micro component. You're already using composites. You've just got two levels of them. But it's not a very sensitive one. And if you're looking for sensitivity and worried about sample size, you want to be careful about that. Because suppose I have two patients that clinically fail. They fail. One patient dies. The other patient survives, clears their symptoms, but they have a micro problem. your outcome doesn't even distinguish between those two patients. And that would seem to be very critical, important to distinguish. So I can think about having ordinal types of outcomes that could distinguish that. And if you've got drugs that are having effects, you've got a measure that's much more sensitive than something binary that's combining patients who fail on a micro outcome and patients who die. So thinking about that, I think, is important. The other sort of aspect of this is the way we analyze things is not really very pragmatic or practical about how patients are doing. Now, there was a couple of presentations today, one that mentioned days in the ICU. Another mentioned ventilator days. You've got to be really careful about how to interpret a summary statistic of those parameters in the context of dying. You say, well, I got ventilator days or I got days in the ICU. Fewer days is better. Patient dies on day 2. Fastest way to me to reduce the number of days of ventilator days or days in the ICU is the faster the patient dies, the smaller the days. The only way I understand that statistic is when you link it to the patient's survival. Now, you can do that with composite endpoints. You start to tell me whether the patient died or whether they survived. If they survived, how long were they on a ventilator? I can build things like that that are sensitive enough to tell me how the patients are doing. And so that's something really to think about. What we're used to doing is analyzing each outcome separately. Let me give you an example. You treat 100 patients. I have an efficacy outcome. I have a safety outcome, all right? I give you the efficacy result, 50% efficacy. I give you the safety outcome, 50% toxicity. Let's suppose they're equally important. Do I have a good drug? Well, let me give you 2 scenarios. One scenario is the 50% of patients who had efficacy are the same patients who had toxicity. That particular case, nobody benefited. Worthless. And we give you a second case. The 50% who would have efficacy are different than the patients who had toxicity. Now we've got a great drug if I can find the right patients. If all I see is separate analysis of each outcome, I don't even see the difference. So we're taking patients in the trial and analyzing the outcomes. We're supposed to be taking the outcomes in the trial and telling you what happens to the patient. And it turns out that a lot of the problems we have start to dissipate if you think about it that way. So one other lack of pragmatism. You run your trial. You analyze efficacy, ITT population. I do my safety analysis, safety population. Then I say, well, let me combine those for some benefit-risk analysis. Who does that benefit-risk analysis apply? I'm not even analyzing them in the same population. I don't even know how to think about that. So now, with these more sensitive endpoints, let me give you an example about, so we've been using it because we think it's more pragmatic and practical about how patients feel, function, and survive. But you can potentially save on sample size. Suppose you got one of your new phages or one of your new drugs out there going up against colistin, right? Now, I think I can beat colistin on efficacy, but maybe I can beat colistin on safety as well, because colistin's got its own safety problems, right? So now when I look at things globally, my drug against colistin, and I start to factor in the fact that I'm also winning on safety, in a global sense, I've got big effects, but if I pan it out to just efficacy and safety in a separate dimension, the effects are smaller. But if I think about that in a strategic way, I can get more efficiency. So I'll move on. I wanted to make a couple of comments about trial networks. The NIH has known for years there's some efficiency there. Now, the question is, it's certainly not going to be cheap, but it may be cheaper than everybody running their own trial, and there's one-offs all over the world. And there's certainly some efficiency from both a standardization of protocol and processes, and you can do multi-arm trials, common control group, and so forth. And you do have to be very careful about how to construct them, because there's some dangerous versions out there that are being marketed. But there's some very good ones, and I think the one that Dr. Rex drew up is the right one, where there's still strong error control and sort of the highest rigor and fundamentals of randomized trials. We're just doing it with a common control group. There are some complexities, a lot of complexities that have to be worked through, including if you got multiple companies in the game, data access and who controls the data and who's looking at the data and when, if there's leaks of information that can jeopardize trials from an operational bias point of view. So that really has to be run with a tight ship. But there are examples. There's one in prostate cancer, for example, STAMPEDE trial, that essentially ran something very similar and is still ongoing and has been very successful. So there are examples where it's worked. There's another thing that might help us is a large umbrella screening process. So if you have an infrastructure by which you can screen patients, and at the time I'm screening them, they have clinical manifestations that I can observe, but I don't know yet pathogen identification or the resistance profile and so forth. Maybe I get that 3 days later. But when I get that information, I can be in a position where if patient A has a certain resistance profile and so forth, they might be eligible for trial over here. The next patient once I get their information, they might be eligible for a different trial. But there's an overarching umbrella that can redirect patients to trials that are looking for patients like them. And that's an a potential. The last thing I would like to make a point on is this particularly came up in the discussion of the phage, of some of the phage discussion about defining what the intervention is. In randomized trials, we tend to think of interventions as sort of physical things. There's a chemical that I came up with. It's got a chemical composition, and I'm going to apply it. But randomized trials actually evaluate strategies of use of interventions. And that strategy includes how it's made, how it's applied. You settle on a dose, you settle on how frequency of, and so forth. Now, what's happening with, say, adaptive phage is that you're going to be applying a strategy that says, I'm going to take a patient's sample, there's going to be some tailoring of the manufacturing of that phage, and then I'm going to apply it. But as long as you define what the strategy is for how you're going to manufacture that phage, how you come up with it, and how you're going to apply it, that's fine. Essentially, each patient is getting their own thing. So it's not well defined in that each patient's getting the same exact intervention, but the strategy that's being applied is the same. And so we have a paper about this coming out. It's called COMPASS, and the acronym stands for Comparing Personalized Antibiotic Strategies, and it's personalized. You're tailoring the treatment to an individual patient based on what they have. And that's fine. There's no issue with that. There's sort of an extra source of variation in how you come up with it, but actually what you're hoping for is you decrease variation in patient response at the end of the day, that everybody's going to be all right. I'll stop there.
Okay. Mark?
Yes, thank you for inviting me actually to the panel. I'm, as you mentioned, CEO of a Swiss biotech company working in AMR. So we're working on bacterial transcription regulators in the restore category and in the augmenting and preventing category. And besides that, I'm also the VP of the BEAM Alliance, which is a European alliance that represents 50 SMEs out of Europe that are all dedicated and working in the field of AMR. And it is very interesting to follow the discussion today that is happening here in the US with FDA, because we in Europe have something quite similar ongoing. We built actually a task force in the BEAM Alliance that is discussing with UCAST and with EMA on new endpoints and trial designs, on surrogate markers, and that are well beyond MIC for some of these new approaches. I think these discussions on both sides of the Atlantic are extremely important, and I'm glad that we're having them and that we can be part of them. What have we heard today, besides quite a lot of diversity and novel approaches, and also interesting trial designs and setups. I think, interestingly enough, when you really narrow it down today, most of the more advanced clinical trials have been still looking at very traditional endpoints, and probably this is in the nature of the beast, because it's the beaten track that we know, and that is more accepted also by funders. So I remain also with one of the comments that has been ongoing through the day, even if it's not the main topic, but the big elephant in the room still remains around pricing and how to make money out of these approaches, because otherwise we don't have funders to allow us to be more adventurous as well. I think some of the oncology field has shown that if there's enough funding and potential out there, you can go quite far off the beaten track and become more innovative. And part of this is missing in AMR for sure. The next step or comment I would like to make is sometimes it helps to create a vision. Where do we want to go in the future? And I'm not sure if we have heard enough about this today. So if we look at the prevention topic, I think it's very interesting what Dave has presented, for instance, was his approach. I think one of the visions could be in the future, because we actually are using antibiotics quite a lot today in terms of prevention. Some of them actually with a labeled indication, which came normally after long-term studies and potentially even meta-analysis and so forth. John can probably talk more about this. But the question is, can we create a vision where in the future we actually separate treatment agents completely from prevention agents with the reasoning of sustainable use? Because if you separate the two, your patient that will fail the prevention therapy and create still an infection has the full arsenal for treatment normally ready, because the resistance is not, there's no cross-resistance. So I think that could be one clear vision. And now the question, of course, comes, how do we study a completely new agent that has not been studied before for treatment, etc., in this prevention? And I think one of the examples from Dave's company, from Asanis, is very nice in this regard. The other point that John brought up in the very beginning is part of the vision is that we are creating treatments for the patient of the future. So when we have very high levels of resistance, etc. Now we're creating something that we cannot really study on today's patients by definition with this. So here's the question: how can we solve this problem, and how can we create something that has a benefit and someone would potentially pay for having it, despite we're not using it at the moment? I'm going to come to some of the ideas for this later. If we look at these visions and how can we now create potentially trials and endpoints that study these, I think one part that definitely helps is, and Scott brought a couple of nice examples, are composite endpoints, where we use them already and where we can potentially expand on them and learn, besides the non-inferiority and treatment outcome endpoints that we have, can we define safety? Can we define also, I think that was very interesting of the presentation from Lee, can we define in the microbiome what she called the microbiome health index? Can we look much more into these kind of endpoints or outcomes where the microbiome and the flora is not affected, where maybe we have thus also less resistance creation in the microbiome, less dissemination of resistance in these kind of endpoints, which is more an endpoint for society rather than for the single patient. Another thing is, can we look at biofilm creation, build-up, etc., because we know that some antibiotics actually push bacteria into biofilm formation. So I think that is an interesting point that we could look at. How to study them more. I think the microbiome health index, I would be interested to discuss more with Lee about it. We are at the beginning of understanding what that actually means for the patients. You know, what is a healthy microbiome, and how can we compare it between different patients, etc. So I think this is a very interesting part that we can look at. Secondly, we heard a lot about clinical trial networks, and Scott actually mentioned something I also wanted to point out is the STAMPEDE trial that I only learned about actually a week ago. I think it's in prostate cancer patients, which is a really interesting trial design that is, running since several years, still ongoing, multiple companies involved, and always has a constant control group, but still the control group is adapted, I think, in time how they read it, how they make the readout. So I think where can this actually be useful in AMR? I go one step beyond the STAMPEDE, beyond the trial design per se, and I'm going towards other trial networks that exist, for instance, in HIV and TB area, where they go to also really adverse settings, if you want to, in terms of the countries where these trials are run. But I think that the ACTG from NIH, the TBTC, and in Europe, the Panacea trial consortia are actually quite successful in recruiting patients at high-quality centers and so forth, and building high-quality centers in these endemic countries for tuberculosis. This could actually help to mimic a bit more in our trials the patient of the future, because we have a higher incidence of MDR and XDR and potentially enrich samples in these kind of patient groups. So I think in terms of recruitment, cost for sponsors, and hopefully also in terms of enriching special patient groups, these trial networks will be of tremendous benefit if that is possible to create. And lastly, I would like to mention something that, I'm actually surprised that no one picked it up so far. Kevin said that in the very beginning is, can we even try to make human challenge models? I think that's quite provocative. But beyond this most provocative step, can we look, and I think this is also something that we as sponsors and developers have to really look at very creatively, is in which indications do we actually still have these unmet medical needs? Can we look at more chronic infections? CF is obviously one example. Can we look at infections where there's a lot of relapse rates, etc.? And can we try to study our new approaches significantly more efficient in these kind of indications? And lastly, can we also look at non-life-threatening indications where we probably don't show for the payer and for a high price an outcome that will immediately make everyone jump on the approach, because now we're changing the world for life-threatening indication, but at least we could show outcomes that build a bridge in an ethical way to study them against placebo, etc., and then create or work towards the armamentarium that is diverse in the future for the patient of the future, having diverse approaches to overcome resistant cases, etc. I think, that was more or less my take-home messages today and some of the thoughts to the topic.
Great, thanks, Mark. John?
Fascinating to listen to all this, and let me again thank the organizers for the opportunity to be here today, and for all of you for hanging on to the bitter end.
And in the basement of the hotel all day long.
It is the basement of the hotel. Well, but this is an important conversation, and we started off the day by saying that some of us have been thinking about this hard for a long, long time, and have desperately wanted to figure out a way to go forward. But it has to be a way forward that that's clear enough to be meaningful to everybody, and wishing won't make this go away. So my takeaways, first, I'm not sure I even know anymore what a non-traditional is. I'll just be really honest about that. I like Dave Mantis saying that to call about antitoxin non-traditional, give me a break. That's actually where it all got started. Well, yeah, you're right. And so it was something that I was thinking about a bit before the meeting. But the various archetypes and all these other things they all apply equally to whether it looks like a standard small molecule or not. By and large, there's one or two corners of it where it's much more divergent. That would be the places where you're clearly working at the host level. That's not something we have traditionally done a lot, though you could call an antibody working from the host level side. I mean, you could call it that. So, and the reason I bring that up is I want us to continue to talk about some of these issues, but I think we should keep in mind that some of them can be generalized to any novel antimicrobial agent, whether it's a large molecule, a small molecule, or a surgical gel, which I also had not known about before, so that was fun to hear about. So with that, sort of two broad directions for comments. One is, it's interesting to see the number of companies that are progressing. Yes, there are challenges, but it's interesting to see how many companies have made a serious foray into phase 2 and phase 3. And the places where they have solved problems are useful lessons for everybody who's coming forward behind them. There's still a lot of work to be done. This is where I get to my list of key challenges. I'll say in advance, these apply equally to traditional and non-traditional products, I think. Cost of goods tends to come up a lot, and that does tend to apply a bit more to, I'm going to call it the a large molecule tends to just be more expensive to make than a small molecule. It's just generally is true. Everybody needs efficient clinical trials, and here I heard conversations that went in two directions. One was that there, I guess it's personally my bias, I think there is still a place for very efficiently delivering the core clinical comparison against a standard drug if you can do one of those. And that could even be a place where a virulence modifier comes into the study. You know, I'm doing a study of, you know, complicated UTI and everything is randomized versus meropenem. And if you'd like your compound to come in and be meropenem plus your virulence inhibitor, you can do that. You're going to need to do that study at some point anyway, and this kind of a program would get you done faster. At least that's the theory. The other direction I heard was about chasing down rare pathogens, and I just want to caution everybody about how hard it is to chase down rare and highly resistant pathogens. It's like chasing the phantom menace. And the best analogy I've got for that is that if you start saying, I want to build a network focused only on XDR pathogens, you quickly run into the XDR Center of Excellence problem, which is there's not any hospital in the world that wants to have a billboard out in front of it that says, come to our hospital. We have the world's most resistant bacteria, but don't worry, we got a drug. No, that's not really very attractive. So one of the things that having a good quality, efficient base trial network would do is let you as a developer spend less money on that and more money going out and playing Go Fish to look for the difficult circumstances, because difficult circumstances tend to, they're slow, small trials. You really have to work hard to find the patients. And, you know, it's going to take some effort, and if you can free up resources from another part of your program, maybe that enables you to do that. Just sort of a thought. The third gap that's common to everybody is payer gaps, and it's been said by several of us today that we have to solve the payer problem, and, you know, cross your fingers, there's going to be some hope for this, but if we don't solve that, we're all in trouble. And I want to end on the notion of, I want to call it different, more sensitive alternative endpoints. And this is one that stands out for lots of reasons. We currently tend to use very simple dichotomous endpoints that squash out a lot of data, and Scott made that really clear in his comments. You cook it down to dead or alive at 42 days. A whole lot of information that's lost in that very, very clear, robust, easy to measure endpoint, but it misses out a lot of stuff, and it also isn't helpful at all in the diseases that are less life-threatening, where the outcome is more like how many surgeries you had to have. You know, that's an important endpoint as well. But we struggle to measure that, I think. So some work on the question of non-mortality endpoints. And there has been a lot of work on that. There are a number of standard infections now where you can use a non-mortality endpoint. I tried to make a list of what those were. Complicated UTI, uncomplicated UTI, community-acquired bacterial pneumonia, skin structure infection. All of those have non-mortality clinical-like endpoints. They really do. We still haven't come up with a good alternative in nosocomial pneumonia. It's tricky there. But we actually, we do have clinical endpoints for a lot of these things, but I won't say they're terribly sensitive, and they may not be very useful measures over time. I think one of the other things we lose is any sense of response over time, and getting better sooner is advantageous, provided you can believe that you've actually measured it in a strong and compelling way. And then the third category of new endpoints is this idea of community-level endpoints. And here, we've heard several presentations about carriage of resistance. I enjoyed Lee's comments about changing the microbiome and thinking about carriage of resistant pathogens. And that it ought to be true that that's relevant. I mean, it has to be true that I can't come down with salmonellosis if I'm not carrying salmonella. I mean, it just has to be true. So clearing me of salmonella has got to be, for me, a good thing. And it's probably also a good thing for my neighbor. I'm not excreting it. And so that has to be valuable at some level. And we do use some ideas like that. I was trying to make a list of the places where I thought we used them. The one that was mentioned early in the day was we have used the surrogate endpoint of precancerous changes with the HPV vaccine. And we've actually approved an agent on that, and people are using that widely. But we also have some places where we use carriage of non-commensal. So in gonorrhea, one of your endpoints is clearance of the organism. And if it's gone from your urinary tract, it's gone. And you can't get gonorrhea again, at least from that one. You can always pick it up again, I suppose. But, and you can't transmit it again, because it's gone. So it feels like there ought to be a way to back into this, saying that if I don't acquire MRSA or you clear me of MRSA, that is helpful because I can't now transmit it. But there are a bunch of issues there about how do you know I've cleared it, how many body sites do you have to sample, and so forth. And finally, there's the ethics then of community-level endpoints. And several of us have commented on this. What is when you study a good clinical practice, when you get trained in good clinical practice, one of the things you learn is you're reminded of the Declaration of Helsinki and its extensions that say that you can't be doing science without paying attention to the individuals you're doing them on. And you're not allowed to do an experiment that benefits, that sort of goes beyond you always have to consider the individual first. And so, true, but as an individual, I also am benefited by the fact that Mark's not carrying MRSA. I mean, it's got to be a good thing, right? So how do we approach that, and can we approach that for some of these tools? It's just a fascinating thing to think about. So I've enjoyed the day very much. I think I'm more confused in some ways than I was when I started, but I do appreciate everybody making the effort to think out loud. And the companies that came and shared your stuff, thank you. It was really fun to hear that variety of techniques and concepts being applied.
Thanks, John, for those comments, and everyone. Everyone went through all of my questions that I had. So
there we go.
I think we're done with this session. We have 5 minutes to 5. I am going to wrap us up, though, and we covered a tremendous amount of ground here. And I'd like to thank all of you for sticking with us 3 levels down in this hotel. One thing that I came away with is I'm just blown away by the amount of technology diversity and the science that's going into all of this, and the different novel approaches to treating and preventing and making existing antibiotics better is just extraordinary. We talked a lot about many different important things today, one of which how flexible and supportive FDA has been, both on the CDER side and on the CBER side, at trying to figure this out and trying to understand what some of the scientific and developmental challenges might be with these more non-traditional approaches. So with that, we've talked, and I'm not going to repeat what but the main points have been what to do about small patient sizes and trial design and the opportunity for network trials. A lot of discussion was on clinical benefit and showing the effect size of, particularly in the superiority studies that are trying to show a demonstrated benefit above and beyond standard of care. And we talked a lot about meaningful clinical endpoints and how that can be challenging with the range of technologies that we've been talking about, but how that also impacts the provider and health system and payers' views of these technologies. We talked a bit about diagnostics and rapid diagnostics and the role that they can play, and some on funding, not just funding for the pool or the push incentives, which BARDA and CARB-X are really out there doing a lot, but also on the need, although this clearly wasn't the topic and we purposefully left this out, but it kept coming back, the need for pool incentives and how not just the push in the pool, but making progress toward addressing some of these scientific challenges. All of these things need to come together if we're going to help the field come through with more innovative ways to treat and prevent infections. So with that, I'd like to thank all of you for joining us, your thoughtful contributions, for all of our panelists and presenters, and all of you for asking really good questions and engaging with us throughout the day. Before we leave, I'd like to thank our colleagues at FDA who helped make today's event possible and helped us along, Ed Cox, Samantha Nambiar, and Scott Stibbets. As well, and especially as well as Kevin Outerson and John Rex for all of your help throughout the years, but in leading up to this event, your contributions were tremendous. And then lastly, I'd like to acknowledge the individuals and staff at the Duke-Margolis Center who helped plan this event: Morgan Romine, Nicholas Harrison, Elizabeth Murphy, Sarah Saprisi, and especially Monica Schneider, who put a lot into this. So thanks to all of you. Good, safe travels back home, and we'll see you soon.